कर रहे हो? ಏರ್ಟೆಲ್ ಕೈ ಕೊಟ್ಟಕೊಂಡು
ಬೇರೆದ್ ಮಾಡ್ದೈತೇನೆ ಕನೆಕ್ಷನ್. ಯಾವ್ದು ಮಾಡಿದೆ?
ಮಾಡಿಲ್ಲ ಇನ್ನು.
5G ಮಾಡು. ಲೈವ್ ಹೋಗಿದ್ಯಾ? ಏನ್ ಬರ್ತಿರುತ್ತೆ ನಾನು ಲೈವರಿಗೆ ಈಗ?
ಬಂತು. So hello students, I'm audible to you all. आवाज आ रहा है? थोड़ा नेटवर्क का इशू है. Just confirm me once if I'm audible. अगर मैं audible हूँ तो बताना. Yes, good evening. Yes. तो जल्दी फटाफट जान कर लो. कितने दिन से पूछ रहे थे कि मैराथन कब आएगा. फाइनली मैराथन लेके आ गए हैं. So we are back with a marathon for the plant sciences, जो सबसे बड़ा सब्जेक्ट है और सबसे इजीएस्ट सब्जेक्ट है, उसके लिए हम मैराथन लेकर आ गए हैं. तो वी विल बी कंप्लीटिंग द मैराथन फॉर बोथ जेनेटिक्स एंड प्लांट ब्रीडिंग. तो हालांकि हम ब्रीडिंग से स्टार्ट कर रहे हैं जो बहुत ही इजी है, तो वहां से स्टार्ट करके उसके बाद जेनेटिक्स कवर करेंगे. वीडियो प्रॉपर है, परफेक्ट है, ऑडियो एंड वीडियो दोनों क्लियर है. So sound is coming but doesn't feel the same on the YouTube. ठीक है, एक मिनट में कंफर्म कर लेता हूँ. ऑल क्लियर है. हाउ मेनी क्लासेस ब्रीडिंग? ठीक है, ब्रीडिंग का जो भी क्वेरीज है तो अभिषेक आके बता देगा. सो आई एम हियर टू जस्ट अपडेट, क्विक अपडेट देने के लिए आया हूँ. तो 6 बजे एक वीडियो आने वाला था, लाइक वी वर इन द प्लानिंग टू पब्लिश अ वीडियो एट 6 PM, जिसके लिए मैंने सुबह क्लास में भी बताया था. आई डोंट नो लाइक कितने लोग याद रखे हो, कितने लोग भूल गए हो, बट यस. सो द वीडियो विल बी लाइव बाय 9 PM तक वो वीडियो लाइव हो जाएगी, अपलोड हो जाएगी. तब तक तुम लोगों का मैराथन भी खत्म हो जाएगा. बीच में मैराथन के बीच में हो सकता है. So as I told you already, like what you have to do instantly after the publishing the video publish होने के बाद मैंने already बता दिया कि क्या करना है. Just giving a simple reminder, quick reminder दे रहा हूँ सभी लोगों को, सभी स्टूडेंट को, सभी प्यारे बच्चों को जिन्होंने एग्री एडिक्ट से पढ़ा है और यहाँ से बहुत अच्छा कंटेंट लिए हो. तो आपके लिए एक रिक्वेस्ट है कि जैसे ही वीडियो आएगा, लाइक आई वांट योर क्विक फीडबैक ऑन द वीडियो. ठीक है, यस. तो स्टार्ट करते हैं. बाकी जो ब्रीडिंग का जितना भी कंटेंट है और जितना भी प्लान है ये अभिषेक सर बता देंगे. और हाँ, इसके साथ साथ मैं जेनेटिक्स का न्यूमेरिकल्स पहले खत्म करूंगा, उसके बाद मैं जेनेटिक्स का मैराथन लूंगा और उसके बाद सीड साइंस का मैराथन लूंगा. ठीक है, तो कोशिश करेंगे कि 20 तक सब कुछ खत्म हो जाए. ठीक है, ओके फाइन. ओके, आ जाओ मिनटो, स्टार्ट करो. So what to do after the video is uploaded? Once the video is uploaded, I want every one of you to comment on that video, share your experience with Agri Addict. ठीक है, आपका सिंपल एक्सपीरियंस शेयर करना and just comment anything on the video, anything that is lively. Which video? So आज एक वीडियो अपलोड होने वाला है Agri Addict से. कुछ सरप्राइज है, कुछ ऐसे कुछ नया, कुछ चीजें हैं जो मैंने बताया है. And if I have to give some hint, like we have conveyed about the journey of Agri Addict. जो स्टोरी है Agri Addict का पूरा बताया मैंने उस वीडियो में. मेरे खुद का जर्नी मैंने शेयर किया है, पूरा 1-2 घंटे का वीडियो है. तो आई वांट यू टू पुश इट, कमेंट, शेयर, लाइक एंड सपोर्ट Agri Addict प्लेटफॉर्म, बिकॉज बहुत मेहनत लगा के बनाया है वीडियो को. ठीक है.
हेलो, गुड इवनिंग एवरीवन. सबको वेलकम मेरा क्लास में. बताओ, कमेंट्स में लगा दो सारे, सब शेयर कर दो वीडियो. अभी व्यूज तो कम आ रहा है यार, शेयर कर दो ना. मैं ऑलरेडी बताया था, मुझे मतलब बूस्ट कहाँ से आएगा? व्यू से आएगा. मैं डायरेक्ट बता रहा हूँ. गुड इवनिंग. ठीक है, हेलो या हरिप्रसाद बता रहा था ब्रीडिंग तो बहुत इजी है। इजी है क्या? इजी है क्या ब्रीडिंग? एक सवाल है, सिंपल का सवाल। ब्रीडिंग, प्लांट ब्रीडिंग इजी है क्या? मैं पूछ रहा हूँ, कमेंट में बताओ। 86 और थोड़ा पुश कर दो, सबको शेयर कर दो, ठीक है। अभिषेक सर मेड इट इजी है रे। वो तो थैंक यू, इट्स इजी व्हेन यू आर हियर सर। Explanation make it easy. Yes sir, with Abhishek sir, if you read it then it is easy. Yes. This is what I wanted. I do our best sir. Easy your, yeah Rimi, your reading has made it easier. The thing is when it will be easy, that to plant breeding. Okay, मैं genetics के बारे में बात नहीं कर रहा है इधर, plant breeding के बारे में बात कर रहा है। When you understand the concepts, when you understand the basics, ठीक है, सब easy होता है। If you know the real background, okay, and the basic of that particular topic, then plant breeding is very easy. If you don't know, actually ना मैं 2 दिन के पहले एक स्टूडेंट्स के साथ इंटरेक्ट कर रहा था, ठीक है। When okay he or she don't know the basic concepts, then they are unable to answer the easiest questions. But if you know the concepts very well, then you can easily answer MSc level, PhD level क्वेश्चंस आल्सो, ठीक है। ठीक है, हम ये मैराथन में मैं 3 पार्ट बना लिया है मैराथन, ठीक है। पहला पार्ट में आपका सारे बेसिक्स कवर हो जाएगा, दूसरा पार्ट में आपका ब्रीडिंग मेथड्स, ठीक है, और प्रिंसिपल्स एंड मेथड्स कवर हो जाएगा सारे सेल्फ एंड क्लास क्रॉप्स में, ठीक है। और थोड़ा स्पेशल ब्रीडिंग में भी वो सेकंड पार्ट में इंक्लूड किया है। और थर्ड पार्ट में, फाइनल पार्ट में आपका सारे बायोटिक स्ट्रेस, एबायोटिक स्ट्रेस, क्वालिटी, ठीक है, और IPR सारे बचा हुआ कंटेंट है ना, उसमें थर्ड पार्ट में कवर करेंगे, ठीक है। 3 पार्ट में हम मैराथन कर रहा है प्लांट ब्रीडिंग के लिए, ठीक है। 4-4 घंटे का सेशन रहेगा, ठीक है। इन 12 आवर्स वी विल कंप्लीट ईच एंड एवरीथिंग विथ रिलेटेड टू प्लांट ब्रीडिंग, द क्विक रिवीजन। ओके, रेडी है क्या? सारे रेडी है क्या? कमेंट में बताओ, हम चालू करेंगे आज का सेशन। बताओ, कमेंट में बताओ, और थोड़ा शेयर कर दो, शेयर भी कर दो। yes, total 3 classes. Nutan, Abhishek sir, hello to you. Yes, Inchan, yeah, welcome to all. Ratnesh, yes sir, good. Yeah. Okay then, yes, we will start today's session. Yeah, as you know guys, I am Abhishek. Okay, I completed my undergraduation from University of Agriculture Sciences, Bangalore. Okay, I got All India rank 21st in ICAR PG exam, All India rank 4th in ICAR PhD exam. Okay, I completed my masters from ICAR IRI New Delhi, currently pursuing my PhD from the same institute, ICAR IRI New Delhi. Okay, also cleared joint CSI UGC NET, ASR BNET in 2021-2022, and recently I visited Rome, okay, Italy, and in Larissa, Greece. Okay, I visited 2 countries for one of my PhD summer school. Okay, we will begin today's session. Okay, in this particular marathon, okay, what we are going to study, history of plant breeding, ठीक है। Domestication and germplasm conservation, next centers of origin, plant introduction, modes of reproduction, pollination in crop plants, ठीक है। उसके बाद आपका 2 mechanisms which will promote the cross pollination, that is self incompatibility and mere sterility, and also they are having some relevance in hybrid seed production. Okay, we will see, we will discuss what exactly these concepts, that too very quickly. Okay, we are not going to discuss very much deep. This is like a quick revision. Okay, as we are already covered each and everything in our master course. Okay, the one who are already took that our master course, for them it is very easy. It's like a revision, and for one who are not yet purchased our course, for them it will be somewhat informative. Okay, they will definitely get some good basic information. ठीक है, 87, अरे थोड़ा पुश कर दो, शेयर कर दो, इंफॉर्मेशन शेयर करना है, ठीक है। प्लांट ब्रीडिंग, what exactly is plant breeding? It is principles and methods needed to be favorably changed to the genetic makeup of a crop plants. Okay, we are using some principles, okay, based on the understanding of genetics of crop plants. Okay, we are having some principles of particular concept, then we will use some methods, okay, the basic breeding methods to develop, to change the genetic makeup of a crop plants. Okay, in turn we will get some good benefits like yield improvement, for disease improvement. Okay, there are some different traits which we have targeted to make them improve through the genetic change. Okay, we will call it as a plant breeding. Simple definition. Okay, इसके बाद it can also be described as plant evolution directed by man. Okay, where there is an involvement of man, okay, in the evolution of a crop plant. Okay, this is also plant breeding. मैं ऑलरेडी एक पूछा था, क्लास में भी पूछा था. Okay, plant breeding is an art or science. Okay, in the beginning years, okay, in the 19th era, 20th, the beginning of 20th century, 21st century, okay, it is like a mixture of arts and science. Okay, we needed some art, okay, the skill, okay, to select some plants based on the phenotypic observation, and it is also science. Okay, we need to understand the basic genetic principles. Okay, the law of dominance, law of segregation, law of independent assortment. Okay, these are the basic principles, okay, that will give the opportunities for the breeder of which are the plants to select. Okay, how the plants are evolving, that's a science also. But nowadays, okay, nowadays we are telling that it's not more than art. Okay, it's now it is not a art anymore. Okay, now it is completely purely science. Okay, I will tell you guys. Okay, we are now seeing the each and every basis of DNA. Okay, we are sequencing it, and we are seeing what is the responsible, okay, which is the sequence responsible for the particular trait. Okay, so it is now, it is not more not an art anymore. Okay, completely we can tell that it is a science now, plant breeding. Okay, if we start with the history of plant breeding, first it will come domestication phase. Okay, what exactly domestication? Process of bringing wild species under human management. एक plant है, for example in maize, teosinte, it is growing somewhere else in the hills, okay, hills of Mexican part. What people did, okay, they took that plants and they are tried to make it cultivable form. Okay, in the process of this period, okay, in the process of this particular generations, it converted, okay, it got evolved to cultivated maize. You can see here, okay, this is wild teosinte. You can see how the plant is looking like. There will be more branches and the seeds will be covered by some husk, or we can tell that it is covered by some odd case in case of maize. Okay, and more number of branches. But modern maize, you can see there will be a single cob. Okay, and your seeds are like without odd husk, odd case, and the single branch. Okay, there will be two genes. Okay, TB1, teosinte branched, and TGA1, teosinte gloom architecture. Okay, these are the two mutations happened in the evolution of maize. Okay, under domestication process. Okay, when from bringing wild species into human management. Okay, these are the two genes, okay, involved in the domestication of maize. Okay, you should remember. This is the domestication. This is the most basic method of plant breeding. ठीक है, क्वेश्चन ऐसे आता है. ठीक है, which is the most basic breeding method? Okay, which is the first step in plant breeding scientifically. Okay, and which is the first method, okay, where we can introduce some variation. ठीक है, इससे क्वेश्चन आता है, उसको आप ध्यान से क्वेश्चन देखना है. ठीक है, मोस्ट बेसिक मेथड विल बी योर डोमेस्टिकेशन. द फर्स्ट साइंटिफिक प्लांट ब्रीडिंग, द साइंटिफिक मेथड ऑफ प्लांट ब्रीडिंग. ओके, दैट विल बी योर सिलेक्शन. ठीक है, डोमिनेंट म्यूटेशन, व्हाट इज इट सर? अरे म्यूटेशन, ठीक है, म्यूटेशन होता है, ये तो रिसेसिव म्यूटेशन है. ये दो जीन्स है ना, TB1, you can see TB1, these are dominant, TGA1, these are dominant genes. Okay, what they will cause teosinte branched. When the TB1 gene present, there will be a more number of branches, and where there is a TGA1, there will be a odd casing in the seeds. When these two genes got mutated to recessive condition, they will lose their functions. Okay, once they lost their functions, okay, these are the two mutations, so that you will get single branch and seeds without odd casing. ठीक है, समझ गया ना नूतन? ठीक है, these are recessive mutation. in the process of domestication they got mutated. ठीक है, मैं क्या बता रहा था, मैं कुछ बता रहा था, हाँ, the most principal and scientific okay method of plant breeding, that will be your selection. ठीक है, अग्री भाई, व्हाट अबाउट ट्रिप्साकम सर, वो तो एक और अलग थियरी था मेज का, ठीक है, अभी हम उसको कंसिडर नहीं कर रहे हैं, ठीक है, थियरी अभी हम, दिस इज द सिंगल म्यूटेशन हैपेंड इन द मेज, ओके, द सिंगल macro mutation, we can also say the major mutation, okay, from teosinte, wild teosinte to modern maize. The tripsacum theory is another one, okay, which now it is failed, okay, we are not agreeing to that particular theory, okay, समझ गया ना, which is the most as I am telling, the most scientific and basic plant breeding method will be your selection. But if they ask most basic method of plant breeding, then it will be your domestication, okay, and the first method of plant breeding where you can introduce some variation, that is your plant introduction, ठीक है, domestication और selection में आप variation introduce नहीं कर सकते हैं, ठीक है, if they are asking where you can introduce some variation, okay, the first or the most basic method of plant breeding where you can introduce some variation, that is your plant introduction, ठीक है, समझ आ रहा है ना? Are you seriously in third year of your BSc? अरे छोड़ दो यार, ठीक है, सिंचन छोड़ दो, ये सब छोड़ दो, इधर कंसंट्रेट कर दो, कौन सा थर्ड ईयर है, कौन सा फोर्थ ईयर है, वो क्यों चाहिए, ठीक है, इधर आ जाओ, नेक्स्ट सिलेक्शन फेज, ठीक है, वाइल्ड पोटैटो था पहले, इट इज वेरी हाई टॉक्सिन, ओके, सोलानम स्टीनोटाटम, this is the wild potato in the Andes, origin of potato, okay, Andes mountains, okay, which will be in North America, which will come in the North America, okay, this is your North America, okay, your Andes will come here, this is the origin of potato in that particular region, Solanum stenotatum, it is wild potato, high toxin, okay, the bitter taste, okay, but course of evolution, okay, people try to select some potatoes which are less toxin, okay, Andes selection, we will also call it as a Andes selection, human selection and domestication, okay, it approximately happened 8,000 years before, okay, then there will be a genetic diversity, there will be so many land races, and you can see there will be a different color potato, and also sweet, large tuber, high yield, okay, and now we are introduced this crop to our India also. These are from North America, our potato, tobacco, okay, your maize, these are introduced crop from the North America. This is the selection phase. The second phase in the history of plant breeding, first will be domestication, without knowing you, okay, you are bringing wild species into human cultivation. Then selection, okay, the intervention of a men, okay, people in the selection process, okay, intervening of a human in the selection process. The example is your potato, okay, how it happened, this is your selection phase. Then you can see Patrick Scherf stated that only heritable variation respond to selection, and that natural variation arises through mutation and hybridization, okay, in the beginning of 18, okay, it is in 19th century, okay, Patrick Scherf will tell that only heritable portion respond to the selection. Then Wilmorin in 1856 developed the progeny test in sugar beet. By seeing only the parent plants, parent crops, you can't go for selection, okay, when there will be some plants, okay, mixture of plants, and it is giving some high sugar content in the sugar beet, okay, you will select easily. But in the upcoming generation, okay, if it is going for segregation, then in the progenies you may not get the high sugar content, so that the progeny test will be very, very important, okay, since the any phenotype, okay, any phenotype will contains your genotype and environment, okay, we don't know whether it is due to environment or it is due to genotype. To know that particular thing, okay, you will go for progeny selection, that means you will grow that particular parent in the next generation, then you will see if you are getting the same yield, then it is due to your genotype, otherwise it may be due to environment. So your Wilmorin, okay, he will give progeny selection in the sugar beet. Now we are following this progeny selection each and every plant breeding methods. Nielsen and Haley provided the genetic basis of individual plant selection. Okay, why you need to go for individual plant selection? Okay, not you will go for some out of 100 plants you will select some 10-20 plants and you will bulk and you will go for next generation. Okay, it's not like that. You will have to select individual plants, then you have to grow that particular progenies, then you will get some information whether it is due to genotype or whether it is due to environment. Okay, Nielsen and DNA. Next, William Johnson. Okay, the famous person, pure line theory. Okay, when this particular concept will come, okay, we'll discuss in self-pointed crops. Okay, in the methods of, okay, principles of self-pointed crop plants. Before that, what you should know, he proposed the pure line theory. What exactly pure line? It is nothing but progeny of single self-pollinated crop plant. Okay, he also told that your phenotype is nothing but it is due to genotype plus environment. Okay, in which crop he gave? In Phaseolus vulgaris. Okay, we will see exactly the pure line theory in the next marathon class. Next, hybridization phase. After your selection phase, next there will be hybridization phase in the history. Babylonians and Assyrian in 700 BC. Okay, what these two guys did? Okay, they pollinated date palm. What is the story? Date palm is dioecious plant. Okay, there will be separate male plants, there will be separate female plants. Okay, they saw some female plants and they are observing from 2 to 3 years. Okay, there will be no seed set in that particular female part. Okay, from a particular female date palm. Okay, they are very curious about it. Okay, why it's happening like that? Then what they did, they took some pollens. Okay, at that particular time they don't know about pollens. Okay, they took some flowers from the male date palm and they pollinated to the female date palm. Then only they got seeds. Okay, this is the one of the basic hybridization program. Okay, Babylonians and Assyrians in the 700 BC. Okay, where in Tigris and Euphrates region, that is your fertile crescent. Okay, what we will call it will come in the region of Turkey. Okay, in the Asia Minor region, in that particular region, they have did it. Then Thomas Fairchild in 1717. Okay, what they have did? They crossed Sweet William with Carnation. Okay, what is the scientific name of this particular Sweet William? Caryophyllus barbatus into Caryophyllus. Sorry. This is Fairchild mule. Okay. In 1717, Thomas Fairchild crossed with Sweet William and Carnation. Okay. Yes, barbatus with Caryophyllus. Barbatus with Dianthus barbatus into Dianthus Caryophyllus. He got Fairchild mule in 1717. You can see the original Fairchild mule, how it will look like. Okay, this is the famous hybridization program in 1717. Then your Knight. Okay, he extensively worked on fruit crops, hybridization in fruit crops. Then Colwriter in tobacco, he worked on hybridization in tobacco crop. Then your Eastern Shell. Okay, the famous geneticist. Okay, and corn breeders. Okay, they gave the phenomenon heterosis, hybrid vigor. Okay, and they have also published 3 back-to-back papers. Okay, it is the basic foundation for your heterosis breeding. Okay, whatever now you are seeing the hybrids. Okay, the principle is given by your Eastern Shell in 1914. Okay, the term heterosis given by Shell. Okay, the dominance hypothesis, over-dominance hypothesis. Okay, one by one. Okay, everything start to publish their works because of this particular works. from eastern shell in corn breeding. ठीक है, ये तो hybridization phase. You should remember. Fairchild mule, okay, between Dianthus barbatus and Dianthus caryophyllus. ठीक है, yes, O dominance theory, yes, good, yes, Preeti, Dianthus caryophyllus, yeah, that's what I told, okay. ठीक है, next phase your distant hybridization. In this class, okay, in this first some starting 20-30 slides, it is just we are introducing plant breeding, okay, the full overall concept, okay, whatever the history, okay, it started from the domestication phase from the particular time, okay, the present status, okay, we will see what exactly happened in the era of plant breeding. ठीक है, शेयर कर दो, सबको शेयर कर दो यार, थोड़ा बढ़ा दो, सबको पहुंचना चाहिए. ठीक है, what exactly I am going to tell in distant hybridization, okay, the beautiful example that is your triticale. ठीक है, ये आपका triticale. ठीक है, in 1890, okay, Rimpu, you should remember his name. ठीक है, Rimpu, what he did, he crossed tetraploid wheat or hexaploid wheat, that is your Triticum turgidum or Triticum aestivum into rye, that is your sickle cereal. ठीक है, now he produced triticale. ठीक है, in 1890. What about this? In 1928, okay, cross between your radish into cabbage, okay, by Karpanchako. ठीक है, in 1928 he crossed Raphina sativus into Brassica oleracea. What he thought, he will get the root of radish and head of cauliflower, but what happened, okay, it got failed and he got the reverse. In reverse case, he got roots of your cabbage, that is Brassica oleracea, and the leaves of radish. ठीक है, this is the one of the again failure. In this case, triticale, now also we are using this particular triticale for maintaining genetic diversity and also as a source of some disease resistance or biotic or abiotic stress resistance genes. Okay, these are two examples for your distant hybridization. There will be so many examples, okay, but these two are major examples in case of distant hybridization. Raphanobrassica intergenic, not successful, yes. Karpanchiko, vice versa, yes. Good, Ratnesh. Intergenic polyploid, roots of cabbage. ठीक है, next phase of novel genetic variation. ठीक है, distant hybridization का phase भी चला गया. Then phase of novel genetic variation, mutations. Okay, anyway we know about spontaneous mutation. There will be so many mutations which are happened in domestication phase also. You can see in the evolution of maize, in the evolution of wheat, in the evolution of cabbage, cauliflower. Okay, there will be so many mutations happened, but those are spontaneous. We don't have any hand in that particular case. This is induced mutations. Okay, Muller in 1927 he told that, and he used X-rays to induce mutation in Drosophila. Okay, he got Nobel Prize in 1946, I think, if I am correct. Okay, 1946 he used X-rays in Drosophila. Okay, immediately in 1928 Stadler used, okay, gamma rays in barley, in wheat, and in maize, in crop plants, okay, to induce some mutations, okay, which maybe we can take it as improvement. Okay, he treated this gamma rays in barley, maize, and wheat. Okay, this is the phase of mutation. Okay, now we are using mutations also. Now we started to use mutations, induced mutations. Next phase will be genetic engineering phase, as you already know. Okay, in 1994, what happened? Can anyone? Anyway, the research and everything started in 1970, 1975 era. But particularly in 1994, there will be some triggered achievement. Okay, the milestone happened in case of genetic engineering. Sir, X-ray is also there. But yeah, but majorly used gamma rays. Okay, in your mind, you can keep like this. Okay, you used particularly gamma rays in case of barley, wheat, and maize. अरे कोई बीटी काटन है क्या? अरे भाई, पर्टिकुलरली आई एम आस्किंग, मैं ऑलरेडी क्लास में भी बताया था, ठीक है? ये तो क्वेश्चन बार बार मतलब सब पेपर में आ रहा है बार बार फिर से. एटलीस्ट यू शुड नो दिस द मेजर अचीवमेंट्स. Okay, from where it started, the first variety in edible crops. कौन बताएगा? गिफ्ट मिलेगा आपको, पक्का गिफ्ट मिलेगा. रतन सिंह बुल्लर, गुड गुड. टोमेटो ना, फ्लेवर सेवर. अरे भाई, ये तो मैं PYQ डिस्कशन में बार बार बोल चुका है ऑलरेडी, ठीक है? फ्लेवर सेवर, एंटीसेंस DNA टेक्नोलॉजी, ओके, पॉलीगैलेक्टोनोज एंजाइम. जाके PYQ देखो एक बार सब लोग, ठीक है? फ्लेवर सेवर आया था 1994 में. उसके बाद इन इंडिया कॉटन, BT कॉटन इन 2002 गॉट अप्रूव्ड फ्रॉम GAC कमिटी, ठीक है? BT कॉटन, ये क्वेश्चन पक्का. अरे भाई, जेनेटिक इंजीनियरिंग फेज में एक तो क्वेश्चन पक्का आएगा, ठीक है? आपको सबको पता होना चाहिए क्या अचीवमेंट हुआ था, ठीक है? दिस इज इन केस ऑफ वर्ल्ड द फर्स्ट, इन केस ऑफ इंडिया द ओनली वन एंड ओनली BT कॉटन, ठीक है? अभी कोई और क्रॉप हम ग्रो नहीं कर रहा है कॉटन में, ठीक है? कॉलजिन कंपनी, गुड. रतन, अब हमारा कोर्स में है क्या? स्टूडेंट है क्या ग्रेड का? रतन सिंह बुल्लर, बताओ, जल्दी बताओ. यस गुड. एंटीसेंस RNA पूजा. रतन सिंह बुल्लर, नो. अरे भाई, आप एक काम करो, ठीक है? क्लास होने के बाद मेरा मतलब अग्रेड कुछ हमारा कॉन्टैक्ट सपोर्ट में आप मैसेज करो एक, ठीक है? आपको गिफ्ट तो पक्का मिलेगा. नेक्स्ट, स्मार्ट बिडिंग. नेक्स्ट पेज विल बी स्मार्ट बिडिंग, ठीक है? क्या एग्जैक्टली द स्मार्ट बिडिंग इज? ओके, वी विल आल्सो कॉल इट एज the varieties. Again one question for you guys. Okay, if anyone tells this, again they will also get some gift. Okay, the ultimate, the last gift means at the end of your preparation you will get some gift which will be useful for you guys. What message do you want to send, sir? अरे भाई, अग्रीडेट वेबसाइट में जाओ, आपको एक नंबर मिलेगा, उस नंबर पे आप एक मैसेज डाल दो बस, ठीक है? अभिषेक सर को कनेक्ट कर दो, ऐसे मैसेज डाल दो, मुझे वो मैसेज आएगा, ठीक है? स्मार्ट ब्रीडिंग बताओ. मैं एक क्वेश्चन पूछ रहा है इधर. स्मार्ट ब्रीडिंग, इसमें द वेराइटीज व्हिच वी विल डेवलप थ्रू द स्मार्ट ब्रीडिंग, ओके, व्हाट वी विल कॉल फॉर देम? देयर विल बी वन वर्ड, ओके, द वेराइटीज व्हिच वी आर डेवलपिंग through the smart breeding technology. Okay, again I will give some hint, ठीक है? Marker based selection. We are using smart breeding is nothing but we are using markers to select some traits without seeing the phenotypes. Okay, develops होता है ना variety, the variety which will develop through this particular smart breeding. Okay, अरे भाई, एक मिनट, मैं पूछ रहा हूँ, उसको एक नाम दिया है. We will call them with some name, the varieties which are developed through smart breeding. Mark-tested variety है तो वो तो generalized हो गया. कविता बट बता रहा है, कविता तो क्रिस्पर क्या सर है, छोड़ दो यार. क्रिस्पर सर, लव फ्रॉम एंटामोलॉजी स्टूडेंट्स. अरे भाई आप क्यों देख रहे हैं क्लास? ठीक है, आपको तो कुछ मिलेगा, कोई दिक्कत नहीं. मेरा क्वेश्चन है ना, द वेराइटीज विच वी विल डेवलप थ्रू स्मार्ट ब्रीडिंग, ओके, वी विल कॉल दोस वेराइटीज विथ सम नेम, विथ सम टर्म. अरे मिल गया. सुपर ऑर्गेनिक, दावा डोलमा. ओके, शी इज एनीवे आवर स्टूडेंट, आई थिंक. दावा, यू आर आवर स्टूडेंट, राइट? अरे मिल गया आंसर, तो मिल गया यार, छोड़ दो यार गणेश। टोबाटो प्रिसिशन ब्रीड वेराइटीज बाय इंजीनियर्ड क्रॉप्स, अरे इसको बताते हैं हम सुपर ऑर्गेनिक्स, मैं ऑलरेडी क्लास में बताया था, ठीक है। दावा यू आर, yes sir, okay, anyway you will also get some gift, yeah, anyway you are we are having your contact details, okay, you will get some gift। सुपर ऑर्गेनिक्स, अभी बताओ पहला वैरायटी कौन सा है? एचएचबी इम्प्रूव्ड, ओके, पर्ल मिलेट सारे लोग डाल दिया था, ठीक है। एचएचबी 67 इम्प्रूव्ड in 2005 and 2006, okay, for downy mildew resistance। Then your improved Pusa Basmati 1 XA13 XA21 for bacterial blight resistance in 2008, okay। Then there will be a in chickpea there will be super andigeri 1, okay, for Fusarium wilt resistance। नोट कर दो, सारे नोट कर दो, ठीक है, ये तो बहुत इंपॉर्टेंट है। और उसके बाद आ गया आपका improved Pusa Samba Masuri, improved Swarna 64, okay, there will be so many varieties, but initially your SHB improved 67 for downy mildew resistance and your improved Pusa Basmati 1 for bacterial blight resistance, okay, XA13 and XA21 gene, ठीक है, ये तो important है। It will, we will call that as a smart breeding phase, okay। After your genetic engineering there will be a boom in the smart breeding, okay, everyone they are using markers, okay, marker assisted breeding, marker assisted backcross breeding, marker assisted recurrent selection, okay, there will be era। Then precision breeding, okay, at this particular time now we are having at this particular phase precision breeding, okay, maybe your genomic selection, okay, genomic selection is a thing, but by using again markers, okay, again by using again you are using markers, you are selecting some better parents for the hybridization program। Another one is gene editing technology, your CRISPR, okay, this is also your precision breeding। Now we are in the era of gene editing, CRISPR-Cas9, okay, prime editing, there will be so many editing technologies, okay। Recently now we have released 2 varieties again, okay, we will discuss in the end of the this particular unit what are those 2 varieties, okay। This is all about your gene editing। Next, objectives of plant breeding, okay, what are the objectives, why we need to do plant breeding, okay, what are the different objectives? First one is your yield, okay, we should increase yield day by day, okay, like year by year। why we need to improve yield? अरे भाई हमारा growing area ना कम होता जा रहा है, ठीक है, the cultivable area कम होता जा रहा है, okay, so we need to increase yield, okay। In particularly in case of wheat, okay, in during 1960, do you know the million ton or how much we are producing at that particular time? 20 to 30 million ton, okay, the overall production। After this, after green revolution, at presently in 2023, as per my knowledge, we have reached 120 million ton in the wheat। area है तो decrease तो हो रहा है, ठीक है, or maybe there will be a equal area, but you can see the yield in case of wheat it got jumped from 30 to 120 million ton, you can see, okay, that's why the main objective is your yield। Then breeding for biotic stress for different insects, for different pests, okay, we are breeding for it। breeding for abiotic stress, breeding for abiotic stress, for drought, for heat stress, as I told in case of wheat now we are experiencing terminal heat stress, okay, during that March April period, okay, there will be so much climate is changing, okay, we are getting so much heat particularly in the March April, okay, so there will be less yield, okay, there will be a stress, okay, we are targeting that particular heat stress also nowadays, okay, it will comes in your abiotic stress। Then breeding for quality, quality is again the major thing, anyway now we are achieved with the yield in case of wheat, now at particular time we are getting 60 to 70 tons per hectare। now we are what we are targeting, now we are targeting zinc, now we are targeting iron to improve quality, okay। उसके बाद क्या आएगा आपका बताओ, यील्ड हो गया, बायोटिक स्ट्रेस हो गया, एबायोटिक स्ट्रेस हो गया, क्वालिटी हो गया. What are the different other objectives? Crop duration. Okay, you need to decrease the crop duration period. Okay, synchronized maturity, breeding for synchronization. Okay, there will be maturity uniformly and with the synchronized form with all the crop plants, all the individuals. Okay, tolerance. Anti-nutritional factors, good. Breeding for to reduce anti-nutritional factor, not to increase. Okay, to reduce anti-nutritional factors. मैं एक लिस्ट ऑलरेडी बताया था, ठीक है, और अब PYQ में भी है. We will discuss also. We will discuss in quality breeding. Okay, the list in which crops which are the anti-nutritional factors. Very important. Okay, they will definitely ask in your exams. Okay, वो भी देख लो एक बार. उसके बाद क्या आएगा? Yes, non-shittering for photo and thermo sensitive reduction. We'll also call it as Devarakonda. Okay, Devarakonda will also call it as photo and thermo insensitive. Okay, we need to improve for the insensitivity. Okay, there will be no impact of your photo period and thermal period. Okay, yes, seed dormancy, earliness. Okay, there will be so many objectives. Okay, basically your yield will be whatever other traits you are targeting, but you also maintain in all other traits. You should also maintain yield as your primary objective. ठीक है, इतना ब्रीडिंग तो आसान है भाई, कुछ नहीं है. ठीक है, अब तो थोड़ा दिमाग लगाना है, ब्रीड करना है बस. अभी बताओ, वेराइटी फॉर न्यू सीजन, गुड. I will tell you. Okay, Madhuri, good. Madhuri is telling one good objective. Variety for new season. क्या है variety for new season? क्या होता है? ठीक है, आपका राइस है ना, कहाँ ग्रो कर रहा, पहले कहाँ कल्टीवेशन हो रहा था, पहले ज्यादा. ओके, वो राइस. ओके, इन विच पार्ट ऑफ इंडिया वी आर ग्रोइंग वेरी लार्जली एंड द ट्रेडिशनली? कोई बता सकते हैं क्या द ट्रेडिशनल ग्रोइंग एरिया ऑफ योर राइस? अरे 103 हो गया व्यूज. हाँ, जबरदस्त. एक 8 लगा दो सारे. पहली बार हमारा सेशन 103 मतलब 100 के ऊपर चला गया है. सबको शेयर कर दो, सबको ये इंफॉर्मेटिव होना चाहिए बस. वेस्ट बंगाल, साउथ इंडिया, या इन द पार्ट ऑफ गोदावरी, तमिलनाडु. ओके, इफ इट इज, मैं तो उतना सही से नहीं कर रहा है, इंडिया को थोड़ा एडजस्ट करो. अरे ठीक है. ठीक है, तमिलनाडु. ओके, वेस्ट गोदावरी रीजन ऑफ आंध्रा, ओडिशा, वेस्ट बंगाल. ठीक है, एंड दिस इज सम ईस्टर्न पार्ट्स. दिस इज द मेजर ट्रेडिशनल ग्रोइंग एरिया ऑफ योर राइस. ठीक है, उसके बाद क्या हुआ अभी? बासमती पता है ना क्या हुआ अभी? ठीक है, यूर पंजाब, हरियाणा, उत्तर प्रदेश, जम्मू कश्मीर, उत्तराखंड, they are also now they are growing your rice crop. That's what we'll call rice is now new seasonal crop for Punjab and Haryana majorly. In Kharif season now they are also growing rice. Wheat का क्या हुआ? Wheat तो पता है ना आपको? Wheat is majorly Haryana, Punjab and Madhya Pradesh. This is the traditional growing area. But yes, good. सिंचन 7 states. अरे यार, आपको तो सब पता है सिंचन को. This is the wheat growing area traditionally, but now we are growing wheat in Bihar, UP and some parts of Jharkhand. You can see this what the new season crop is. ठीक है, समझ गए ना? Yes, बताओ, समझ गए ना? अचीवमेंट्स में जाएंगे हम लोग. समझ गए ना? बताओ सर, सारे बताओ. yes, good. ऐसे ही रहना, ठीक है। कौन कौन अटेंड किया है, लाइव सेशन अटेंड कर रहा है अभी? 100 के ऊपर है लोग। लास्ट तक जुड़े रहना, ठीक है। सारे रिवीजन हो जाएगा, ठीक है, सेल्फ इनकम्पैटिबिलिटी तक। एक ही क्लास में कभी भी, मतलब कहा भी आपको नहीं मिलेगा ऐसे कंटेंट। अचीवमेंट्स, फेजेस हो गया, ऑब्जेक्टिव्स हो गया, ठीक है, अचीवमेंट्स। अभी हम 14 स्लाइड में है, 14-15 स्लाइड में है। पता है स्लाइड्स कितना है? 100 है, ठीक है, 100 के ऊपर है। सब मिलेगा आज, सब रिवीजन हो जाएगा। अचीवमेंट्स ऑफ प्लांट ब्रीडिंग, okay, where we will, whenever we will talk about achievement, okay, in case of, in the scenario of Indian case, okay, we will always starts with semi-dwarf, okay, whether it may be in wheat or rice, okay, the green revolution phase. From that particular phase, as I told, okay, the yield increased, okay, 10%, 10% increase हुआ है, from 20-30 million ton to not 10%, around 5 to 6%. Okay, now we are in the 120 million ton in case of wheat. कैसे हुआ वो प्रोसेस? क्या हुआ था, जर्नी क्या है उसका? एवरीथिंग विल स्टार्टेड विथ सेमी ड्वार्फ wheat. श्योर सर, 100%। सिमिट मेक्सिको, अरे सिमिट का वो स्पेलिंग मिस्टेक डाल रहा है रत्नेश। आपको ये स्पेसिफिक चीज सही से याद होना चाहिए। मे बी दे कैन आस्क द फुल फॉर्म ऑफ योर सिमिट, ठीक है, एक बार जाके देखो सिमिट क्या होता है, उसका फुल फॉर्म सब आपको याद रहना चाहिए, ठीक है, कोई दिक्कत नहीं, आप एक बार जाके देखो। सिमिट एज गुड कोई डाला है अभी 94, 86, अरे अरे जा रहे, ये बहुत इंपॉर्टेंट स्लाइड्स है, 1, 2, 3, ठीक है, जुड़े रहो। सेमी-ड्वार्फ wheat क्या हुआ, कैसे? Okay, there will be a Gonzalo Inazuka. Okay, what he did, he crossed, okay, the native Japanese variety, that is Daruma. Okay, he is scientist from the Japan, okay, in that particular 1930. It is Japanese variety Daruma. He crossed with Fulds. It is American variety, American tall variety, but it is giving some good yield. Okay, now they are growing in Japan. Okay, Daruma is their own Japanese variety, which is very short in nature. Okay, then they, in the cross they get Daruma Fulds. For this progeny, they crossed with Turkey Red. Okay, now we got Narin 10 variety. This is the development of Narin 10. Okay, from Gonzalo Inazuka. When Salman, okay, he is some agronomist, okay, after World War II in 1945, at that particular time he visited Japan, okay, to see the crops, okay, after World War II, what happened to the different agroclimatic zones crops. He visited to Japan, okay, Salman, and he collected 16 varieties of wheat from the Japan, and he sent those particular varieties to Orville Vogel. Okay, your Salmon 16 varieties. Okay, he posted or he just gave it to Vogel. Okay, he is one of the scientists in US Department of Agriculture, Washington DC. Okay, Vogel. He gave these 16 varieties to Vogel. Then what he did? Okay, he crossed with particular winter varieties. Anyway, we know already what exactly winter wheat and spring wheat. If you don't know, if anyone don't know what exactly difference between winter wheat and spring wheat, you can just go to our first PYQ discussion in 2020 paper discussion. I have already explained it very well. Okay, Vogel will develop by using those 16 varieties, which also include your Narin 10. Okay, he crossed with some winter wheat varieties and developed gains. Okay, Vogel gains are in the background of winter wheat. ठीक है, हम क्या ग्रो करता है, हम लोग, हमारा पाकिस्तान लोग, हमारा इंडिया लोग, ठीक है, इंडिया एंड पाकिस्तान, हम कौन सा वैरायटी, कौन सा स्प्रिंग और विंटर व्हीट, मैं ऑलरेडी बताया था ये चीज, कौन सा वैरायटी हम लोग ग्रो करता है, स्प्रिंग व्हीट, गुड लकी पटेल, स्प्रिंग, ओके, बट ये डेवलप्ड इन द बैकग्राउंड ऑफ विंटर व्हीट, ठीक है, कौन, वोगेल, then these gains and the Norrington he received from Norman Borlaug in the CIMIT, okay, that is the Central Institute for Improvement of Wheat and Maize Crop, okay, the CJR Institute in Mexico. He received these gains and he transferred, okay, the dwarf trait into the spring wheat, okay, spring wheat, कौन किया ट्रांसफर स्प्रिंग व्हीट के लिए, आपका नॉर्मन बोरलॉग, okay, then he introduced those varieties to India as Sonara 64, okay, Lerma Rosa in India, ठीक है, Mexi Park variety, Mexi Park for Pakistan, okay, India and Pakistan are major wheat growing areas, okay, that's why they developed spring wheat in the background of the Sonara 64, Maharashan and Mexipac. They introduced to India in the 1960, 62, 64 during that years, okay, so this is what exactly happened in case of Green Revolution, okay, the journey of Norrington from Japan to USDA, then USDA to Mexico, then Mexico to India and Pakistan, ठीक है, समझ गए ना, Daruma into faults into Turkey red, Norrington, it is done by Gonzalo Inazuka, then Solomon, okay, when we visited to Japan, he collected 16 varieties of wheat which includes Norrington and he sent it to Vogel, we is working in USDA, and instead of Washington, he transferred those traits into winter wheat, okay, called them as gains, then gains will given to Norman Borlaug, he transferred to spring wheat, and we introduced Sonara 64 Larmarajo in India, Mexipac in Pakistan, clear है ना, अरे सोनालिका कल्याण सोनार secondary, okay, after we got this particular Sonara 64 and Larmaraja, we, as per our Indian condition, we hybridized or we selected some of the progenies from that particular parent varieties, now and then we released those are Sonara 64 and Kalyan Sona, ठीक है, ये तो secondary introduction. Norman Borlaug worked on many segregating generation, yes, there will be so many segregation generation, okay, there will be one particular cross, one particular targeted cross, then you got these varieties, ठीक है, then what exactly you are in Norrington, what are the genes responsible for semidwarfism, ये तो आपको नहीं पूछेगा मेरा हिसाब से, ठीक है, one who are preparing for PhD maybe they can target this particular part, but for you guys what you want to know, your RST gene is dominant mutation, okay, पहले RST ऐसे था, ठीक है, small RST 1 RST 2, ओके, इसलिए उसकी मतलब म्यूटेशन के बाद वो डोमिनेंट हो गया RST1 RST2, okay, these are dominant mutations in the green revolution, okay, not in case of rice it is recessive mutation, but in case of wheat this is dominant mutation, पहले ऐसे था, ठीक है, small RST, actually what the role of this particular RST genes, they will code for Della protein, what exactly your Della protein will do, it will inhibit the growth, okay, it will inhibit the particular GA genes so that you will get a tall plant, okay, you can see in the normal pathway when there is an RST genes, okay, RST B1A, RST D1A, or we can also tell small RST1, small RST2 in the recessive condition. Okay, they will bind to receptor, okay, the gibberellic acid receptors, okay, there will be a complex, then it will recognize DELA protein, okay. This particular complex, GA complex, will recognize when there is a normal genes, they will recognize DELA protein, okay. Then DELA protein is degraded by the proteasome. When this particular gibberellic acid will recognize normal DELA protein, okay, it will be degraded. Your DELA protein will be degraded. So there is no DELA protein now. Growth genes will be in the on condition. As I told, your DELA protein will inhibit the growth, but this particular DELA protein, which is coded by RST genes, or normal DELA protein, it will be recognized by normal GA and degraded by the proteasome. So growth genes will be on. In this particular original case, growth genes will be on. So you will get tall plant. ठीक है, समझ गए ना? Suppress gibberellic acid formation. Exactly. Okay, your DELA protein will suppress the growth genes. Okay, when they are in the activated condition, but the normal DELA protein will recognize by GA, the primary mechanism. They will recognize by GA and they will degrade it. So there will be no DELA protein, so your plant will grow. But in case of dominant mutation, the RST mutation, it will also code for DELA protein. But these DELA proteins are insensitive. There will be a mutation, right? So there will be some modification in DELA protein. So they will be insensitive to this particular complex, GA complex. So this GA will not recognize this particular mutated DELA protein. These are mutated DELA protein. Okay, they will not recognize. And your DELA proteins are free to act now. Okay, since they are not recognized by GA due to insensitivity, due to the mutation, okay, now they are active condition. Okay, they will suppress the growth. They will not degrade. So they will act on the growth genes. Okay, growth genes will be off now. Though there will be no growth genes, so there will be semi dwarf in the plants. ठीक है, आप क्या याद करना है इधर, एक simple, I will tell in simple language. ठीक है, your normal RST genes which are in recessive condition, okay, they will code for DELA protein. Okay, your DELA protein are normal wild type DELA protein. They will easily recognize by GA, okay, gibberellic acid complex. Then they will degrade this DELA protein. Okay, then there will be no functional of this DELA protein. So your growth genes will be on and they will grow tall. Okay, the main function of DELA protein is to inhibit the growth genes. Okay, but they will not inhibit here since the GI complex will recognize and degrade it. These are original DELA protein. But in case of mutation, the dominant mutation, your DELA protein or mutated DELA protein, okay, they will not recognize by normal GA. Okay, then they are free now. Okay, they are not recognized by GA and they are not degraded. So they are work to free now. So they will act on growth genes so that you will get a semi dwarf. Simple चीज, ठीक है, ज्यादा सोचना मत, ठीक है. PhD, one who are preparing for PhD exams, okay, one who are preparing for ASRB exams, then it is very important one. कुछ कोई बता रहा है, yes, the DELA protein are mutated, does their function is also not changed. वही बता रहे हैं भाई, ठीक है, they are insensitive mutation, ठीक है सागर, insensitive mutation, not completely non-functional mutation. That's what I am telling. They are only insensitive to GA. ठीक है, वर्क उनका फंक्शन तो चेंज नहीं हो रहा है, इधर फंक्शन तो नॉर्मल फंक्शन कर रहा है वो, ठीक है. दे विल रिप्रेस दी ग्रोथ जीन्स, ठीक है, बट they are insensitive, just they are insensitive to the gibberellic acid. Is there any change in the function of DELA protein with respect to inhibiting growth genes? Okay, now you are understand, I think. Okay, they were only insensitive, those are not non-functional mutations, ठीक है. सिंपल चीज, सेमी ड्वार्फ राइस, वीट तो हो गया, अभी राइस लाइक पिनत्राम फ्लावर. नूतन तो पता नहीं क्यों सेल्फ इनकम्पैटिबिलिटी आ गया बीच में. सेमी ड्वार्फ राइस, ठीक है. The simple gene is GA20 oxidase. Okay, this is the gene responsible for your बताओ कोई. बताओ. Sorry. Semi-dwarf in rice. This will be one of the enzyme in the pathway of gibberellic acid formation. ठीक है. GA20 oxidase 2. Now what will happen when this gene got mutated? Okay, the normal SD1 gene codes for GA20 oxidase, but when there is a mutation, recessive mutation, this is recessive mutation, okay, but your RST is dominant mutation. When there is a mutation to recessive condition, the activity of this GA20 oxidase will inhibit. There is no GA20 oxidase now, okay, the pathway will not happen, so there is no formation of your gibberellic acid, okay, in case of SD1, okay, then there will be a no growth. सेमी ड्वार्फिज्म सिंपल. राइस में तो सिंपल है केस, ठीक है. व्हीट में थोड़ा कॉम्प्लेक्स है, मतलब इफ यू नो द कॉन्सेप्ट एज आई टोल्ड, okay, it's also very easy. In case of rice, there will be GA20 oxidase 2, okay, when there is a SD1 gene, when it got mutated, there is no GA20 oxidase which is responsible for the pathway for the formation of GA, so there is no GA, so you will get a semi dwarfism, ठीक है. Yes, good, GA20 oxidase. Then what happened in the history of semi dwarfism in case of rice, ठीक है. Taichung native 1, the TN1, the first semi dwarf variety in case of the world, okay, which is from Taiwan, okay, cross between DG Wujen and Toyon Hunj, ठीक है. This is the first variety. We also introduced this particular variety when there is a famine. You have heard that, I think, Bengal famine. There will be a famine with particular to where there is a cultivation of rice. Okay, during that period we have introduced this particular TN variety. Okay, this is developed in Taiwan, cross between DG Wujen and Toyon Hunj, the first semi dwarf variety. Okay, then in Irri Philippines. Okay, again there is one more CGR institute as like CIMIT. Okay, they what they have did, they have crossed DG Wujen with Peta. Peta is an Indonesian variety. Okay, DG Wujen is Taiwan semi-dwarf variety. Then they have developed IR8, okay, and it is introduced to India in 1966. क्या है IR8? Miracle rice, okay, we will call it as a miracle rice. There will be a boom in the production of rice during that particular 1966-1967. Again, it got improved. There will be a 3x 4x improvement in the yield due to semi dwarfism, okay, in 1966. There will be so many varieties after this. IR8, IR24, IR36, okay, IR64. There will be so many varieties got released after IR8, okay. In India, Jaya, we have developed for our Indian condition, we have developed Jaya, which is cross between TN1 and T141, okay. Jaya is miracle rice in India. Good. IR8 is international miracle rice. सिंचन बता रहा है, ठीक है सिंचन. We will move somewhat fast. Next, novelization of Indian canes, ठीक है. C.A. Barber and Venkataraman in SBI Coimbatore. Okay, what they have did? Saccharum officinarum. This is noble cane. We will call this as a noble cane. Okay, this is very high sugar thick cane. Okay, your Saccharum barberry, our Indian cane, ठीक है, Indian cane, low sugar, thin stem, but it is hardiness, good hardiness, and also it will be suitable for growing in North Indian regions. Okay, it is very highly adapted. Saccharum spontaneum, wild cane. Okay, resistance to pest and diseases. ठीक है, that is your Saccharum spontaneum. क्या करता है बार बार वेंकटरामन, ठीक है, दोनों मिला के कुछ क्रॉस बनता है, ऐसे बनता है, पूरा रेवोल्यूशन होता है नोबलाइजेशन केन में, ठीक है. They will cross Saccharum officinarum into Saccharum barberry in the first phase. Okay, what they will do? They will transfer this high sugar and thick canes to low sugar and thin stem Saccharum barberry. Okay. Anyway, these Saccharum officinarum are not adaptable, not suited for North Indian condition. So they have crossed with to Saccharum barberry. Those are Indian canes. Okay. So what you will get? You will get thick stem, high sugar, and hardiness. Then you will cross these particular hybrids or varieties whichever they developed with the Saccharum spontaneum, okay, for the resistance to disease and pest, for abiotic stress. Okay, this is what we will call nobilization of cane. Okay, the cross between the noble cane with the Indian canes, then with the wild canes. There is one variety, Co 205, the famous variety which is developed through nobilization of Indian canes. Okay, from C.A. Barber and Venkataraman. Okay, you should remember that particular variety. We will call it as a wonder cane also. Then hybrid development, the phase of hybrid development in case of India. Okay, 1957. Okay, AICRP started for maize through the help of Rockefeller Foundation in India with the ICR. In 1961, hybrid in maize. Okay, Ganga 101. Okay, the hybrid got developed. There will be a starting of hybrid era in case of 1961 for maize. In 1964, sorghum. Okay, CSH1. Okay, combined sorghum hybrid 1 in 1964. In 1965, bajra HB1, hybrid bajra 1. Okay, after that there will be so many hybrid varieties got released in safflower, in sunflower, in rice. Okay, era got started. That is your hybrid development, the achievements in case of hybrid development in the beginning era. Yeah, Deccan Ranjit. Okay, there will be so many varieties started, but Ganga 1, Ganga 101, Tikin Utan वो तो शुरू में रिलीज किया था. Co 205 or 491, which is wonder cane. साजन Co 205 is noble cane. ठीक है, नहीं बताया था क्या? ये तो पहले ही रिलीज हुआ था, the first nobilization during the nobilization of cane. Then आपका 419, Co 419. This is wonder cane. Okay, what I have told is Co 205 and wonder cane. Wonder cane is your Co 419. ठीक है, समझ गए ना? हाँ, वही बता रहे सिंचन. Wonder cane is your Co 419. Then hybrid cotton in 1970, the first intra-hirsutum hybrid H4 was developed at Gujarat Agricultural University from C.T. Patel. Okay, the first intra-hirsutum. Okay, between the same species. Okay, Gossypium hirsutum. Then Varalakshmi 1972. Okay, world first interspecific hybrid from B.H. Katkari at US Dharwad. Okay, first interspecific. ये तो intraspecific है. Okay, intra-hirsutum H4, but it is interspecific Varalakshmi. Okay, hybrid cotton, the era of hybrid cotton, the achievement in case of hybrid cotton. Then transgenic breeding. Insect resistant BT cotton cultivation in India started in 2002. As I told, it got approved from GEAC in 2002. In case of India, the one and only crop which we are growing in Indian case. Okay, that is your BT cotton. Bikaneri Narma and a hybrid NH4 is an indigenous BT cotton. Okay, that means we could develop. Okay, खुद हम डेवलप किया है ICAR इंस्टीट्यूट से और तो प्राइवेट कंपनी से जुड़ के बीकानेर नरमा और hybrid NH4 is developed from our in our country. Okay, but BT cotton, okay, there will be so many MNCs, the events which developed, okay, those are from other countries. Okay, mainly your in case of BT cotton, okay, Monsanto worked very from the so many years, then they got released in India. Okay, that is from MNC. Again, it is from the outside also. ठीक है, ये तो दोनों improvement ये transgenic breeding में. Then in molecular breeding, as I told, okay, I already discussed it. Your HHB 67 improved. Your Pusa Basmati 1 improved Pusa Basmati 1. Then Annigeri 1 in chickpea. Note करो तो सारे note करो. ठीक है, downy mildew resistance. This is bacterial blight, BLB resistance. ठीक है, QRSG1 and 2 genes. ठीक है, in case of HB1. In Pusa Basmati XA13, XA21 gene. In Annigeri 1, FOC1 gene. ठीक है, these are some important varieties. After this, there will be a improved Samba Masuri, improved Sorana. ठीक है, your sub 1, Sorana sub 1, also improved Sorana sub 1. Then okay, these are some important varieties, but these are the major improvement in through the molecular breeding. Then आपका precision breeding, that is your gene editing. Okay, DRR done 100 and Pusa DST 1. आ गया इधर देखो. ठीक है, what exactly they have released, how they have worked it. Okay, first one is your from Indian Institute of Rice Research from Hyderabad. Okay, they worked with Samba Masuri. Okay, they took Samba Masuri, the initial variety. Then they edited one gene, that is cytokinin oxidase. They targeted this particular cytokinin oxidase 2. What it will do? Cytokinin oxidase 2 will inhibit, okay, or repress the production of cytokinin. Okay, it will inhibit the cytokinin production, so there will be no cell division properly. Okay, but what they will target this particular gene, so then they will switch off. Okay, they have edited this particular gene, then there will be no further functional of this particular gene, so there will be a accumulation of cytokinin, normal accumulation of cytokinin, so there will be a increase in the cell floral parts growth, so that you will get a more number of grains. Okay, more number of grains in Samba Masuri. So they developed DRR done 100. We will also call it as a Kamala. Okay, this is the first variety. They targeted for more number of grains. Then from MT1010, okay, they released Pusa DST1 variety. Okay, they have targeted here for drought and salt tolerance. This is from IARI New Delhi. Okay, is your, as I told, DGICR. Okay, कौन है DGICR? हाँ, DGICR कौन है? मिनिस्टर, यूनियन मिनिस्टर. ठीक है, ये आपका यूनियन मिनिस्टर. बताओ कोई. ML Jat. ठीक है, DGICR will be your Dr. ML Jat and Union Minister will be शिवराज सिंह चौहान. ठीक है, श्रीनिवास, ये तो IRA का डायरेक्टर है डॉक्टर श्रीनिवास राव. ठीक है, डॉक्टर श्रीनिवास राव. ये तो डॉक्टर विश्वनाथन is responsible for पूसा DST1, ठीक है, is सुंदरम, ठीक है, responsible for your DRR than 100. अरे व्यू कम हो रहा है, क्यों लोग छोड़ के जा रहा है? इंपॉर्टेंट बता रहे हैं मैं, सारे इंपॉर्टेंट है, सब कुछ इंपॉर्टेंट है। अनडिजायरेबल जेनेटिक कॉन्सिक्वेंसेस ऑफ प्लांट ब्रीडिंग, अचीवमेंट्स तो हो गया, ठीक है। Next you can see, we will see what are the undesirable genetic consequences of plant breeding. That is, one is your genetic erosion, okay, rapid depletion of genetic variability due to replacement of heterogenic local varieties. When you will develop some new varieties, okay, which are more uniform, okay, you will push for the cultivation, okay, what will happen? There will be a rapid depletion of genetic diversity, right? Okay, what the varieties you are giving, those are homogeneous, okay, homogeneous in nature, uniform, so you are making the variability reduction, that we will call it as a genetic erosion. Narrow genetic base, okay, the varieties again homogeneous, okay, they are not having that broad genetic variability, okay, there will be reduction in genetic variability present among varieties, so there will be a narrow genetic base. Due to this particular narrow genetic base, okay, there will be a development of diseases and pests, loss of particular varieties or particular crop, that is genetic vulnerability, okay, they are like susceptible for disease and pest, susceptible for every stress due to the narrow genetic base, that is genetic vulnerability. Then yield plateau, okay, genetic variability for yield will eventually be exhausted, okay, now it is already exhausted, as I told, now we have reached the highest yield level and there is no variability for yield only, now it is exhausted, okay, that is yield plateau. These are some genetic consequences of your plant breeding, okay, where you are trying to improve the different traits, okay, now we are experiencing this particular consequences, okay, vulnerability repeated sir, अनडिजायरेबल कॉम्बिनेशन लाइक टोमेटो एंड रेफोना ब्रासिका, वो तो छोड़ दो, ये वो तो मतलब कुछ बस एग्जांपल है, ठीक है ना, that is not that much influence in the plant breeding, but other, okay, when you are releasing varieties, those are very homogeneous uniformity, then there will be a different cases. कोई पूछ रहा है जेनेटिक वल्नरेबिलिटी, ठीक है, वल्नरेबिलिटी इज नथिंग बट, मतलब ऐसे हमारा फ्रेंड्स ग्रुप में हम बताया था, बता देंगे ना, अरे आप तो सबको वल्नरेबल है, ठीक है, तू वल्नरेबल है इस चीजें के लिए, ठीक है, किसी को मिल्क के लिए वल्नरेबल है, किसी को सम ईट, मतलब सारे समर में आप वल्नरेबल है, ऐसे बताते हैं ना, क्यों होता है वैसे, ठीक है, वल्नरेबल इज नथिंग बट ससेप्टेबल टू एनी डिसीज एंड पेस्ट. Why you are susceptible? Due to homogeneity, okay, due to homogeneous in nature, there is no variability, okay, if any one race attacks you, then your complete crop will be failure, ठीक है, आप मतलब heterogeneous material तो heterogeneous हुआ तो एक variety survive होगा, एक variety death होगा, so there will be some maintenance, but if your variety is homogeneous, then there will be a complete reduction in case of particular disease development. So due to narrow genetic base, okay, when you are breeding for homogeneity, then there will be a narrow genetic base, so it will make crop genetic, means it will make crop for vulnerable for plant and diseases, that we will call it as a genetic vulnerability, we are making it, ठीक है, समझ गए ना? बुलार बताओ, सिंचन, tends to susceptible or decrease tends to susceptible, वो तो सही है, ठीक है. देन इंडियन प्लांट ब्रीडर्स, एनीवे मैं तो सबको डिस्कस किया है ऑलरेडी, ठीक है, टीएस वेंकटरमण, नोबलेशन, बीपी पाल, NP series of wheat varieties, okay, new pusa varieties, NP series are nothing but new pusa varieties in the initial period, okay, your BP Pal is a wheat breeder and is also rose breeder, but you have developed some NP series wheats, okay, new pusa varieties, now they are obsolete varieties, now we are not growing it, okay. MS Swaminathan, father of green revolution in India, okay, there will be connection between Norman Borlaug and MS Swaminathan, okay, after he got introduced some Sonara 64 Lerma Rajo, then there will be secondary introduction, okay, those are Sonalika, Kalyan Sona, okay, which are from our MS Swaminathan. He also got Bharat Ratna, ठीक है. पुष्कर नाथ है पोटैटो ब्रीडर, ये तो नहीं पूछेगा. NGP राव सोरगम ब्रीडर, okay, the famous सोरगम ब्रीडर भी पूछ सकता है. ठीक है, रामैया राइस ब्रीडर, ओके, रामदान सिंह एक बार पूछा था शायद, ओके, इज आल्सो फ्रॉम व्हीट ब्रीडर, अतवाल pearl millet breeder, pearl millet, अतवाल, since they are very prominent in case of green, in the era of green revolution, in the era of hybrid development in the initial phases, okay, that's why you should know their names, ठीक है, NGP राव sorghum breeder, रामैया rice breeder, ठीक है, अतवाल pearl millet breeder, okay, you can see A2 A3 cytoplasm, cytoplasm कहाँ आता है, cytoplasm motility, ठीक है, वो तो हाइब्रिड डेवलपमेंट में आता है ना, इसलिए थोड़ा इनिशियल फेज में वो कुछ अचीवमेंट्स किया है, इसलिए सीटी पटेल एज आई टोल्ड फादर ऑफ हाइब्रिड कॉटन, बख्शी राम यादव शुगर केन ब्रीडर. नाउ यू शुड आल्सो रिमेंबर डॉक्टर AK सिंह, ओके, रिसेंटली ही गॉट पद्मश्री, ठीक है, पद्मश्री, ही इज रिटायर्ड डायरेक्टर ऑफ IARI न्यू दिल्ली, आल्सो फेमस, ओके, बासमती breeder, maybe there will be a question, maybe they can ask, ठीक है, ये तो आपको सबको बताना चाहिए. बॉयसन मेज ब्रीडर विजाली, ठीक है, धर्मपाल आयल ब्रीडर, गुड, okay, these are some prominent breeders, okay, as I told, yes, good, yeah, about father of plant breeding, Google tell Mendel in a mock test there is a Wilmorin. अरे यार ये तो सारे छोड़ दो ना, they will not ask you who is the father of plant breeding, there is no one, as my, if I want to tell father of plant breeding is nothing, father of plant breeding is farmer, okay, the domestication शुरू हुआ था ना, वही फादर है प्लांट ब्रीडिंग का. जेनेटिक्स को हम बता सकते हैं, मेंडल प्रिंसिपल्स सारे की दिया था मेंडल, प्लांट ब्रीडिंग में ऐसा नहीं है, ठीक है, ये तो मैं शुरू में बताया था, ये आर्ट्स एंड साइंस, आर्ट्स फार्मर, ठीक है, ये तो डोमेस्टिकेशन फेज, ऐसे सिली क्वेश्चंस नहीं पूछेगा, ठीक है, छोड़ दो, ऐसे टाइम वेस्ट मत करना. कौन पूछा था ये, रतन, ये छोड़ दो सारे. Rajaram wheat breeder, yes, good. Next phase will be domestication, okay, we will see the particular chapter, domestication. Domestication bringing wild species under human management, okay, for example, in case of brassica, you can see this is the particular wild mustard. If you see the mustard, how it got domesticated based on the human intervention, okay, this particular mustard plant, you can see if they are particularly selecting for lateral buds over so many generations, over so many years, it got converted to broccoli, okay. If they are particularly taking terminal bud, the terminal buds, your cabbage, okay, the large terminal bud, when farmers or when humans are selecting for particularly flowers and stems, okay, the sterile flower clusters, that is your cauliflower, the bud will be cabbage, flower will be cauliflower. When they are selecting for only leaves from the wild mustard, they are intensively, generation after generation, they are selecting for leaves, then they got kale, large leaves. When they are selecting for stem base, they got kohlrabi, okay, for the stem base. Then for the lateral buds, okay, the large lateral buds, when they selected for large lateral buds, okay, the Brussels sprouts, ठीक है. देखो सारे मतलब डोमेस्टिकेशन कैसे हो, एक ब्रासिका मस्टर्ड प्लांट्स है, ठीक है, सारे दूसरा क्रॉप्स डोमेस्टिकेटेड हुआ है, ठीक है, देयर इज नथिंग बट द ह्यूमन इंटरवेंशन, then there will be domestication, okay, bringing wild species, wild plants under human management. It is an ongoing process that continues human needs, okay, and priorities change over time. Example for recent domestication, that is Jatropha curcas, okay, for biodiesel, okay, for biodiesel, mainly for ethanol, okay. Our Indian government now promoting, okay, the ethanol E80, okay, the blending of this particular ethanol with the diesel, okay, that's why this particular crop now it is got importance. Next, Kala Zira, okay, Burnum persicum, it is also now recently got domesticated, okay. New crop improvement, okay, the wild rice, what is the Alta, okay, it is tetraploid rice, now it is under the domestication, okay, maybe in future maybe we will grow as a major crop, who knows. Okay, these are some recent domestication, selection under domestication. Okay, as I told, domestication is nothing but intervention of humans, right? Okay, intervention of humans while bringing the wild species under human cultivation. There will be two, natural selection and artificial selection. Natural selection is nothing but your environment, okay, the temperature, the rainfall. These are some factors. They will try to select some plants, okay, whether they are suitable for my extreme condition. Okay, in North India there will be a temperature extremity, heat extremity. Okay, based on that crop will got evolve in that particular region. For South India there will be a humidity. Okay, there will be some less temperature. Based on that crop will evolve in that particular location. In North America, okay, there will be a high harsh winter. Okay, based on the different natural conditions, based on different environment, okay, the crops will get evolved and domesticated. That is your natural selection. Artificial selection, okay, we will do selection. Okay, our ants, okay, they will intervening in the selection process. That is your artificial selection. Okay, there will be a different types of selection in case of artificial selection. That is your directional selection, stabilizing selection, disruptive selection. In directional selection, what we will do? Okay, in this particular case, okay, if you took a graph, if you take a graph which are low yield, high yield, we will always select for high yield portion. Okay, we will select this particular portion. One of the extreme in directional selection, we will select one of the extreme phenotypes, very high yielding phenotypes. That is your directional selection. What will happen here? What we are exactly selecting for? We are selecting for dominance. Okay, we are selecting for dominance condition, one of the extreme phenotypes. Okay, समझ रहा है ना? In stabilizing selection, okay, if you take this as a graph, okay, you are selecting for intermediate genotypes. Okay, the intermediate genotypes, the one who are giving intermediate phenotype, that is your stabilizing selection. You are maintaining variability. Okay, you are try to maintaining variability. Here you will select the gene action will be additive. There is no dominance, means you are selecting for both the alleles. Okay, that's why we are selecting for additive. Here we are selecting for major allele, dominant allele. That is your directional dominance. For extreme phenotype, one of the extreme, again one of the extreme. But in case of here, you are selecting for intermediate types. Okay, here in case of disruptive selection, if you look on graph, you are selecting for both extremes. Disruptive, you are disrupting the population. Okay, you are making some noise in the population. Okay, it will create the variation. Okay, it will create variation. You are selecting for low yield also, you are selecting for high yield also. Okay, yeah, it will select for both dominance and additive. When you are selecting for high phenotype and low phenotype, what it will do? It will create two separate population. Okay, there will be a genetic diversity increases, and you are making two different population. Over the years, it will create separate species. Okay, this will be separate species and this will be separate species. We will call this as a sympatric speciation. Okay, there will be two types of speciation. One is allopatric speciation, another one is sympatric speciation. Here, where there is a geographical barrier, ठीक है, एक टोबैको वैरायटी था, फॉर एग्जांपल टोबैको छोड़ दो, फेमस हमारा पोटैटो, ठीक है, एक पोटैटो वैरायटी था, नॉर्थ अमेरिका में ग्रो हो रहा था, मीन सब कल्टिवेबल कर रहा था फार्मर्स, मैं कुछ उस पे सैंपल लेके मैं इंडिया में लेके आता हूँ, ठीक है, ओवर द इयर्स, ओके, द सेम पोटैटो वैरायटी, okay, there will be a cultivable in case of North America and India. The same variety I took from there, and there will be over the generation, maybe after 1000 years, after 2000 years, it will grow as a separate species, and that will be a separate species due to the large random accumulation of mutations. That potato will be different, this potato will be different. Why? Due to the geographical barrier. Okay, due to geographical barrier, that is in South America, this is in India. Due to geographical barriers, there will be a speciation that we will call as a allopatric speciation. But what about sympatric speciation? There is no geographical barrier. Okay, in India only we are growing potato, and I am selecting the potato for some chemicals, for some carbohydrate content, starch content, high starch content and low starch content. Okay, in the same area we are trying to separate both of them. Okay, reproductively, means selection is nothing but you are giving a barrier for reproduction. You are selecting separately or selecting separately, and after some years, again after some 1000 or 1500 years, okay, there will be a two separate species. Okay, there will be a two separate species that will call as a sympatric speciation. Okay, but in case of there is a barrier in geographical manner, then it will call it as a allopatric speciation. ठीक है, ये तो तीन डिफरेंट सिलेक्शन है, डायरेक्शनल, स्टेबलाइजिंग, डिस्ट्रप्टिव. कोई पूछा था डाउट ग्रुप में, हमारा मास्टर ग्रुप में, रिगार्डिंग डिस्ट्रप्टिव सिलेक्शन and directional selection. Okay, the one of the extreme phenotype you are selecting, then it is directional selection. Okay, it is dominance gene action. In case of stabilizing selection, okay, you are maintaining variability again. Okay, and you are selecting the intermediate phenotypes. But in case of disruptive selection, you are selecting extreme phenotypes. ठीक है, बस इतना ही बेसिक. नेक्स्ट जर्मप्लाज्म. ठीक है, हो गया आपको, डोमेस्टिकेशन तो हो गया, सिलेक्शन भी हो गया. Germplasm is total hereditary material. Okay, all the genes and alleles. Okay, we will call them as a alleles and genes of all the particular species. Okay, the total hereditary material, or we can also tell as it is a pool of cultivars. Okay, the pool of different individuals or cultivars of particular species. वो भी बता सकते हैं. Present in crop species and its wild relatives. ठीक है, if we are talking about the germplasm of wheat, okay, it will cover each and everything, the cultivable species, wild relatives. ठीक है, सारे वो include हो जाएगा उसमें. We will also call this as a genetic resources or gene pool. ठीक है, सारे pool है. कौन सा components है इसमें? What are the different components? Your land races. Okay, the primitive cultivars in the farmer's field. Okay, they are growing from so many years, from the so many generations. Okay, those are very primitive, and they are also source for different disease, pest resistance, your obsolete varieties. Okay, for example, your NP series. Okay, the BB-PAL, the developed from BB-PAL, NP series, new Pusa varieties of your particular wheat crop. Okay, those are obsolete varieties. That is nothing but those are very famous during that particular time. Okay, after that, the development of new modern varieties. Okay, and now they are not under the cultivation. Okay, those are your obsolete varieties. Then modern cultivar, the present cultivars. Okay, your in case of wheat, SD3086, SD3386, okay, 3385. These are some 3471. Okay, there is no need to worry about these numbers, but these are some recent varieties, modern cultivars. In case of rice, okay, Pusa Basmati 1845, Pusa Basmati 1847, okay, 1980, 1979, Pusa Basmati 1985. These are some modern varieties. Okay, Pusa Basmati 1509. Okay, these are some modern varieties of rice at present condition, the present time. Okay, next, advanced homozygous breeding material. ये breeding material मतलब क्या है exactly? The in components of germplasm, your advanced breeding material also one of the component. Those are nothing but they are about to release. Okay, they are in the trial, or maybe they are in the field trial, in the breeder, in the particular with a breeder, they are about to release. Those we will call as a advanced breeding material. ठीक है, ज़्यादा confusion मत होना, मतलब होने के लिए जरूरी नहीं है. Wild forms of cultivated species. Wild forms. For example, for wheat, okay, what is the wild form? Triticum murata, Triticum monococcum, how it got evolved. For example, for rice, okay, Vareza sativa, there will be a Vareza glabrima, Vareza rufifogan. Okay, these are some wild forms. Okay, those we will also include in the germplasm. Wild relatives. Okay, in case of rice, there will be so many wild relatives. Vareza nivara, Vareza minuta. समझ रहे हैं ना? This will consider as a wild relatives. And mutants. Okay, whenever we treat some mutation to this particular variety, then we will get some other lines, right? Some mutated stocks. Okay, we will call them as a mutants. ठीक है, कुछ questions. Directional maximum fitness, good, most used in plant breeding. Yes, when we are doing directional selection, we are targeting dominance and for extreme phenotype. Okay, again, so that we can go for fitness, maximum fitness. In that case, your yield will be your fitness. Okay, genetic variance decreases in both stabilizing and directional selection. Sir, what is difference between variety and hybrid? अरे भाई ये तो पूरा बेसिक हो गया, हम डिस्कस करेंगे. हाइब्रिड ब्रीडिंग आता है ना, उसमें डिस्कस करेंगे. एडिटिव जीन एक्शन इज स्टेबलाइजिंग एंड इन डायरेक्शनल डोमिनेंस इज फेवर. ठीक है, वही बता रहा है सिंचन. फिर से राइस इज पैरेलल एवोल्यूशन. Parallel evolution nothing but there will be evolution of Indian rice, evolution of China or Japonica rice, and evolution of African rice. There will be 3 different parallel evolution. That's why we'll call it as parallel evolution. Too extreme. Sinchan, I did not understand sympatrical. अरे भाई इतना मैं clearly बताया था. Okay, when there is an 2 extreme phenotypes, we are selecting for 2 extreme phenotypes without geographical barriers in the same location. After so many generations, they will turn to 2 different species. Simple. ठीक है. Again मोहन पूछ रहा है, मैं बताया था ना मोहन. We'll discuss it. Primitive variety, living form as variety of past K65. DBW, those are varieties from again from AWBR kernel. Okay, those are also recent modern varieties. अब समझ आया. अरे ठीक है. Okay, we will continue now. Then gene pools. Okay, it is given by Erlan and David in 1971. Okay, primary gene pool, secondary gene pool, tertiary gene pool 4. Simple. What you should remember here. In primary gene pool, hybridization is easy. Pools are nothing but we will make a group. Okay, now we are having germplasm for particularly for wheat crop. We are having Triticum murato, we are having Triticum monococcum, Turgidum, Aestivum, okay, Triticum zuchovski, okay, Triticum, there will be so many varieties of wild relatives of wheat. Okay, we will make it as a, first we will take them as a germplasms. Then we should make a group among themselves. Primary, secondary and tertiary like that. Okay, what we will do? The species, the germplasms, which are, we can do the hybridization very easy among themselves. We will keep them in a particular group. Hybridization is very easy among themselves. Okay, and they are also fully fertile. We will keep them as a one group. That is your primary gene pool. ठीक है, समझ गए ना? Then secondary gene pool. There will be some difficulty in hybridization. Okay, since there will be a difference in floral structure, chromosome level. Okay, there will be some difficulty, but we can make a crosses. Okay, we can do a hybridization process. Okay, hybrids are partially fertile in that case. Okay, that is your secondary gene pool. Next, tertiary gene pool. There will be a greater difficulty. Okay, for example, there will be 2x and 6x genome size between 2 different germplasms. Okay, and there will be a different times of flowering, and also there will be a different floral structures, floral biology. So there will be completely difficult. In that case, we can use some breed species in between to transfer for the breed species, then to the cultivated species. Okay, for the primary gene pool. Okay, where hybrids are often inviable or completely sterile. Okay, that in that for germplasms, those germplasms will keep it in a germplasm 3. Okay, sorry, gene pool 3. There will be greater difficulty. What you should remember, hybridization is easy in primary gene pool. There will be some difficulty in gene pool 2, but we can do. but in case of gene pool 3, there will be a greater difficulty. Hybrids are fully fertile in GP1. Hybrids are partially fertile in GP2. In tertiary gene pool, hybrids are often inviable or sterile. Okay, the recent one, that is gene pool 4. Okay, we will also call it as a gene ocean. ये तो हमारा अभी recent ICAR ARS एग्जाम, ASRB नेट एग्जाम, उसमें पूछा था. ठीक है, GP4. GP4 includes your transgene, synthetic DNA constructs or sequences from unrelated organisms. ओके, उसको हम जीन पूल 4 में डिवाइड किया है. आपका बीटी कार्टन है ना. ओके, इन कार्टन germplasms, कार्टन जीन पूल. ओके, द बीटी कार्टन, that is the gene from another, there will be one gene, BT gene. We took it from Bacillus thuringiensis from bacteria, so that we will include those transgene, okay, in the tertiary, or sorry, tertiary, not tertiary, in the quaternary gene pool. That is your gene ocean. Okay, now we can include the each and everything, okay, whatever the germplasms which are developed through transgenic technology, synthetics, gene editing, okay, we will include in a gene pool 4. Okay, the recent one. This is all about your gene pool. Okay. When Erlan given, it was only 3 gene pool now. Yes, it is recently added. The basic 3 gene pool, okay, was given by Erlan and divided in 1971. बाबा, वो तो आपका ट्रांसजीन ये सारे अभी भी आए ना during 1990s, 2000. Barnes and Barstar, yes, we can include in the particular gene ocean. ठीक है, एक मिनट में थोड़ा एनर्जी ड्रिंक पियेंगे. 2 घंटा हो गया ना. हाइब्रिड टू वेराइटी तो व्यत्यास ಏನು सर? अरे मोहन, भी बताना है क्या तुझे? You want to know itself, Mohan? I will tell you. There will be a separate class for breeding methods. Definitely, I will explain very clearly. Yes, this is energy drink. Okay, given by your admin cum owner, Hari Prasad. हम आधा भी खत्म नहीं किया है. ठीक है, थोड़ा जल्दी करेंगे अभी. 63 व्यूअर्स. अरे भाई 100 से 60 आ गए क्या? वो लोग सोच रहे हैं तो इम्पोर्टेंट नहीं है. अरे भाई एक ही क्लास में 7 to 8 चैप्टर्स कवर कर रहा है हम लोग. ठीक है, इम्पोर्टेंट तो रहेगा, इंफॉर्मेशन भी रहेगा. And as I told, 1 hour our students, for them it is completely revision. Slides तो सेम ही है, उधर से उठा के आया है. मतलब इंफॉर्मेशन कुछ ज्यादा मिलेगा इधर क्लास में. मैं in classes maybe I forgot to tell something. Here maybe I am telling that extra informations. Okay, that will be, so you should be very careful. ठीक है, so that be with me in the class till the end. Then germplasm conservation. ठीक है, in situ germplasm conservation. First one, there will be a in situ and ex situ. Okay, in situ conservation is nothing but you are conserving the plants in their natural habitat, in their own natural habitat where they are growing. You are conserving in the same region so that you can give opportunity for the evolution. For example, national parks, biosphere reserves, gene sanctuaries. Okay, एक PYQ डिस्कशन में डिस्कस किया था पहले ऑलरेडी. ठीक है, कौन से नेशनल पार्क से? गिर नेशनल पार्क, जिम कॉर्बेट. ओके, योर नागरवड़े रिजर्व, that is also national park again. Bandipur. There will be so many national parks. They will come in the Institute Germplasm Conservation. Then biosphere reserves. Okay, biosphere reserves are nothing but in national parks there will be complete elimination of human involvement. Okay, as a human you are not able to enter to that particular boundary. But in case of biosphere reserve, there will be some involvement of humans in the outer boundary. Okay, so that you can enter that particular region and you can use some as a local people, you can use forest species for the timber purpose, for grazing purpose in case of biosphere reserves. But in case of national parks, there will be complete, okay, complete avoidance of human interventions. Okay, and gene sanctuaries. Okay, there will be so many gene sanctuaries in Meghalaya for citrus. Okay, for banana, like that there will be a gene sanctuaries where in the original place you are try to conserve. That is in situ conservation, best located in the center of origin of crops. Okay, where there is a center of origin for particular crops, okay, we will make them as the in situ germplasm conservation centers. In India, NBPGR will look into it. Okay, for gene sanctuaries, for example, as I told, citrus in Meghalaya, Musa, Varaiza, Mangifera in northeastern India. Okay, these are gene sanctuaries. National parks, as I told, I've already explained it. Next, what are the merits? Conserve natural genetic diversity, allows natural evolution and formation of new genes. Okay, no risk for the laboratory storage. What are the demerits? Difficult to establish and maintain due to human presence, human pressure. Maybe there will be involvement of humans, then they will destroy. So that is a demerit, and cannot conserve crop varieties. Okay, there will be a mutation. If you allow evolution, there will be a mutations, there will be changes in case of cultivable species. So it is again demerit. Okay, then ex-situ germplasm conservation. Okay, means conserving plant genetic material outside of its natural habitat. Outside of natural habitat means in the artificial condition. Okay, seed gene banks, field gene banks, shoot tip gene banks, cell and organ gene, shoot tip gene banks and cell organ gene banks. These are like through tissue culture methods. Okay, your DNA gene banks, you are conserving germplasms in the DNA level. Okay, DNA gene banks. Okay, in the crop, and also you can go for this shoot tip gene banks, cell and organ gene banks in the cryo preservation. Cryopreservation, nothing but minus 196 degrees Celsius in liquid nitrogen. Okay, you can also store like them. What about seed gene banks? Okay, and field gene banks. First we will see. Anyway, these are some tissue culture methods and cryopreservation. Okay, the most important ones are your seed gene banks and field gene banks. Field gene banks are nothing but you will grow them in a field and you will harvest the seeds every year. Okay, so that you can maintain, conserve and seed and multiply the seeds. What about seed gene banks? Seed gene banks are nothing but you are storing as the seeds. Okay, you are conserving in the form of seeds. Okay, there will be a 3 different seed bank collection. Okay, base collection, active collection and working collection. Field gene bank for recalcitrant seed. Yes, good, exactly, Vijayali. Field gene banks are for recalcitrant seeds, where there is a difficulty to store the seeds, so that we can sow the seeds in the field and we can maintain as a crop plants. For example, in case of your jackfruit, okay, in case of mango, okay, we will maintain them as a field gene banks. But where there is orthodox seeds, okay, we can store them as a seed. One is your base collection, active collection, working collection. Okay, what are the different conditions? Okay, it will be for 50 to 100 years. You can store the seeds. Okay, in case of active collection, for 8 to 10 years. Okay, in case of working collection, okay, will always dependent on working collection as a breeder. Okay, for 2 to 3 years. Okay, yeah, minus 196 वो तो हो गया. ठीक है, liquid nitrogen. Next, the moisture, seed moisture level. In which moisture we should store? In case of base collection, we can store them in the 5 degree plus or minus 2 to 3, 5% moisture. Here 5 to 8% moisture. Okay, here around 10 to 12 पॉइंट, मतलब नॉर्मल इन नॉर्मल टेम्परेचर, ठीक है। देन आपका ईयर ऑफ स्टोरेज हो गया, टेम्परेचर सॉरी मॉइस्चर हो गया, नेक्स्ट टेम्परेचर, ओके, अराउंड -20. Also we can go up to -196 degree temperature, okay. In case of active collection, it will be around 0 to 5 degrees Celsius. Here in working collection, okay, normal like 8 to 10 degrees Celsius, okay, or else we can go up to 15 degrees Celsius. There will be different values in different books, but you should know the basics, okay. As a base collection for more years, for the long years, there will be a less seed moisture and less temperature, okay, -20 degree normally, or you can go up to -196, then 0 to 5 degree, then 5 to 10 or up to 15, the normal working condition, okay. Your base collection you will store for longer duration, okay, as I told, so that we will also store in as duplicate, since for longer duration we don't know, okay, their viability and their vigor may be lost, so that we'll make it in a duplicate condition. Your active condition, okay, only we will use whenever there is a need, okay, whenever there is a need or for multiplication purpose, but in case of working collection it will be regular use by breeders for research, okay, for immediately if you want you can take it from working collection, okay. This is like for regular use. This is only for multiplication purpose whenever we need to multiply. The base collection will be for 50 to 100 years, okay, as a backup, okay. This is the simple 3 different collections. Base collection used for regeneration purpose. ये तो longer duration, as I told, whenever there is some natural calamities, whenever there is some, as I told, okay, there is one beautiful example, okay, in Nicaragua, Syria, they have stored the barley seeds and there will be so many different germplasms in their gene banks, and also they have duplicated the same genome resources, genetic resources in seed vault in Norway. As I told, there will be a gene bank, okay, in Norway seed vault. They are stored there also. When there is some recent war in Syria, okay, their own gene bank got destroyed in that particular situation. So then what they did, they retrieved their germplasms from their duplicated germplasm conservation, so from seed gene bank, okay, which is located in Norway, okay, the Doomswald, seed Doomswald, okay. Yes, security purpose. Good, Nutan. Then what is the role of NBPGR? Okay, what is the role of NBPGR in the germplasm conservation? सिंचन पूछ रहा है वही, किसके लिए वही, क्या पूछ रहा है तू, बताओ क्या चाहिए सिंचन? Svalbard Global Seed Vault, good, Vijayali, which is located in Norway, as I told. Nothing, sir. ठीक है. Evolution of MBPGR, the National Gene Bank, okay. As I told, in 1935, initial recognition for the need of centralized body in India. Okay, in 1937, there is a publication of landmark paper regarding germplasm conservation. In 1946, it started as a plant introduction scheme. This also is discussed in PYQ discussion also. Then 1961, the division of plant introduction separated as division of plant introduction, means everything under control of IRI. But in 1976, it separated as National Bureau of Plant Introduction. 1976, National Bureau of Plant Introduction. In 1977, National Bureau of Plant Genetic Resources. Okay, dono means under ICR. Till now, these are division and plant introduction scheme under IARI. Okay, then in 1976 under ICR National Bureau of Plant Introduction, in 1977 National Bureau of Plant Genetic Resources. ठीक है, उसके बाद क्या हुआ 1996 the National Gene Bank, the first National Gene Bank established in 1996 under NBPGR. Okay, in 2021 it got world second largest gene bank. Okay, world second largest gene bank which is located in NBPGR. Okay, we should be very proud of that. In 2025-26 now means in recent they are planning for second gene bank. ठीक है, ये तो टाइमलाइन है आपका NBPGR का. The next one is that is in national level. What about international level? In 1974 International Board for Plant Genetic Resources, it started as ICAR in 1974. In 1991 International Plant Genetic Resource Institute. Okay, in 1991 it got the new name, that is International Plant Genetic Resource Institute. ठीक है. In 1994 it got merged with INIBAP, that is International Network for the Improvement of Banana and Plantation, that is your INIBAP. INIBAP and IPGRI, they merged in 1994. Okay, the new name, that is Biodiversity International. Okay, Biodiversity International, they merged and they got new name Biodiversity International. Recently, okay, again they got merged with CAAT, that is International Center for Tropical Agriculture, that is in Colombia. They merged now. Now we are calling it as Alliance of Biodiversity International and CAAT. Okay, Alliance of, at present status it is Alliance of Biodiversity International. Biodiversity International, how it got came? It came from IPGRI and INIBAP. Now Alliance of Biodiversity International with the CAAT. Okay, recently. This is the International Institute for Plant Genetic Resources. As I told, our NBPGR National Gene Bank is second world largest gene bank with accession approximately 4,71,000 accessions. The first one is NPGS, National Plant Germplasm System, which is located in USA, around more than 6 lakh germplasms. Okay, the third one will be our Ricarda Gene Bank, then CIMIT. Okay, what are the activities of germplasm activities? What are the different activities? Okay, first one is your exploration and collection. Okay, you should go for collection, you should go for exploration where there is a need of collection. Okay, the varieties are some plant species, they are about to extinct. Okay, so you should conserve them. And when in the centers of origin there will be lot of diversity. For your breeding program, the source of breeding will be, as I told, variation. For that variation you should go for exploration and collection. Okay, in the centers of origin the first activity will be exploration, then you should collect those germplasms. It will be useful for your breeding program. Then after that, conservation. Okay, after exploration you should go for conservation. How you will conserve? In situ, ex situ. Okay, as we already discussed about it, in situ, ex situ. In situ there will be a national parks, reserves, gene sanctuaries. In ex situ, seed gene bank, field gene bank, shoot tip culture, meristem culture, tissue culture, different tissue culture methods, cell and organ culture. Okay, DNA gene banks. Then evolution. Then you will go for evolution. Which are the traits you are interested, which are the germplasms you are interested, you will go for evolution in the field. Then you will document it. Okay, based on the different traits, you will document which are the germplasms giving, which are the different traits and their measurements. Okay, then based on the interest of breeders, okay, they can apply for the gene banks. I need this particular germplasms for my breeding program. Okay, based on that they will distribute. Okay, your NBPGR will distribute the particular germplasms and you can utilize those in the breeding programs. Okay, These are the simple activities in the germplasm activities. ठीक है, वो तो हो गया, germplasm conservation हो गया. Now we are entering into centers of origin. Okay, we'll see. That will be one beautiful phenomenon, Wavelian mimicry. Okay, I will start with this. Everyone knows, our students already knows about it. Okay, what exactly the Wavelian mimicry? Anyway, we are talking about Wavella. Okay, who give the concept of centers of origin, so that we will see what exactly the Wavelian mimicry, the origin of rye and oats. Yes, in the beginning, traditional cultivated crops, your rye and oats are weeds. Okay, in Near East region, okay, in the Near East warm climate, okay, where Near East matlab your Turkey, Syria, okay, Arabian countries, okay, in that particular region there will be a warm climate. So in the wheat and barley growing areas, okay, there will be a weeds, okay, weedy oat and weedy rice. They will be growing as weeds. Okay, so farmers will remove those weeds, and there will be some remaining weeds. They will mix with the wheat seeds. So there will be a mixture. When farmers try to migrate from south to north, that means from the nearest region to the upper region, okay, temperate region, towards Uzbekistan, towards Kazakhstan, okay, towards Ukraine, Russia. Okay, they will migrate in the past years. Okay, they also took these wheat seeds. When they took wheat seeds, there will be mixture, right? Your wheat oat and wheat rye. So when they move to the temperate region, they will try to grow that particular crop. Your wheat, okay, your wheat and barley got failed there due to the temperate region, but rye and oats, okay, they are tolerant for the cold temperature. So they got domesticated in the northern Europe. Your wheat and oats. Okay, you can see here they are weeds in the hot climate, but in temperate region they are converted into cultivable crops. Your rye and oats. Okay, this for this particular phenomenon we will call it as a Wavelian mimicry. Origin of rye and oats. Okay, this phenomenon given by Nikolai Wavello. Okay, Nikolai Wavello. What exactly the center of origin? The most genetic diversity of a cultivated plants is concentrated in a few geographical regions. We will call it as the centers of diversity. First, centers of diversity. The most genetic diversity, okay, concentrated in particular geographical area. Then the concept was proposed by N.I. Wavello in 1926. He suggested that crops evolved from species in a region showing maximum diversity. Okay, the crops will evolve, okay, from the region where the crops are showing maximum diversity. For example, wheat. Wheat is showing maximum diversity in Asian minor region. Again, the same area, the Turkey, Syria, okay, those area, the wheat crops is showing maximum diversity. So we consider that particular region as a centers of origin. Okay, also known as centers of domestication. Okay, in the beginning era, means in 2026, when N.I. Wavello gave these particular terms, okay, everything is like similar. Centers of diversity, centers of origin. Okay, he recommended all words. But what happened later? Okay, some of region like Ethiopia, okay, some of the region like Ethiopia shows high diversity for cultivated crops but lack their wild relatives. To explain this, Wavello proposed primary and secondary centers of origin. Okay, in Ethiopia, okay, they show higher diversity of cultivated crop species. There is having some diversity, but there are no wild relatives. Okay, how we can say, okay, without wild relatives, how we can say that is the centers of origin? So he proposed primary and secondary centers of origin. Your primary centers are areas where crops were first domesticated and wild relatives are presented and high diversity. Okay, first domesticated and wild relatives are present. Your secondary centers areas where crops spread later and developed considerably diversity, even though they did not originated there. Your primary centers are exactly the centers of origin, okay, where they got domesticated and wild relatives are present. But your secondary centers, they are nothing but they are having high diversity, but crops are not originated there. Okay, the simple difference between primary and secondary centers of origin. Okay, as per Wavillo. Okay, NI Wavillo also established Institute of Plant Industry in Leningrad in Russia. Okay, in Russia. He is from Russia. Institute of Plant Industry, which is nothing but gene bank. Okay, anyway, he is like a good traveler. He explored so many countries. Okay, for example, 64 countries. You can imagine, just imagine, during 1920s, he explored 64 countries and he collected each and every seeds, okay, wherever is required, and established first gene bank. First gene bank, that is Institute of Plant Industry in Leningrad. He also proposed law of homologous series in variation. What it will tell? Law of homologous series, the variation present in particular species is also present in related species. Okay, simple. ज़्यादा सोचने के लिए जरूरत नहीं है. What exactly? Law of homologous series, which is proposed by N.I. Wavillo. You are having some different rice species, which is one is your Vareza sativa, that is your Indian rice. Vareza rufifogan or glabrima, African rice. Okay, the variation present in your indica rice, the particular species Vareza sativa, there is also present in the African rice. Okay, or if you go somewhat more different species, for example, the variation present in rice, this is complete different species, also present in the related species. For example, your Triticum aestivum. There are two, both are from cereals. Okay, there is some relation between them, right? We can see there will be similar looking phenotypes between rice and wheat. Yes, there will be some different plant character, but you can see there will be some similarity. That's what exactly he told. Okay, the variation present in particular species also present in that related species. Okay, tetraploid and hexaploid wheat. Yeah, between tetraploid and hexaploid wheat, between African rice, Indian rice, between rice and wheat. Okay, there is some similarity, right? That's what your Wavillo told. Okay, what are the different centers of origin in 1926? Okay, he given 8 regions. Okay, first he given 8 regions of centers of origin. You should remember, not 9, 8. Then classified them into 11 original regions. Okay, they divided into 11 regions plus added one more centers of origin. नूतन कुछ दे रहा है. Out. नूतन क्या बताओ. Okay, first this is China region. Okay, this will be first region, the China. Then Indo, मतलब Hindustan region. First initially given as Hindustan region. Then it divided into Indo-Burma and Siam, Malaya, Java region. ठीक है, China, Indo-Burma region, Siam, Malaya, Java region. ठीक है. Then Central Asia, which contains the Uzbekistan, Tajikistan, Kazakhstan, सारे आता है ना वो region. ठीक है. Then Asia Minor, Turkey, Syria. Okay, इधर आता है, सारे इधर आता है. ठीक है. The main fertile crescent बोलता है ना, fertile crescent is nothing but the origin of wheat. The crop got domesticated. Your Assyrian Babylonians, सारे आता है ना, ये रीजन से आते हैं, एशिया माइनर, टर्की, सीरिया रीजन से, ठीक है। द फेमस रिवर यूफ्रेटिस एंड टाइग्रिस, okay, it will also from this particular region where the agriculture started, okay, that is your Asia Minor. Then Mediterranean region, famous for olives, okay, your southern Greece, southern Italy, okay, everything will come here, Mediterranean region. Then Abyssinian region, Ethiopian region, okay, Abyssinians, this particular region. Then your North America, in North America also Peru region, Chile, Paraguay and Uruguay, classified into 3 different regions, okay. This is your USA region, the newly added last region, okay, and this is Mexican region where your maize got evolved, okay. समझ आया ना? 8, there will be 8 major region. If I want to tell 8 region, first is your China, then Hindustan region, second one Asia Central, Asia Minor, 4 हो गया. 5 Mediterranean region, then 6 your Abyssinia or Ethiopian region. Next South America, okay, the South American region and North American region, ठीक है. This is the different 8 region, but later he divided Hindustan region as Indo-Burma and Java, Siam, Malaya region, and North American region into 3 region, that is your Peru, Chile, this is Chile, and Paraguay and Uruguay region, ठीक है. और he added one to the new region, that is USA, ठीक है. इसलिए वो 8 से 11 plus 1 हो गया, 11 plus 1 that is 12, ठीक है. ये तो आपको in 1935 these were expanded, okay. In 1926 he gave original centers of origin. In 1935 expanded to 11 center by subdividing your Hindustan region into Indo-Burma, Siam, Malaya, Java centers, and South American region into Peru, Chile, Brazil, and Paraguay region, and adding your USA center. USA center is center of origin for your sunflower and Jerusalem artichoke, ठीक है. Modern classification, there more one more classification from in 1982, Zevan and Dewitt, okay, proposed 12 major centers of diversity, including Chinese, Japanese, Hindustan. There will be complete different classification, okay. But what you should know at this particular concept, Zevan and Dewitt gave 12 major centers of diversity, उतना है बस. उनका classification important तो नहीं है, okay. The important is Vavillo classification, okay. 8 into 11 plus 1, that you should remember. ये तो छोड़ दो, सारे छोड़ दो, जरूरत नहीं है. These are some those 11 plus 1. Abyssinian center, famously it is for barley and triticum, okay, and also safflower, sesame, castor and coffee, okay. Our coffee got introduced from that Ethiopian region, okay. In Karnataka it got introduced from Baba during Baba Budangiri, okay. There is a region, okay, the coffee got introduced there from this particular African countries, Ethiopia, that is from Abyssinian center, you should remember, okay. Secondary तो नहीं पूछेगा, ज्यादा मत सोचो. Primary centers of origin they will definitely ask. Asian Minor center, triticum species, from where? Asian Minor, you should remember, Turkey and Syria region, okay, from the Fertile Crescent. Others are like carrot, cabbage, okay, utna your wheat and rye. Central American region, Central America, those are maize. Central America matlab your Mexican region, okay, this region, this particular region, maize, okay, maize and rajma, which are very important, and your cotton, okay. Central Asia region, Central Asia like Afghanistan, Tajikistan, Uzbekistan, okay. Apple है क्या? Apple, your linseed, safflower, okay. Onion and garlic, from this particular Afghanistan center, and apple. Okay, you should remember. China is soybean and radish. They will ask soybean from China. And Hindustan center, rice, pigeon pea, important chickpea, all pulses which are from our center. Brinjal, okay, mango, orange, coconut, banana. Okay, there will be so many crops from our region. Mediterranean region, as I told, Titicum like it is like merging with everyone, but the main center will be Asia Minor for wheat. And as I told, onion, garlic, beet, lettuce, and also olives, clovers and olives. Okay. South American centers from Peru, Chile, Brazil, Andes mountains. Okay, आपका क्या पोटैटो कहाँ से? पोटैटो, मेज, पाइनएप्पल, ओके, टोमेटो, टोबैको, सारे ठीक है, tobacco, potato, solanaceous species from the South American centers. That is Peru, Chile and Brazil and Paraguay. US center, as I told, your sunflower and Jerusalem artichoke, the recently added centers. There will be some criticism. Centers of diversity are not always centers of origin. That's why your Wavilla made it primary centers and secondary centers. But some regions show high crop diversity but lack wild relatives. Okay, so the crops are not originated there. Environmental diversity, okay, different climates, mountains, valleys increases the variation within the species. Okay, wherever there is a different environmental condition, for example, in Western Ghats, okay, there will be some different species are available. Okay, so that there will be variation in this particular Western Ghat regions. Many centers of diversity lies in the mountain valley regions, which naturally promote variation. Mountain regions will promote variation, but your mountain regions are not centers of origin, but there is a variations present there. So we can't tell that as the centers of origin. That's why there will be a criticism, right? Centers of diversity can shift over the time. Okay, centers of diversity can shift over the time due to crop improvement. Okay, but the centers of origin should be the same place, right? From the origin, from the particular era. But your diversity can change, they can shift over the time. Some crops have more than one centers of diversity. Okay, making the true centers of origin difficult to identify. You can see in case of wheat, there will be diversity in Asia Minor, in case of Central Asia, in case of Mediterranean region, in case of Ethiopian region. Okay, you can see here your Triticum species in Asia Minor region, in Abyssinian center, in Central Asia, in Mediterranean region. There will be diversity, and that's why they are like given in all the given in all the centers of origin. In all centers of origin, he mentioned wheat, so there will be a confusion, right? Some crops having more than one centers of diversity, making the true centers of origin difficulty to identify. Therefore, these regions are better to be called centers of diversity rather than centers of origin. Okay, so now that's why at this particular present time we are mostly using the term centers of diversity rather than centers of origin. We don't know where exactly the crops are originated from due to the different reasons. As I told, your environmental thing and your diversity will shift over the time due to so many reasons. Okay, now we are not using the term centers of origin. Okay, we are using centers of diversity. Then Zhukovsky, again in 1965, you should know the name. Okay, proposed 12 mega gene center. Okay, for crop genes, for crop diversity, for micro gene, as like mega and micro centers of diversity. Okay, micro centers are small areas within the large centers of diversity as having higher diversity. Okay, evolution occurs faster in micro center, making them important for germplasm collection. There will be a mega gene center. Okay, he proposed 12 mega gene centers, Zhukovsky. Okay, for particular crop. Okay, if you consider this as a mega gene center, at particular core, okay, there will be high diversity of croplands. So we'll call this as a micro center as per the Zukowski classification. Okay, what are those 12 megagene center? We don't need it. Okay, only we want to know the particular 8 region converted into 11 plus 1 Wavilo, according to Wavilo. That is more important. These are different classification given by different scientists. वो तो centers of origin. ठीक है, हम अभी at present time what we will call? We will not call it as a centers of origin. We will call them as a centers of diversity. Okay, we don't want confusion. Okay, we don't know exactly from where the crops got originated, so we will call them as a centers of diversity. Better than over your centers of origin. ठीक है. Next, plant introduction. We already saw that centers of origin. Now we are entering into the crop improvement, the different breeding methods. Or plant introduction also one of the breeding method. ठीक है. Urban center and non-center classification. Center and non-center classifications are nothing but your primary, similarly same as a primary and secondary centers of origin. ठीक है. Erlan also gave the center and non-center classification. Similar, that center is nothing but your centers of origin. Okay. Non-centers are nothing but they are not particularly centers of origin, they are centers of diversity. ठीक है विजयाली. Those are similar to primary secondary classification as given by Wavilo. He just gave the name center and non-center classification. ठीक है विजयाली, बताओ क्लियर है ना? Sir, brinjal in China also. Yes, exactly. That's what confusion. Abhijit Tiwari. That's what the confusion is. Brinjal got originated in China also. Brinjal got originated in Indo-Burma region also. So there will be a confusion. So now we are left that particular centers of origin concept. Okay, the centers of diversity. So brinjal is having centers of diversity in both Indo-Burma region and China center. Okay, if they ask in your exam, they will mostly target the major crops like soybean. Okay, there is no confusion for soybean. For triticum, Aestium also they will not ask. If they ask, you should choose Asia Minor region. Okay, the Tigris and Euphrates region, Fertile Crescent. Okay, we will move. For plant introduction, it is nothing but a process of moving a plant into new area where it has not been grown before. Okay. For example, potato, tobacco, your tomato, okay, your maize, rubber, coffee, these are all introduced crops. ठीक है, हम तो पहले से नहीं ग्रो कर रहा था हमारा रीजन में. ठीक है, ये सारे इंट्रोड्यूस्ड क्रॉप्स है. Exotic material, bringing plants from other countries, domestic movement, moving a crop within the same country. Okay, as I told, growing rice in Punjab, grapes in Haryana. पंजाब में तो पहले से राइस ग्रो नहीं कर रहा था, अभी हम मूव किया है वेस्ट बंगाल रीजन, दैट नॉर्थ ईस्टर्न रीजन से. ठीक है, ग्रेप्स, ओके, विच आर द ट्रेडिशनल रीजन्स आर महाराष्ट्र, ओके, द सोलापुर रीजन and the northern Karnataka like Belagaum, okay, Bagalkot, that particular region, and some Kolar, the Bangalore, near Bangalore in southern Karnataka. These are some traditional grape growing area, but now grapes are growing in Haryana also. Okay, this is the domestic movement, the domestic introduction of plant. Okay, the plant introduction. Exotic means we are bringing from the outside of the country. Your tobacco, tomato, potato from North America. Those are exotic material. Primary introduction, as I told, the very important example are your Sonara 64, okay, Lerma, Rosa, which are like same as it is from the outside of the country, we have introduced into the Indian condition. Those are your primary introduction. By using those primary introduction, okay, we will use them as parents in next breeding program, or we will use in hybridization program, or we will do some selection in those particular varieties. Then we will release, for example, your Kalyan Sona and Sonalika. These are secondary introduction. Primary introduction as it is from outside the country, we will push into our farmers. That is primary introduction. Second introduction क्या होता है? From the same varieties, we will do some hybridization program, or you will use some selection among themselves. Then we will release some varieties according to our condition. वो second introduction होता है. ठीक है? Yeah, good रिमी है क्या? वसुंधरा, वसुप्रदा, ठीक है, विजयाली, ठीक है, गुड. उतना है, सिंपल. ठीक है? This is the list of the crop plants. Okay, we introduced from different countries: potato, tomato, tobacco. Okay, these are 3 from South American regions. Okay, we introduced into India: cherries, grapes, cherry and grapes in 1300 AD from Afghanistan. ठीक है? From Muslim invaders. Okay, as I told, cherries, grapes, apples, onion, garlic, these are from Afghanistan region. Okay, from the Muslim invaders. Muslim invaders कौन है? Muslim invaders आपका मुगल्स. ठीक है? वो सारे आता है ना आपका पूरा इन 1300 AD पीछे, वही ये लोग आपका ग्रेप्स, चेरीज, एप्पल्स लेके आया था उधर से. नेक्स्ट ग्राउंडनट, मेज. ठीक है? ग्राउंडनट तो ब्राजील से आया, पोर्चुगीज. मेज आल्सो फ्रॉम पोर्चुगीज. मेज कहाँ से? अगेन फ्रॉम मेक्सिकन, फ्रॉम सेंट्रल अमेरिकन. योर कॉफी, एज आई टोल्ड, साउथ ईस्ट अफ्रीका, Ethiopian region. Okay, then your tea and lychee and locust. टी, लीची एंड लोकेट फ्रॉम चीना. टी, लीची, टी कैसे आया था वो तो मैं ऑलरेडी एक स्टोरी बताया था अच्छे से. ठीक है? वो तो हमारे स्टूडेंट्स को पता है. It is actually they have planned, British has planned and sent on person to that particular tea growing region. Okay, he took those seedlings and gave to British, then introduced into northeastern region in India. Okay, so that's why we are growing in the particular region very commercially. From China, from the East India Company, and your cabbage and cauliflower from Mediterranean region, from East India Company again, and your anato, mahogany from West Indies, from East India Company. West Indies कहाँ आता है? देखो इधर आता है. Okay, this is your West Indies, the islands. Okay, आपका ठीक है, mahogany and anato. Okay, these are from West Indies country, from by East India Company. The National Bureau of Plant Genetic Resources is the main authority for the plant introduction in India. ठीक है? NBPGR for agriculture crops, horticulture plants and medicinal and aromatic plants is the main agency for the introduction. Forest Research Institute, FRI, for introduction and testing of forest and germplasms. Botanical Survey of India for botanical and medicinal importance. Central Research Institutes. Okay, for example, your Central Research Institute are nothing but your ICAR institutes. Okay, your IIRR for rice. Okay, IAWBR for wheat. Okay, for sugar cane again, Indian Institute of Sugar Cane Research, Lucknow, SBI, Coimbatore. For tobacco, there is ICAR Institute in Rajahmundry in Andhra Pradesh. For potato, again in Shimla there is a institute for potato. Okay, there will be a ICAR Institute. Those are responsible for the introduction of particular crop plants. But your NBPGR is the main authority. Okay, for any introduction, you should go through the NBPGR. Introduction. How we will do the introduction of the particular germplasm? First will be procurement. How we will procure the germplasms? NBPGR is sole agency for all introduction, as I told. You should go through NBPGR. Okay, what are the requirements? हमेशा बता रहा था मैं, ठीक है, सब क्लास में बताया है ऑलरेडी. What are the requirements? One is phyto sanitary certificate. Another one is import permit. जल्दी बताओ, कोई जल्दी बताओ, कौन इंपोर्ट परमिट देता है? ठीक है, कौन phytosanitary certificate देता है? Who will give phytosanitary certificate and who will give import permit for introduction of any germplasm? बताओ कमेंट बॉक्स में। अरे सारे at the end of the class till the end of the class you should be with me, ठीक है। अभी आधा आधा खत्म हुआ है 50 स्लाइड, अभी भी 50 स्लाइड से सेल्फ इनकम्पैटिबिलिटी तक जाना है, ठीक है। फिर से वो जाके बुक खोलना है, बुक पढ़ा मतलब बुक में देखना है, सब छोड़ दो, ठीक है। मेरा साथ रहो, बस ये 3 मैराथन क्लास को रिवीजन करो फिर से, ठीक है, सब कवर हो जाएगा। कुछ कुछ पॉइंट्स तो मिस हो सकता है बट कॉन्सेप्ट तो सब कुछ मिलेगा, ठीक है। इंपोर्ट सर्टिफिकेट DPPQS देता है क्या? बीट बाउंस, अरे भाई आप जाके मेरा PYQ देखना है, जरूरी है आपके लिए। तो कौन कौन DPPQS बता रहा है ना, मधु हेगड़े, चैतन्य देवरकोंडा, विजया अली, डिलीट कर रहे हैं सारे अभी, ये सारे रिट्रैक्ट कर रहा है आपका मैसेज, ये सारे लोग जाके मेरा PYQ देखना ही देखना है, ठीक है। इस पर एक क्वेश्चन आना चाहिए। यस, गुड। योर import permit, it will be given by NBPGR, okay, the director of NBPGR, phytosanitary certificate from agency from outside country, okay, from outside country, okay, one who are sending your germplasms from that particular country, there will be agency to take care of germplasm, they will give your phytosanitary certificate। DPPQS बता रहे हैं, अरे भाई क्यों क्यों ऐसा हो गया? एक्सपोर्टिंग कंट्री गुड। नेक्स्ट क्वारंटाइन, इधर आता है आपका DPPQS, ठीक है, DPPQS, ठीक है। कोई बता रहा था ना DPPQS क्वारंटाइन में आता है, अकॉर्डिंग टू दिस पर्टिकुलर DPPQS, ओके, you should do normally average 40 days, quarantine will be for 40 days and they will also give some norms, okay, how you should do the quarantine, you should follow DPPQS rules and regulations। कहाँ है DPPQS, कोई बताओ। next you will do the cataloging, you will see each and everything, okay, what are the requirements, what are the information they have given, each and everything, then you will do the evaluation of that particular introduced material, everything will be take care of NBPGR, ठीक है। it will see, okay, it will see for the material if there any contamination, okay, everything it will see, then there will be multiplication and they will distribute, okay। if the application from 2 or 3 agencies or the material should need in the large quantity, then they will do multiplication, they will do the distribution of that particular germplasm, okay। Faridabad, good। Cataloging is nothing but after the quarantine they will see, okay, for which purpose they need material, okay, what are the output and what are the roles of that particular institute, okay, why they are needed for that particular material germplasms, it will check it will see for each and everything and they will catalog it, okay। Cataloging is they will maintain each and everything data, then they will go for further thing। this is the simple thing, you should remember phytosanitary certificate and import permit who will give, important है, ठीक है। then purpose of plant introduction of entirely newly crop, ठीक है। मैं बार और बता के ऑलरेडी तक गए, पोटैटो टोमेटो, ठीक है, पोटैटो टोमेटो मेज टोबैको, ये सारे इंट्रोड्यूस्ड क्रॉप है एज एंटायरली न्यू क्रॉप, ओके। लॉन्चिंग न्यू वेराइटीज, ओके, प्योर सोनारा 64, ओके, धर्मा रोजो एंड क्लार्क, ये सोयाबीन वैरायटी है USA से, ठीक है। केंट वोट का वैरायटी है, Okay, these are all some different varieties which are introduced directly. Okay, crop improvement in hybridization. For example, Seox is introduced variety from USA, crossed with Miruti. Okay, Miruti. Okay, Pusa Ruby. Okay, they are used in the hybridization process. Okay, for example, there will be so many. Your Solar 64 Larva also, they are also used in hybridization program. That's why you got Kalyan and Sonalika. Okay, there will be so many examples, and also they will definitely not ask these basic are already they are like old achievements or improvements. They will target the recent ones. As I told, I will again and again telling in the each and every classes. Okay, बार बार मैं बता रहा है, ठीक है? If they ask, they will ask the recent achievements. Protection from pests and diseases. Okay, protection from pests and diseases matlab if in some particular growing area, if the occurrence of some pest and disease, to protect that, okay, we will introduce that particular crop to the another region. Okay, for example, coffee got infested from different diseases and pests in the Sri Lanka region, so they try to move that particular varieties in different regions. Okay, like that we can protect them from the pests and diseases. Scientific research we can use in scientific research, okay, by introducing the particular crop plants as the aesthetic value. Okay, your for example is water hyacinth. They have introduced that particular crop as for a aesthetic value, but now it turned as a weed. Your lantana camera, okay, for the along roadside, okay, it will be look like very good, okay, like flower. It is like a flower. Your lantana camera, so that we tried to import means introduce that particular crop in India, but now it is turned into a weed. ठीक है, वो भी एक आपका पर्पस था, एस्थेटिक वैल्यू. ठीक है. What are the demerits of your plant introduction? Entry of weeds, as I told, your Argemona mexicana introduced unintentionally. Okay, it is not in the particular in Indian case scenario, but when you are introducing some other crop plants, your Argemona mexicana introduced. Okay, Icornia crassipis, water hyacinth, as I told, introduced as ornamental, but now it is a weed. Your Phalaris minor, Geunsa, famously we will call in Hindi Geunsa. Okay, Geunsa, introduced along with a weed seed. Okay, it is also your Phalaris minor is not from our country again. Okay, introduction of diseases, late blight of potato introduced from Europe, flax smut of wheat from Australia, coffee rust from Ceylon. Okay, these are some different demerits. Your bunchy top of banana again from Ceylon. Okay, potato tuber moth from Italy, woolly apid of apple, fruit scale of citrus. These are the different diseases or different insect pests from the when we are doing the plant introduction. Okay, these are the demerits. गिल दंडा, गिल्ली दंडा, नूतन क्या है वो गिल्ली दंडा, बताओ मुझे भी कुछ. Ornamental turned weeds, water hyacinth again used as a ornamental, now it is turned into weed. Lantana camera will famously called as a curry. ठीक है, it is also like turned into weed. Threat for ecological balance, your eucalyptus species introduced from Australia. Okay, we introduced from Australia for timber purpose, and also it is as having some medicinal importance. Your eucalyptus, Nilgiri, we'll call it as a Nilgiri. Okay, no, but it is due to its deep root system. Okay, depletion of subsoil and underground water threatening to ecological balance. It will what it will do? It will take water from the subsoil and underground water. Okay, due to that there will be a threat for ecosystem balance. Sir, one MCQ for entry of weed. मतलब क्या पूछा था क्या? We learn from Icornia crassipis. Not everything looking beautiful is good. Good, Shinchan. Good. अरे मैं ये बीच में लगाया था इसलिए आ गया. अभी देखो ये अभी हम आधा मतलब 50% में. ठीक है. For one who don't know about our courses, ठीक है, now the mock test series will be available for 399. ठीक है. For one fact I will give you here, among there will be a 3,500 students are appearing for this particular ICAR PG plant science exam. the data, the exact data I am telling. Okay, around 3,500 people will appear for ICAR PG plant science exam. Among that, okay, 2,000 students, okay, 2,000 students already enrolled for our test series. Frankly, I am telling. Okay, and also for our master course, okay, this is our master course, around 1,200 plus students already enrolled for our courses. So if you are not yet registered to our courses, it's time to buy our particular test series. Okay, I will not recommend, okay, personally, that particular master course. Okay, this is already late to cover each and everything, video sessions, complete details in our master course. But if you are not yet registered for test series, okay, you should purchase our test series today itself. Okay, otherwise you are definitely missing it. If I want to say, if you are not enrolled into our test series, already you are out of competition, already you are out of the match. So go today itself, you should buy the plant science test series only for $399. It also covers each and everything, the different mock tests, okay, the discussion, which are the questions are in the mock test, okay, PYQs, most expected series, PDFs, PPTs, okay, whatever we did in the YouTube series, everything you will get in this particular test series. Okay, if you are having interested, you can also buy our master course for 1,999. Okay, and one more watching this particular marathon video, and one more preparing for next year, okay, for 2027, you are getting our particular course in 4,999. And for next year, but for next year means for 2028 course, for 2028 we are launched already course for 8,999. Okay, if you are already delayed, one more preparing for next year, okay, you should go and purchase. And the again, one of the important course, okay, ASR Minute plus ICR PhD for genetics and plant breeding. That validity is 365 days from the date of purchasing, from the date of registration. Okay, available only for 6,999. Okay, this is ASR Minute plus ICR PhD. Both the syllabus we have covered. Okay, anyway, our students already knows about it. Okay, what are the different according to syllabus, what are the different sections we have made it, and what are the different topics we are already covered it. For every year there will be a live class sessions, and when we are purchasing now, there will be already recorded classes for them also. Okay, in all courses we are having both live and recorded sessions. Okay, Pooja ICRS sir. Flourish, not flourish. Another noun name of flourish minor also Gehu ka mama. Yes, Nutan, you are from Nutan. Where are you from? गेहूं का मामा बोलता है उसको, ठीक है. मैं भी मतलब वीट में काम कर रहा है, मैं तो साउथ से हूँ. एक स्टोरी बताता हूँ बीच में, 2 मिनट. मैं तो साउथ से हूँ, ठीक है. मुझे मतलब ये सारे पता नहीं था, मतलब कॉमन लैंग्वेज हिंदी में क्या बोलता है उसको. नूतन एमपी से, ठीक है, इसलिए पता है आपको. मेरा एक गाइड था, मतलब मेरा साइंटिस्ट और प्रिंसिपल साइंटिस्ट है, वीट में ज्यादा काम किया है ऑलरेडी. मैं बताया था ना, HD 3086 3386 सारे उनका अंडर आया है, ठीक है. वो तो थोड़ा नॉलेजेबल पर्सन है फील्ड में, मतलब सब पता है उनको. फील्ड में आया था एक दिन, मैं क्या उसमें मतलब कुछ क्रॉसिंग उसमें लगा हुआ था, कुछ क्रॉसिंग कर रहा था फील्ड में, ठीक है. मैं तो मुझे समझ नहीं आ रहा था वीट और फेरारिस में वो दोनों डिफरेंस में, ठीक है. मैं मिस्टेकनली, ओके, मैं वो फेरारिस माइनर है ना, उसमें देख रहा था, स्पाइक ढूंढ रहा था. अरे भाई इसमें स्पाइक कहाँ है, स्पाइक ही नहीं है, दोनों सेम लग रहा है, वीट और फेरारिस माइनर, ठीक है. उसी समय सर आया था हमारा मतलब गाइड, ठीक है. वो बताया था, अरे भाई वो गेहूं का मामा है, ठीक है. वो गेहूं नहीं है, ठीक है, ऐसे बताया था. उस समय मुझे वो सारे बताया था. वो there will be some pigment in that particular Phalaris minor. Okay, we can easily distinguish between wheat and आपका गेहूं का मामा, ठीक है। अरे भाई वो गेव का मामा है। पिंक रूट तो पता नहीं, पिंक रूट वो तो मुझे पता नहीं। देयर विल बी ए anthocyanin पिगमेंटेशन इन दी स्टेम, ओके, इन फिलारिस माइनर। ठीक है, हम चलेंगे अभी गेव का मामा छोड़ देंगे, अभी थोड़ा फास्ट चलेंगे। ठीक है, टर्म अडेप्टेबिलिटी, स्टेबिलिटी, एक्लामेडेशन। ओके, these are the three terms always confusing. ठीक है, your adaptability is nothing but genetic ability to perform well in many environments, as I told. Okay, there will be one variety. Okay, we will release for all agroclimatic zones. For example, for southern Karnataka, Tamil Nadu region, for example rice, and for West Bengal, and for Haryana, Punjab region. That particular variety is performing in all the environments. How it is performing? It is there will be some adjustments, there will be some buffering. Okay, homeostasis. Due to those mechanisms, it is performing well in all other environments. That's we will call as the adaptability. Okay, it is well adapted in all the conditions. Okay, it is trying to give the yield, maximum yield. Not always maximum yield, but it will try to give that particular yield. Okay, that is we will call as adaptability. What about stability? Consistency of a performance across environments. Okay, that means in all other environments, okay, it is trying to give consistent performance. ठीक है, इधर 10 kg दे रहा है, उधर भी 10 kg दे रहा है, पंजाब में भी 10 kg रहा है, 10 kg दे रहा है, ये तो स्टेबिलिटी है। ठीक है, हमेशा हम स्टेबिलिटी के पीछे भागता है ब्रीडर्स। एज ए ब्रीडर, ओके, we will see the stability. We should need the stability. Okay, what about acclimatization? Okay, acclimatization is nothing but the physiological adjustment to a new environment. ठीक है, whenever the crop we got introduced from other countries, okay, initially it will try to adapt. Okay, it will try to adapt and it will change some physiological adjustment for the new environment. Okay, that's we will call it as a acclimatization. ठीक है, adaptability is nothing but the genetic ability to perform well in many environments. Stability is consistent performance. ठीक है, डिफरेंस तो बस इतना ही है। स्टेबिलिटी इज कंसिस्टेंसी। ठीक है, वो तो याद रखना है। मोड्स ऑफ रिप्रोडक्शन, द नेक्स्ट टॉपिक। ओके, सिंपल है, वो बेसिक मोड्स ऑफ रिप्रोडक्शन विल बी वेरी बेसिक फॉर एनी breeding programs. You should know the modes of reproduction and pollination of that particular crop, so that you can easily decide, okay, which is the breeding method we should adapt to that particular crop. If it is self, it is cross. Okay, maybe whether it is due to asexual reproduction or sexual reproduction. Asexual reproduction हो रहा है, for example in case of potato, we can take sugarcane, udhar cuttings where the reproduction will be vegetative reproduction. Okay, at that particular crop plants, okay, if you try for pedigree, bulk breeding, backrest breeding, वो तो नहीं चलेगा यार उधर। ठीक है, उधर कौन सा हम ब्रीडिंग मेथड यूज करना है? क्लोनल सिलेक्शन, म्यूटेशंस। ओके, उधर जाके हम वैरायटी डेवलप करता है, मैं कुछ सिलेक्शन करता है पेडिग्री में, क्रॉस करके F1, F2, F3 देखता है, वो तो नहीं चलेगा। फॉर दैट्स व्हाट एग्जैक्टली, for clonal propagated crop plants, for asexually propagated crop plants, for sexually propagated crop plants, there will be different breeding methods. Okay, if you know the basic things, बस आप इजीली से आप बता सकते हैं कौन सा ब्रीडिंग कहाँ यूज करना है। ठीक है, वही है ऑब्जेक्टिव इस पर्टिकुलर टॉपिक का। ओके, विल सी व्हाट एग्जैक्टली द मोड ऑफ रिप्रोडक्शन नाउ। क्रॉप प्लांट शो टू मेन मोड्स ऑफ रिप्रोडक्शन, वन इज योर एसेक्सुअल रिप्रोडक्शन, अनदर इज सेक्सुअल रिप्रोडक्शन। ठीक है, एसेक्सुअल रिप्रोडक्शन में क्या होता है? वेजिटेटिव रिप्रोडक्शन, न्यू प्लांट्स अराइज फ्रॉम द प्लांट्स पार्ट्स लाइक स्टेम, रूट, लीफ। ठीक है, योर अपोमिक्सिस, एम्ब्रियोस डेवलप फ्रॉम without fertilization. Okay, asexual reproduction is nothing but which is there is no fusion of male and female gametes. Okay, उसमें दो आता है, वेजिटेटिव रिप्रोडक्शन, अपोमिक्सिस। वेजिटेटिव रिप्रोडक्शन में न्यू प्लांट्स arises from the plant parts like stem से, root से, leaf से हम प्रोपोगेट कर सकता है, वो है वेजिटेटिव रिप्रोडक्शन। अपोमिक्सिस है ना, वो भी एसेक्सुअल है, मतलब देयर इज नो फ्यूजन ऑफ मेल एंड फीमेल गैमेट्स, okay, but there will be a formation of seeds in case of apomixis. In case of vegetative reproduction, there will be cuttings, the different things. But in case of apomixis, there is a production of seed, but without fusion of male and female gametes. That's why we'll call it as a asexual reproduction. Okay, how it will your embryo develop without fertilization? We will see. Okay, we'll see. Another is your sexual reproduction. Sexual reproduction is nothing but fusion of male and female gametes. It will create variation. Okay, or sexual reproduction will create variation. Natural vegetative again in vegetative reproduction, again natural and artificial. Okay, in natural vegetative reproduction, first one is your underground modified stem. Okay, underground में आपका stem है ना, वो modify होता है. Okay, where your food will store, okay, it will modified. Your stem is going to modify here. Okay, in this particular thing, underground modified stem stores food and have buds. These buds grow into shoots, form new plants. Okay, underground stem से ना उधर से buds आता है. For example, bulb, your onion, that is underground stem only. Okay, your garlic, वो bulb से आता है ना, garlic और onion में, वो stem में, ठीक है, वो modified होके उधर food store होता है. That is your onion and garlic will come in bulb. Your rhizome, your ginger and turmeric, टर्मरिक तो देखा है ना, ठीक है, वो भी अंडरग्राउंड मतलब साइल के अंदर आता है, मतलब मॉडिफाई होता है स्टेम, अंडरग्राउंड स्टेम, विल कॉल इट एज अ राइजोम. ओके, यू आर इन जिंजर एंड टर्मरिक. कार्म, योर बुंडा, कोलाकेशिया एंटिक्वोरम एंड अर्बी, कोलाकेशिया एस्कुलेंटा, दोस आर विल कॉल देम एज ए कार्म, दोस आर आल्सो योर अंडरग्राउंड स्टेम. देन सब एरियल स्टेम्स, there will be some aerial parts, they will expose to aerial parts. These modifications grow near the soil surface and root at specific intervals and forms new plants like runners, your strawberry, okay, stolons, mint, suckers, banana and date palm. If you see the banana, okay, there will be some different seedlings, we'll call them as a actually suckers. Okay, they will comes in the along with the main stem. We will separate, we will dig that from the main parent and we will separate as a separate parent. So if you plant that particular sucker, then again it will grow like a main banana plant. Okay, that we will call as a suckers. Subaerial, those are subaerial stems. There will be some exposure to the out of the soil. Okay, bulbils, these are modified flowers. Okay, from flowers there will be some modified structures that directly grow into new plants without forming seeds. These developments does not involve fertilization. Okay, that's why we are calling it as a vegetative structures. In garlic, okay, in garlic, the lower flowers of the inflorescence naturally forms bulbils. Okay, flower structure will transfer into the new buds. We will call them as a bulbils. Okay, sword sucker in banana, good. Sinchan, water sucker, yes, water sucker. In pomegranate, we will call them as water suckers. Again, there will be some suckers will comes out of the in the pomegranate plants also, but we will not use them as a propagating material. We will cut them. We will remove them. Okay, agave, good. Pineapple have crown, sucker, slip. Yes, there will be so many different vegetative propagation. Okay, these are some different, the main important natural vegetative propagation. What about artificial? Okay, here there is no natural, we are doing it. Okay, your cuttings, stem cuttings, sugarcane, grapes, roses, and your budding will also take buds. Okay, budding again in the roses. Okay, the root cuttings, sweet potato, citrus. Okay, will take roots and will propagate them. Okay, layering and grafting, widely used in fruit trees. Okay, mango and apple. Okay, what we will do? This is your stem. Okay, we will remove the bark of this particular region and we will give some media so that there will be a formation of roots. Then we will separate it and we will try to make a new plant. ठीक है, वो तो वही है ना आपका layering and grafting. ठीक है, pomegranate में ज्यादा करता है. And mango and apple. Okay, tissue culture, this is modern micro propagation technique. Okay, where it is used for rapid and disease free resistance plants. Okay, we will take some 0.5 mm meristem and we will try to grow in a means in artificial media. Okay, we'll try to provide everything artificially. That is your tissue culture. Okay, these are some artificial vegetative reproduction. What are the significance? It will maintain genetic stability. That means it produces clones. Clones are nothing but the no fusion of gametes, and your offsprings are similar to your parents. Okay, in case of genotype and phenotype. Okay, those we call it as a clones from this single mother plant. We will make them some more than 1500 individuals, offsprings. Every individuals are similar. Okay, that's why we'll call them as a clones. We can maintain the genetic stability. Okay, clones are heterozygous, homogeneous. वही है, एक बात बताता हूँ. ठीक है, clones are nothing but the same offsprings as it is of mother parent. Okay, if mother parent is homozygous homogeneous, then your clones are also homozygous homogeneous. If mother plants are heterozygous and homogeneous, then your clones also heterozygous heterogeneous. Okay, but why we will always call clones are heterozygous and homogeneous? Will these clones, that this particular vegetative reproduction, okay, will only use in case of asexual reproduction crop plants. Okay, asexual reproduction as crop plants are nothing but your ornamentals like fruit crops. Okay, the horticulture crops. Okay, those are like heterozygous condition only. Okay, those are actually cross-planted crop plants, but in nature, okay, there is less chance to make them as a cross-planted crop plants. Okay, they have adopted the asexual reproduction. Okay, like this budding. Okay, the natural ones. Okay, subaerial stems. Okay, the modified stems. Okay, they have adopted their own propagation methods. Okay, since the particular crops, okay, where we will produce clones are heterozygous and homogeneous, your clones also heterozygous and homogeneous. Yes, they are immortal. They are immortal, nothing but they are always maintaining genetic stability. Okay, nothing but they are maintaining genetic stability. There is no mixture of female and male gametes. That's why they are highly stable and immortal. Means they are not going to die. That means they are not going to die. Not going to die means they will maintain their stability, and they are the true to type. They will maintain the true to typeness. Maintenance of heterozygosity. Again, okay, since our asexual reproduction crop plants are heterozygous in nature, it will be maintained always. Rapid multiplication. Okay, utilization of bud mutation. Utilization of bud mutation is nothing but if any single branch of a plant develops into beneficial mutation, okay, in the big tree, okay, for any particular fruit crop, if any single branch got mutated due to some reasons, okay, it can be cut and propagated vegetatively to create new entirely different variety. ठीक है, ये भी मतलब कर रहा है हम लोग. ठीक है, ब्रीडिंग में वो भी एक सिग्निफिकेंस है. ठीक है, essential understanding before we are going into the apomixis. Okay, you should know the basic thing. ये तो फ्लावर है. ठीक है, उसमें आपका गाइनोशियम एंड एंड्रोशियम आएगा. ठीक है, एंड्रोशियम में क्या आएगा आपका स्टेमन, देन योर एंथर. This is very important. Okay, as per our case, in case of breeding, and this is gynoecium, stigma, style and ovary. This is very important. ठीक है, ये ovary है. ठीक है, देखो इधर what exactly the ovary is. This is ovary. In particular, each and every ovary, there will be so many number of ovules. This is ovule, one ovule. Okay, there will be a number of ovules. Okay, at this particular ovule, okay, you can see this ovary will got develop into fruit, and your ovule within the particular ovule, there will be so many ovules. As I told, those ovules will develop. This is the particular ovule. These ovules will develop into the seeds. Okay, for example, now you can imagine that, okay, this is orange flower. ठीक है, as I told, your ovary है ना, ये ovary, ये तो fruit, it will develop into fruit. ठीक है, your orange contains so many seeds. As I told, those are from ovules. Okay, each ovule will convert into seed. Okay, you can see this is the ovule. This is chalazal end. One end will be chalazal end, another one will be micropyle end. Okay, where there is entry of pollen, we will call that as a micropyle end. Okay, and the nucellus, the nourishing, this is nourishing cells for the embryo sac. Okay, this is your embryo sac. Okay, where exactly your zygote will form. Around this there will be nourishing cells, that we will call them as a nucellus, and the outer boundary for nucellus there will be integument cells. This is embryo sac. Okay, ये तो important है इतना. ठीक है, this is the structure of ovule. Next, we will see what exactly apomixis is now. Anyway, you know, now you got the what exactly ovule, how it will be. Ovule is, there will be number of ovules in the ovary. Okay, each ovule will produce each particular seed. Okay, the term apomixis is derived from the Greek word, okay, apo, without, and mixis, sexual or union or mixing. Okay, apomixis is nothing but without mixing of gametes. Apo is away, okay, away or without mixing. Mixis is mixing, that is your male and female gametes. Without mixing male and female gametes, there will be a production of seed. Okay, it refers the development of an embryo. Embryo will enter with nothing but seed. Okay, and seed without the fusion of male and female gametes. How we are differentiated it? Okay, your apomixis, ampemixis, and apomixis. Ampemixis is nothing, mixing of your male and female gametes. That is your sexual reproduction. Apomixis is a sexual reproduction. Okay, apomixis is nothing but a sexual reproduction. Within that apomixis, okay, there will be agamospore and vegetative reproduction. Broadly, your apomixis will also covers the vegetative reproduction. But, okay, but in the general explanation, okay, we will remove this vegetative reproduction as a separate thing and apomixis as a single part. But, okay, but as the scientific articles, your apomixis also contains vegetative reproduction. One is vegetative reproduction and another one is agamospermy. Okay, agamospermy is broadly, we will call it as a apomixis. Okay, then your agamospore, that is your apomixis, recurrent and non recurrent. Recurrent में क्या होता है, आपका सीड डेवलप होते हैं ना, सारे डिप्लॉयड कंडीशन में रहता है. So your recurrent will be, everything will be diploid condition, so that it will pass into generation to generation. So your progenies will come again, there will be a next progenies. Okay, due to diploid condition there will be continuation in the life cycle. Okay, that is your recurrent. But in non recurrent, okay, they will end up with applied, okay, applied plants, so that it will be for only one generation, there will be no continuation of generation. That is your non recurrent. Okay, in recurrent apomixis there will be two types, adventitious embryony and gametophytic apomixis. What is a adventitious embryony? Okay, adventitious embryony is nothing but there will be a direct, okay, there will be a direct formation of embryo from the somatic cell, there will be embryo formation. So there is no intermediate gametophytic formation here. We will also call this adventitious embryony as sporophytic. Okay, we will also call this particular adventitious embryony as sporophytic. Okay, that means there is no intermediate gametophytic. Gametophyte क्या होता है, आपका embryo sac होता है gametophyte, that is in the natural sexual reproduction your embryo sac will be gametophyte. But in case of adventitious embryony there is no formation of embryo sac. Directly your somatic cells will convert into embryo. Yeah, your somatic cells are nothing but nucellus. Okay, from those cells there will be a formation of direct embryo. Okay, but in case of gametophytic apomixis there is a intermediate embryo sac formation. Okay, from your megaspore mother cell or any other somatic cell, okay, there is a formation of embryo sac first. Then that particular embryo sac, okay, you can see gametophytic apomixis, there will be two types. Okay, then particular embryo sac will convert. Based on that embryo sac, there will be a different. You can see here in embryo sac, there will be a synergids, okay, synergids, antipodal cells, polar nuclei, okay, and egg cell. There will be egg cell here. Based on that, on from which cell, from which embryo sac cell, there will be development of seed. Okay, again based on that, there will be a different term. Okay, parthenogenesis, apospory. Okay, but now, okay, not apospory, apogamy. But from which cell your embryo sac is forming, that is based on these two terms, apospory, diplospory. We will see here. Okay, your apomixis is contains your vegetative propagation and agamospory. Okay, agamospory. Agamospory is nothing but again we will call it as apomixis. No problem. Okay, your agamospory is two types: adventitious embryony and gametophytic apomixis. Your adventitious embryony also we will call it as a sporophytic apomixis. ठीक है? अच्छे से देखो, इंपॉर्टेंट है apomixis. इतना इजीली क्लासिफिकेशन कोई नहीं बता सकता है. ठीक है? Now, adventitious embryony, gametophytic apomixis. What I am telling, your adventitious embryony or sporophytic apomixis, it will start with the sporophyte from the diploid cell, okay, somatic cell. Again, it will end up with sporophyte directly. There is no intermediate gametophyte formation. Your gametophyte is nothing but embryo sac. Okay, we will see in the sexual reproduction how your embryo sac will form each and everything. But at this particular point of time, you should know, okay, there is no embryo sac formation in case of adventitious embryony. From direct somatic cells, for example, your nucellus, you can see from these nucellus cells, okay, these cells, there will be a formation of embryo. That we will call it as a sporophyte. Okay, this is responsible for polyembryonic condition. Okay, from each ovule, as I told, there will be one seed. So from one seed there will be one seedling will come, but due to this nucellus they are converting into embryos, there will be from each ovule there will be a multiple embryos, polyembryonis. ठीक है, इसलिए ये polyembryonic condition cause करता है. इस किसमें? For citrus, mango. Okay, you can see from each citrus seed or mango seed, sometimes you will get 2 or 3, 4 seedlings. ठीक है, 2, 3, 4 seedlings आता है ना, किस लिए आता है? ये polyembryonic condition से आता है. ठीक है, there will be embryo formation from nucellus cells, so that there will be a more number of embryos in the single seed. But generally, single seed contains single embryo. When there is a normal sexual reproduction, okay, the male and female gametes will merge and there will be embryo formation in particular ovule. So you will get single embryo. So single seed, single embryo, single seedling. But in this in particular adventitious embryony, your nucellus cells around that particular embryo sac will form the embryos. So there will be so many chances. So you will get a polyembryonic condition. ठीक है, ये तो adventitious embryony है. मेडिका को बता रहे embryo sac only from the MMC. Yes, embryo sac can form. Generally, it will form from the megaspore mother cell, medicago. But in apomixis, it is different. You can see here. No, gametophytic apomixis is there is a intermediate gametophyte formation. Gametophyte formation is nothing but this embryo sac formation. In adventitious embryony, ठीक है, ये नहीं फॉर्म होता है, इधर से डायरेक्ट न्यूक्लियस सेल से ना, इधर से आपका एम्ब्रियोस आता है. ठीक है, बट in gametophytic apomixis, your sporophyte, okay, there will be two process. From archisporium cell, that is your megaspore mother cell, there is a megaspore mother cell in case of your ovary. Okay, that is, we'll also call it as the acrosporium cells. From if your gametophyte develop from this acrosporium cell, okay, we will call it as a diplospory. If your gametophyte is forming from somatic cell, any other cell of your ovule, any other cell matlab from nucellus or any other integument cells, okay, then we'll call it as a apospory. In gametophytic apomixis, there is a formation of embryo sac, but from which cell there is a formation of your embryo sac? If it is normally from megaspore mother cell, okay, megaspore mother cell, that will be in diploid condition again. So you will get again, if it is producing diploid embryo sac, you should remember here it is recurrent apomixis. So you will get diploid embryo sac. Normally in sexual reproduction you will get a haploid embryo sac, but here you will get a diploid embryo sac. If it is from a megaspore mother cell, that is acrosporium cell, you will call it as diplospory. If it is from the somatic cell, any other than your MMC, okay, maybe from your nucellus or integuments or any other cells, we will call it as a apospory. Okay, आपको gametophyte तो form हो गया अभी. Based on the gametophyte formation, you will call it as a diplospory, apospory. Then in that particular embryo sac, okay, there will be a synergids, there will be antipodals, and there will be a egg cell. Okay, if your sporophyte, that is your embryo, if is forming from egg cell, then we will call it as a parthenogenesis. Okay, parthenogenesis. If it is from other than egg cell, okay, then we will call it as a apogamy. Okay, itna hi. Your diplospory and apospory will related to formation of embryo sac, that is your gametophyte, but your parthenogenesis and apogamy will tells that from which cells of embryo sac is producing your embryo. If it is from egg cell, parthenogenesis. If it is from other than egg cell, that is your synergids or antipodals, then we will call it as a apogamy. And this apogamy we will also call it as a, okay, somatic parthenogenesis. Okay, sorry, gonial. Gonial is your egg cell, from the egg cell, and that is from somatic parthenogenesis. That is your apogamy. Clear? We will discuss syngamy, everything. Okay. Synergid, apospory. Yes, the synergids and antipodals, that we will call it as apospory. Sorry, apospory. अरे apospory नहीं, ये apogamy. ठीक है. Apospory and diplospory are the terms related to formation of your embryo sac. ठीक है. Then after embryo sac formation, there will be a formation of embryo from that cells of embryo sac. If it is from egg cell, then we will call it as a parthenogenesis. If it is from other than egg cell, then we will call it as a apogamy. ठीक है. Yes, good. Okay, can we move? This is what exactly they are telling. Types of recurrent apomixis under recurrent apomixis. Okay, adventitious embryony, that is also called it as a sporophytic apomixis. In this mode, embryos are developed directly from the diploid vegetative sporophytic cells, other than means cells of the ovule, such as nucellus, integuments, or chalaza. Okay, bypassing the formation of embryo sac entirely. Okay, from nucellus, from integuments, from chalaza cells. Okay, without passing the embryo sac formation, okay, there will be a formation of embryo, directly embryo, genetically identical to the parent. Okay, there will be a adventitious embryony. The polyembryony condition will appear there only. Okay, the characteristics which are where multiple embryos developed within the single seed. Okay, example mango, citrus, and orchids. Okay, your gametophytic apomixis, there is an intermediate gametophyte formation. Here an embryo sac is formed, but it is unreduced, it is diploid. Okay, if it is applied, then it is a non-recurrent apomixis. It is recurrent apomixis. because normal meiosis is either modified or omitted. ठीक है, there is a process of apomeiosis. We'll call it as a apomeiosis. There is a different number of genes again due to for the apomeiosis condition, but there is no requirement. Okay, it is not important for you guys. We will call that as apomeiosis. You should know. Oh, there is a diploid unreduced embryo sac formation from the MMC. Okay, generally there is a reduced embryo sac formation. Okay, we'll discuss in sexual reproduction, but there is a condition apomeiosis. In that particular, this apomeiosis, it is related to this gametophytic apomixis. Okay, the reason for gametophytic apomixis, there is a two pathway, apospory, diplospory. The diploid embryo sac developed from somatic cells of the ovule. Somatic cells of the ovules are nothing but your nucellus, integuments and chalaza. From those cells, if there is a formation of embryo sac, then we will call it as apospory. From the embryo sac is developed from the megaspore mother cell, that is your archisporium cell, cell. If you are developing that particular embryo sac from that megaspore mother cell, then we will call it as a diplospory. But in adventitious embryony, there is no formation of an embryo sac. From the somatic cells, directly it will form embryo. That's why you can see more embryos within the seed, polyembryonic condition. But here, there is a only formation of particular embryos. If it is embryo sac, there is formation embryo sac first. If it is from somatic cells, then we will call it as apospory. If it is from megaspore mother cell, then we will call it as a diplospory. You should remember the examples: Aeraceum, Malus and Orchids in case of gametophytic apomixis, Taxum, Taraxacum and Tripsaxum, example for diplospory. Okay? Then the pathways of embryo development, based on pathways of embryo development. Okay, you can see here, this is based on the whether an embryo sac is formed or not. If it is embryo sac is forming, then gametophytic apomixis. If there is no embryo sac, adventitious embryony. Okay, based on which cell, from which cell your embryo sac is forming, your apospory and diplospory. Then after embryo sac formation तो हो गया, ठीक है, apospory और diplospory. Through apospory और diplospory से embryo sac तो formation हो गया है अभी embryo development कैसे embryo develop हो रहा है, उसके हिसाब से again there will be a different classification. You can see here, parthenogenesis. The embryo develops directly from the egg cell without fertilization. As I told, there will be embryo sac. Okay, there will be a 3 cell here, there will be a 3 cell here, there will be a 2 polar nuclei here. Okay, antipodal cells, synergids, and this is your egg cell. This is your egg cell. Okay, these are polar nuclei. The embryo developed directly from the egg cell. From the egg cell, if your embryo is getting developed, then we will call it as a parthenogenesis. Okay, your gonial parthenogenesis from the egg cell and somatic parthenogenesis from the other diploid cell within the embryo sac. Okay, we will also call this as a apogamy. Okay, if anything other than egg cell, okay, from these cells, from antipodal cells or synergids, okay, we will call it as a somatic parthenogenesis. Otherwise, apogamy. Same, both are same. Okay, the embryo developed from the cell in the embryo sac other than the egg cell. Then pseudogamy, a unique state where pollination is required to stimulate the endosperm development. Okay, for this particular endosperm development, okay, the polar nuclei will turn into endosperm. To that, there is a requirement of mimicking pollination. Okay, there is a to stimulate the endosperm via fertilization of the secondary nucleus. Okay, there is a fusion of another gamete, okay, from this particular male gamete, so there will be formation of secondary nucleus. Okay, ये तो चाहिए, but the egg cell itself remain unfertilized. Your egg cell will not fertilize. That is your pseudogamy. Okay, where there is a requirement of pollination, okay, to convert your egg cell into embryo. That is your pseudogamy. But semigamy क्या होता है? Semigamy is rare form of apomixis where sperm cell enters the egg cell. In pseudogamy, there is no enter of sperm cell, but in case of semigamy, your sperm cell will enters the egg, but fails to fuse its nucleus with the egg nucleus. Okay, it will come here and it will fuse with this particular sperm cell, but the nucleus of both sperm and egg will not fuse. Okay, instead of forming normal hybrid, both male and female nuclei divide independently. Okay, both your male and female nuclei divide independently and they will form the chimera. Chimera is the thing, but in the single plant there will be two different genetic constitution. Okay, independently within the same embryo, this results in a genetic mosaic or chimera. Okay, it is technically considered as apomixis because there is no fusion of male and female gametes. Okay, cotton example is cotton for semigamy. Okay, how your embryo will develop from the embryo sac cells, that is your different classification: parthenogenesis, apogamy, pseudogamy and semigamy. Okay, अपोस्पोरी एंड एडवेंशियस एम्ब्रियोनी डिफरेंस. अरे भाई क्या डिफरेंस पूछ रहा है. अपोस्पोरी में एम्ब्रियो सैक फॉर्म होता है, एडवेंशियस एम्ब्रियोनी में एम्ब्रियो सैक ही नहीं आता है. मैं वही डिफरेंस बता रहा है ना. फॉर ICAR PhD आल्सो. अरे भाई हम ऑलरेडी कवर किया है ICAR PhD के लिए क्लासेस. हमारे वेबसाइट में जाओ, देयर विल बी सो मेनी कोर्सेस. ओके, यू कैन इजीली गेट सो मच इंफॉर्मेशन. Explain about submitting of submitting inbreeding depression. अरे मोहन अभी कौन सा चल रहा है? आपको हाइब्रिड का वैरायटी पूछ रहा है. सब आएगा मैराथन में. ठीक है, थोड़ा पेशेंस में रहना है मोहन. आप सब क्लासेस अटेंड करना है, इसलिए आपको सब पता चलेगा. सबमिटिंग ऑफ सबमिटिंग सब बता देंगे मैं. वो तो आना चाहिए ना कांसेप्ट. बीच में घुसना है क्या? सिनर्जिट्स, एंटीपोडल्स, एम्ब्रियो सैक, अपोस्पोरी. फिर से पूछ रहा है, 2-3 बार पूछ रहा है आप. एक्सप्लेन अबाउट सबमिटिंग ऑफ सबमिटिंग. कहाँ से मोहन बताओ आप. सूडोगैमी एग्जांपल. सूडोगैमी मतलब ऐसे पर्टिकुलर क्रॉप तो नहीं है. देयर विल बी सो मेनी इन सो मेनी क्रॉप्स यू कैन ऑब्जर्व दैट पर्टिकुलर phenomenon. Okay, not fertilized egg in both semigamy and yes, there is no fertilization in both semigamy and pseudogamy. Yes, medicago. ठीक है. सिंचन कुछ डाल रहा है क्या? But how to detect the detection of apomixis in crop plants? How we can detect the apomixis? If it is obligate apomixis, okay, you can see the similar offsprings. You can see if it is obligate apomixis, what are the progenies you are getting? They are very similar. Since there is no fusion of male and female gametes, obligate means it is completely apomictic. Whatever you are getting, the embryo is from somatic cell. There is no meiosis, there is apomeiosis. Whatever the genotypes, offsprings you are getting, they are very similar to parent crop plants. So you can easily identify, oh yeah, there is apomixis. What about facultative apomixis? Out of some progenies, okay, 50% or there will be some 50 to 60% progenies will be similar to your parent plants. So that there is a facultative apomixis. No inbreeding depression after selfing. Okay, where you will mostly see apomixis in if you take any cross-pollinated crop plants. Okay, if there is an selfing, self-pollination is happening, if there is a fusion of male and female gametes. Okay, when you do the selfing, there is a exposure of recessive alleles, then there will be inbreeding depression. Okay, but if it is apomixis, there is no inbreeding depression, there is no sexual reproduction, so you can see the inbreeding depression. So there is no inbreeding depression. And polyembryony, as I told, okay, if you are seeing polyembryony, that is exactly due to apomixis. Okay, these are different methods how you can detect the apomixis. अरे फिर से मोहन डाल रहा है क्या? Apogamy, अरे obligate only by apomixis, but facultative is sometimes sexual the reproduction occurs. Yes, visually. Sir, now how many slides remaining? Because I am very hungry. अरे खाना लेके आ जाओ बेटो मेरे साथ, अभी मैं भी खाना नहीं खाया है अभी तक, ठीक है सर? how many? Okay, by using morphological markers too. Morphological markers nothing but those are very similar. Okay, if there are similar to your crop plants, be your parents, then you can see na why you need again morphological markers. Done sir, ठीक है। सिंचन जाओ 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 खाना लेके आ जाओ। मैं अभी 64 67 चल रहा है और 20 स्लाइड से we will crisply we will see males retained self incompatibility, that's it, ठीक है। how we can develop apomictic lines? Okay, we can transfer genes from wild species. Okay, there are some different species we have transferred it, the genes which are responsible for apomeiosis. Okay, the genes which are responsible for apomeiosis are responsible for apomixis. Okay, we will transfer those genes so that we can develop apomictic lines. Okay, there are some examples. Okay, in case of maize from Tripsacum dactyloids, we have transferred it. In pearl millet, Panicidum squamalatum, in beta species, sugar beet, in Elaemus reticulus, in wheat we have transferred it. There will be some induced mutation in sorghum. Okay, there is some current event. Okay, in sorghum and bajra, they are trying to develop apomictic lines. Okay, for the fixation of heterosis. Okay, once you will develop hybrids, okay, then you introduce apomixis genes, then there is a continuous apomixis. Once you develop hybrid, then there is no need to maintain it. Okay, if it is apomixis, it will automatically you can maintain hybrid, right? Okay, hybrid is nothing but capital A small a. Okay, you will develop hybrids. Then if it is self pollination, okay, there is segregation. Every year you should produce hybrid seeds and you should give farmers. But if there is a apomixis, okay, once you develop hybrid, then what will happen? It will maintain this particular genotype since there is no fusion of male and female gametes. Okay, whatever in the parent, okay, it will pass through megaspore. Then your megaspore will go to apomeiosis from diploid to diploid again. So from AA to AA, so you can maintain, right? This is what they are trying to do in bajra and in sorghum. Okay, at this particular time, the recent paper they also published it. Okay, how we can maintain this in sorghum, how we can use apomixis for the maintenance of hybrid. Okay, then interspecific hybridization, you can develop apomictic lines through the cross between, like example, wheat and oat, barley and wheat. Okay, there's some crosses they have identified this particular hybridization. Okay, then application of apomixis, as I told, fixation of heterosis. You can fix the heterotic combination. As I told, you can fix this capital AA condition for the multiple generation. If it is apomixis, so there is no fusion of male and female gametes, so you can maintain. This is the application, one of the application. Okay, production of homozygous lines. How we can produce homozygous lines? Okay, you will first, what you will do, you will isolate haploids through apomixis, through non-recurrent apomixis. Okay, you will get haploids from the non-recurrent apomixis. Okay, your egg cell or any other cells will develop into the complete plant. Those are haploids. You can doubling the chromosome of that particular haploid. So if your plant is capital A, your haploid gamete will be capital A, then your plant will be capital A. Then you can double it, capital A, capital A. Okay, you can produce homozygous lines. Okay, production of hybrids. Hybrids are nothing but crossing two facultative apomictic plants. Obligate apomixis में तो हम सेक्सुअल रिप्रोडक्शन ही नहीं है, ठीक है, कंप्लीट ऑब्जेंस, बट फैकल्टी apomixis में हम कुछ सेक्सुअल रिप्रोडक्शन भी देख सकते हैं ना, क्रॉस पोलिनेशन भी कर सकते हैं. यू कैन क्रॉस बिटवीन टू facultative apomictic plants. Okay, we will call it as a hybrid. We will call that particular thing as a hybrid. Hybrids we can use where, okay, in case of some asexual reproduction plants where there is a complete impossible to produce the normal hybrids at that particular time where there is a mixture of this particular apomixis also. There you can use this faculty of apomixis. You can form a hybrid, okay, so that you can use that in a crop improvement program. Those hybrids are maybe they are having high vigor, okay, they are having high yield, okay, resistance to disease and pest. Okay, this is the different applications of apomixis. Okay, one is main application is fixation of heterosis. ठीक है, सेक्सुअल रिप्रोडक्शन हम अपोमिक्सिस देख लिया, वेजिटेटिव प्रोपगेशन तो देख लिया, ठीक है। थोड़ा मैं आप 2 मिनट मुझे टाइम दे दो, आपको किसी को खाना है तो आप लेके आ जाओ बेटा, मैं 2 मिनट में आएंगे, थोड़ा एनर्जी ड्रिंक लेके आएंगे। ಆಗಿಲ್ಲ ಒಂದೇ ದಿನ ನಾನು ಇಲ್ಲ I want to eat sir. अरे आवाज आ रहा है ना मेरा, बताओ कोई। एडवेंशियस एम्ब्रियो एंड अपोस्पोरी डिफरेंस, अरे मैं बताया था ना, इन बोथ नुसेलस एंड इंटेगुमेंट्स ओनली कन्वर्टेड इनटू एम्ब्रियोस। अरे सोमन गौड़ा, कोई बताओ इसको आंसर बताओ कोई। ही इज टेलिंग एडवेंशियस एम्ब्रियो एंड अपोस्पोरी डिफरेंस पूछ रहा है, इन बोथ नुसेलस एंड इंटेगुमेंट्स ओनली कन्वर्टेड टू एम्ब्रियोस, ऐसे बता रहा है। कोई बता सकते हैं क्या इसको, मतलब आंसर। He is telling in both nucellus and integuments, yes, it will start from both nucellus and integuments only in adventitious embryo and apospory. Okay, I will agree for that. He is telling only converted to embryo sac. बताओ कोई बताओ आंसर। ओके कोई बताया ना, एडवेंशियस स्पोरोफाइटिक नो एम्ब्रियोसैक फॉर्मेशन, ठीक है। आज साजन बता रहा है अपोस्पोरी अंडर गेमेटोफाइटिक एम्ब्रियोसैक इज फॉर्म्ड ब्रदर विजयाली, एडवेंशियस एम्ब्रियोनी इज नॉट। मैं इतना बार बता दिया, there is absence of embryo sac in adventitious embryony, that's why we'll call it as adventitious, it is skipping the embryo sac formation। अरे भाई सोमन गौड़ा, कहाँ से आप सोमन? किसी को पूछा था ना मैं कहाँ से। सोमन गौड़ा, मोहन, हाँ मोहन को पूछा था। कहाँ से मोहन? मोहन आप बताओ कहाँ से? मोहन बताओ। सिंचन कहाँ लेके आया क्या? ओके नेमानी, आपका नाम उधर साजन आ रहा है इसलिए मुझे कैसे पता चलेगा नेमानी। नाइस में स्लाइस, अरे स्लाइस नहीं है, वो एनर्जी ड्रिंक है, ठीक है, ठीक है। सेक्सुअल रिप्रोडक्शन, नाउ वी हैव सीन, हेलो, मेरा आवाज आ रहा है? बताओ, कोई बताओ, आवाज आ रहा है क्या सही से? All good. Okay. Now we will see sexual reproduction. Okay. There is a formation of sporogenesis. First, there will be formation of spore. Then formation of gametogen, means gametophyte. First, there is a, in both male and female side, we need spores. Okay. Then those spores will convert into gametophyte. Male gametophyte, female gametophyte. The formation of spore, we will call it as a sporogenesis. Then formation of gametophyte from that particular spores, we will call gametogenesis. ठीक है, विजुअली हिंदी डिडंट नो सर, यस कंटिन्यू, सब लाइक कर दो सर को, सेंचुरी हो जाएगा। अरे मैं अभी 60 हो रहा है, सो यू आस्क्ड द क्वेश्चन, आई डिडंट आंसर। ठीक है, सबको शेयर कर देना, किसी को सोने सोने मतलब, सो यू आस्क्ड द क्वेश्चन, आई डिडंट, ठीक है कोई दिक्कत नहीं, ठीक है, विल सी, सबको शेयर कर दो अभी, ठीक है, उठा दो, नींद से उठा दो सबको। स्पोर, ठीक है, द फॉर्मेशन ऑफ स्पोर will sporogenesis, then formation of gametophyte, that particular from particular spore we will call it as a gametogenesis. Sporogenesis is the process of forming applied spore from the diploid mother cell. Now we are into the sexual, proper sexual reproduction, okay, not like apomixis. In apomixis there is a formation of diploid spore from the diploid mother cell, but here ऐसा नहीं है, ठीक है, ये तो पूरा commercial sexual reproduction, ठीक है, process of forming applied spore from the diploid mother cell. This is the stage where meiosis occurs, normal meiosis, there apomeiosis, reducing the chromosome number by half. First there is a meiosis, so from the particular parent cell there will be formation of spore through the meiosis, then that particular spore will convert into gametophyte. In sporogenesis there will be two types, microsporogenesis and megasporogenesis, ठीक है, the formation of microspore within the anthers, that we will call it as a microsporogenesis. The formation of the development of pollen grains, okay, वही है आपका microsporogenesis, ठीक है, diploid pollen mother cell convert into 4 applied microspore, 4 different pollens, that is your microsporogenesis. Megasporogenesis is formation of megaspore within the ovule, that is your diploid megaspore mother cell will convert into 4 applied megaspore, that is your megasporogenesis, okay, but out of 4 megaspore only usually one will survive, all the other 3 are degenerate, but in case of microsporogenesis 4 pollen grains will produce. Then from that pollen grain, okay, there is a microgametogenesis. Again, how it will happen, the process where microspore develop into a mature pollen grain, there will be particular microspore formation, then that microspore will develop into pollen grain, okay, that is male gametophyte, okay, eventually produces male gametes. Here in case of gametogenesis there is a mitosis, ठीक है, mitosis will occur, how it will occur we'll see. In megagametogenesis that particular megaspore will convert into, okay, female gametophyte, that is your embryo sac, ठीक है, first there is a formation of megaspore, then that megaspore will convert into embryo sac, okay, we'll see in the diagrammatical way so that you can understand easily. This is the megasporogenesis and megagametogenesis, okay, that is female part, okay, in female part how it will develop, it will start, this is your gynoecium, as I told, ovary will convert into fruit, your ovule will convert into seed, each ovule convert into each seed, ठीक है, if you take the longitudinal section of your ovule, this particular ovule, you can see there is a megaspore mother cell, for each ovule there is a megaspore mother cell, ठीक है, that particular megaspore mother cell will be in 2n condition, there is a first meiotic division, so that you will get two spores. First meiosis, there will be in meiosis, there will be a meiosis 1, meiosis 2. There is a second meiotic division. Okay, there is nothing but your mitosis. Second meiosis is nothing but mitosis. Okay, the NN condition will get your 4. This is normal meiosis. Okay, this is process is normal meiosis till this. You will get 4 different megaspore. ठीक है, उसमें ना 3 degenerate होता है हमेशा female in crop plants. Okay, there will be a in normal all angiosperms and gymnosperms. Okay, there is a degeneration of this particular 3 megaspores. There is only 1 megaspore will be active. Okay, functional. This is megasporogenesis, formation of megaspore from megaspore mother cell from 2n to n. Then this n will maintain as a n. If your n should maintain as a n, then there is a mitosis. This particular megaspore will convert into first mitosis, then 2 cell, then second mitosis 4 cell, then third mitosis 8 cell. Okay, there should be 3 mitosis. Then there is a formation of your megagametophyte. That is nothing but your embryo sac. Okay, you can see antipodal cells. There will be out of 8 cells, 3 will move antipodal cells, 2 will be remain in the central. Okay, that is your polar nuclei. Okay, what will happen? This will merge. Okay, and make it as a single cell and 2 nuclei. Then egg cell, there will be a egg cell in the center and synergids. Okay, and we will call it as a 8 nuclei, 7 cell, 8 nuclei stage. Okay, why we will call it as a 7 cell, 8 nuclei stage? Okay, 3 cell and 1, 6, 7 cells. But nuclei, this will not merge polar nuclei. There will be 2 nuclei. So there will be a 2 nuclei. Okay, means 2 meiosis and 3 mitosis in female gamete formation take place. Yes, Shinchan. Like how many mitosis or meiosis required to form 100 seeds? अरे वो तो पहले मेगा गैमेटो मतलब मेगास्पोरोजेनेसिस गैमेटोगैमेटिस खत्म हुआ, अभी माइक्रो मतलब देखेंगे, उसके बाद पता चलेगा कितना 1000 सीड्स के लिए कितना माइटोसिस और मियोसिस चाहिए. ठीक है, वो गया. ठीक है, देन now you can see here. This is about female part. Now we are seeing male part. Your anther, if you take the cross section of this particular anther, you can see like this. You can see there will be polar mother cell in each part of your 4 different parts of your anther. That particular polar mother cell, okay, in 2n condition. So there will be a 1 meiosis. Okay, that is your first meiotic and second meiotic division. You can see there is a pollen grain from 2n to n. This particular pollen grain, you can see that is, we will call it as a microspore. Again, one mitosis will happen. So there will be a generative nucleus and vegetative or tube nucleus. We will call it as a binucleate pollen. Then this will again mitosis will happen. Okay, in the generative nucleus, only in generative nucleus. Okay, you can see it is trinucleate pollen. Trinucleate. There will be 3. 1, 2, 3. There will be a 2 sperms. Okay, from the generative nucleus, 2 sperms, and your vegetative nucleus, just it will, we will call it as a tube nucleus. Okay, it will give the narration to the pollen tube growth. ठीक है, ये तो it will disappear after pollen tube germination. Okay, out of 2 sperms, one will fuse with your egg cell, another will fuse with your polar nuclei. Okay, whatever will fuse with your egg cell, we will call it as a, what we will call, that is your primary fertilization, and your secondary fertilization, that is your with polar nuclei. Okay, that's why we'll call it as a, both will call it as a triple fusion or syngamy. Yes, we'll call it as a syngamy. Okay. 60% 2n. अरे अभी बताओ, कोई बताओ, एक पूछ रहा था, for 100 seed formation, how many mitosis or meiosis required? Syngamy is nothing but male and female. Normal sexual reproduction is your syngamy. ठीक है? And triple fusion is your fusion of polar nuclei with the another sperm. That is your triple fusion. Okay, we'll consider these two will commonly called as, okay, including these two will commonly called as double fertilization. ठीक है? Your syngamy plus triple fusion. Your syngamy is fusion of male and female gametes, normal embryo formation. Your triple fusion is fusion of polar nuclei with male gamete, another male gamete. Okay, both we will call it as a double fertilization. ठीक है? बताओ 100 सीड्स के लिए 125 कैसे? 125 मतलब you can see we are talking about meiosis. How many meiosis we needed? In the male part, okay, for each meiosis, you will get 4 microspores. 4 microspores. If you want 100 seeds, okay, if you want 100 seeds, then we need 100 pollens first. For 100 pollens, okay, if one give 4 microspore, your 25 meiosis will give 100 pollens. We need 100 pollens. Okay, through 25 meiosis. For 100 egg cell, for 100 egg cell, you need 100 meiosis. Since each meiosis will give only 1 megaspore, then 1 megaspore will convert into 1 embryo sac. Okay, you need 100 meiosis for 100 embryo sac and 100 egg cell formation. Okay, total 125 for 100 seed formation. ठीक है? कोई पूछा था ना? कौन पूछा था क्वेश्चन? मेडिकोस कौन सा नाम लिखा है आपका यूट्यूब में? ठीक है, गुड 40% एंजियोस्पर्म 3n स्टेज, राइट? समझ नहीं आ रहा है आपका क्वेश्चन? सो मोड्स ऑफ पॉलिनेशन, ठीक है, जल्दी थोड़ा अभी जल्दी करेंगे. ठीक है, मोड्स ऑफ पॉलिनेशन. रिप्रोडक्शन तो हो गया, सेक्सुअल, एसेक्सुअल, वेजिटेटिव, अपोमिक्सिस सब कुछ अभी पता चल गया. मोड्स ऑफ पॉलिनेशन. पॉलिनेशन इज द फिजिकल ट्रांसफर ऑफ पोलन ग्रेन फ्रॉम द एंथर टू स्टिग्मा. सिंपल. ओके, एंथर से स्टिग्मा के ऊपर जा रहा है आपका पोलन, वही उनको मतलब विल कॉल इट एज ए पॉलिनेशन. ओके, देयर विल बी ए डिफरेंट काइंड्स ऑफ पॉलिनेशन. सेल्फ पॉलिनेशन विल कॉल इट एज ऑटोगेमी, ट्रांसफर ऑफ पोलन विद इन द सेम फ्लावर. ठीक है, क्रॉस पॉलिनेशन, अलोगेमी. ओके, ट्रांसफर ऑफ पोलन फ्रॉम द एंथर ऑफ फ्रॉम पर्टिकुलर वन प्लांट टू स्टिग्मा ऑफ द डिफरेंट प्लांट, अनदर प्लांट. ठीक है, वो तो क्रॉस पॉलिनेशन हो गया, दैट इज अलोगेमी. व्हाट अबाउट गेटिनोगेमी? ट्रांसफर ऑफ पोलन from the flower to another flower of the same plant. In maize, okay, you can see in maize there will be a, if it is maize, there will be a tassel. Okay, and there will be a cob from tassel to cob. Okay, from the same plant, transfer of pollen from this flower of this particular male plant to female plant. Okay, उसको बोलते हैं हम गेटिनोगेमी. It is same as your self pollination, identical to your self pollination. Okay, that is getonogamy. Then xenogamy, true cross pollination between flowers of different plants. That is nothing but your cross pollination. Okay, that is we will call it as a xenogamy. Okay. Agree by 300, 3 intro 100, total 500 metasis. Mango for getonogamy, yes. Where there is a monoecious condition. Where there is a monoecious condition. Monoecious condition is nothing but the plant is having different male and female flowers. Okay, you can see the getonogamy. Okay, all monoecious comes and will having a mode of pollination, that is getonogamy. Okay. Then mechanism promoting self pollination. Which are the mechanisms which promote the self pollination? Homogamy. Example mirabilis, jalapant, potato. क्या homogamy मतलब? The flowers will mature in the same time. Okay, if it is they are maturing in the same time, then they will promote self pollination, right? That is your homogamy. Okay, what about cleistogamy? Flowers never opens at all. Okay, they will never opens in case of cleistogamy. Mom's air marathon record break today. Next, chasmogamy. Cleistogamy, they will never open at all. But chasmogamy, the flower will open, but after pollination. ठीक है, उसमें ओपनिंग तो होता है, पॉलिनेशन तो ऑलरेडी खत्म होता है, मतलब खत्म होने के बाद ओपन होता है. ठीक है, उसको बताते हैं चैस्मोगमी. कोई एग्जैक्टली मुझे एग्जांपल बताओ क्लिस्टोगमी और चैस्मोगमी के लिए. ठीक है, आप कमेंट में डाल दो, हम दूसरा भी डिस्कस करेंगे. हिडन स्टेमन एंड स्टिग्मा. Stamens and stigma are covered by the other floral parts. In case of your pulses, okay, there will be keel petal, then standard petal. Okay, there will be complete covering of your stamens and okay stigma. So obviously it will promote the self pollination. Okay, in legumes they are enclosed by keel petal. Example, all pulses, your pea, moong bean, urd bean, soya bean, gram. Okay, this is also promote self pollination. Then close position of anthers and stigma, mostly seen in tomato and brinjal. Okay, there will be a close attachment within the stigma and anthers, so that there will be a mechanism. So it will promote self pollination. मैं पूछा था examples पूछा था. Wheat cleistogamy, rice, wheat, chasmo, rice. Okay, your chasmogamy, that means flowers will open after pollination. Example, rice, wheat, barley. ठीक है. But when you comes to cleistogamy, flowers never opens at all. ठीक है. Wheat, oat, all not wheat. Okay, some varieties of wheat. Okay, and also you can take examples of pulses. Okay, the flowers never opens at all. Okay, some varieties of wheat and barley. Okay, which are examples for cleistogamy. Yes. Brinjal heterostyly. Heterostyly, okay, will promote self or cross pollination. कोई बताओ. Peanut is for cleistogamy. Yeah, pulses we can put it in cleistogamy, but not all wheat. Some varieties of wheat. ठीक है. Rice will chasmogamy. Heterostyly, बताओ कोई. Heterostyly will promote self or cross. Means in chasmogamy there is chance of cross pollination. That's what Nemani. Chasmogamy, rice, oats is good. Cross pollination in wheat percentage. अरे वो तो 2-3% से होता है बस. ठीक है, it is highly self-pollinated crop plant wheat. Heterostyly ना, मतलब visually brinjal heterostyly also, but that will comes under your, it will promote your cross pollination, right? Heterostyly is nothing but there will be a difference, okay, in the stamens positions, style position. Yeah. Then mechanism of promoting cross pollination, decline or unisexuality. ठीक है, In that particular condition, monoecious and dioecious. Okay, monoecious are nothing but your maize, for good example. Okay, in dioecious, date palm, female and male flowers in the same plant. Okay, monoecious. So there will be a different maturity. So there will be a possible chances of cross pollination. In case of dioecious also, there is complete cross pollination. In case of dioecious, there should be a crossing between different plants, since some of the plants will only having male flowers, some of the plants only having female flowers. Then dichogamy, like homogamy, there is same maturity, but dichogamy, protogyny and protoandry. ठीक है, कोई एग्जांपल लगा दो दोनों को, प्रोटोएंड्री और प्रोटोगाइनी को. या पपाया इज dioecious, गुड. पपाया, ठीक है. शुगर बीट, यस, दैट इज योर monoecious. Yes. Pearl millet, protogyny, good. Pearl millet, that is your bajra. For protoandry लगा दो. Yes, good. Maize. In protogyny, your female part, that is your gynoecium, will mature first. In protoandry, your male part, okay, that is your androecium, will mature first. ठीक है. Yes, maize, good. Then heterostyly, as someone told, brinjal. Okay, there is a difference in case of the position of stamen and stigma. So it will also promote the cross pollination. Ergogamy, in lucerne, there is an cellulose layer. Okay, there is a cellulose layer in the stigmatic surface. When the honeybees or any bees, okay, will come and will disrupt that particular layer, then only there is a pollination. ठीक है, वो तो क्रॉस पोलिनेशन प्रमोट करता है. When bees will come from, they will take pollen from another plant and bees will come here and they will disrupt that particular cellulose layer, then there is a pollination. Self pollination तो नहीं होता है easily, and your self incompatibility and male sterility. These are the two mechanisms we will discuss now after this. Okay, then we will wind up. Okay, till now आप रहना चाहिए. There will be again important concepts. Both are, they will definitely ask one of the two questions by self incompatibility and male sterility. ठीक है, these are the different mechanisms. Tripping mechanism, yes, for this we will call it as a tripping mechanism. Bees will come and they will disrupt that physical barrier. Yes, good. Alpha is nothing but your lucerne. ठीक है, और क्या बता रहे, कुछ बता रहे क्या, और क्या क्या क्या, प्रोटोएंड्री, yes, ठीक है, lucerne, yes, tripping mechanism, that's what we will call. And often cross pollinated crop plants, okay, where there is a cross pollination ranges from 5 to 30%. Okay, we will call them as a often cross pollinated. Genetic makeup is intermediate between both self and cross pollinated crop plants. Okay, मतलब completely or self are completely there. They wanted homozygous condition and they wanted heterozygous condition, cross pollinated crop species. But in often cross pollinated, there will be intermediate preference. Okay, your jowar, cotton, pigeon pea, safflower. Okay, these are often cross pollinated crop plants. Both self and cross pollination breeding methods can be used. Okay, again, hybrids are often superior. Why we need to study the modes of reproduction and the pollination? Okay, to choose which breeding method we want to use. ठीक है, अभी एक क्रॉस पोलिनेटेड क्रॉप है, आप उधर जाके मैं पेडिग्री करता हूँ, ठीक है, मैं बल्क ब्रीडिंग तो कर सकता है, ठीक है, पेडिग्री में सेल्फिंग होता है ना. इफ यू आर डूइंग सेल्फिंग, ओके, इन केस ऑफ क्रॉस पोलिनेटेड क्रॉप प्लांट्स, दिस विल बी इनब्रीडिंग डिप्रेशन बेटा. ठीक है, उधर आप जाके हम पेडिग्री नहीं कर सकते, प्योर लाइन नहीं कर सकते. ठीक है, क्रॉस पोलिनेटेड में हम पॉपुलेशन इंप्रूवमेंट करना है. ठीक है, वी शुड मेंटेन हेटेरोजाइगोसिटी. दैट्स व्हाई यू शुड नो, okay, the nature of reproduction, the nature of pollination. Okay, if it is self pollinated crop plant, okay, then there if you want to use some hybrid seed production, then there will be manual emasculation, there is complex. Okay, that's why you should know the basic modes of reproduction and pollination so that you can Based on that you can go for different breeding methods. ठीक है, पता चलेगा धीरे धीरे सब कुछ पता चलेगा. ठीक है. Determination of modes of reproduction. How you can determine? Okay, floral observation. Based on floral biology, you can easily say, okay, if there are no, okay, if there are no, by seeing chasmogamy, clistogamy, the flowers never opens at all, you can easily say na that is self-pollination. Okay, based on the floral observation, isolation test. Okay, if you grow that particular, for example, if date palm, okay, एक plant लेके date palm, you can grow somewhere else individually, isolation. There are no other date palms around it. ठीक है. अभी बताओ उसमें सीड सेट होता है क्या डेट पाम में? बताओ, कोई बताओ. आपका पपाया ले लो. You can see in papaya, ठीक है, when there is an fruit development, उसका आसपास ही कोई पपाया प्लांट रहेगा. ठीक है. If your papaya plant is isolated, okay, it is growing isolation, it will never set the seeds. Okay, fruit तो development हो जाएगा, seed set तो नहीं हो पाएगा. ठीक है, वही है आपका. You can easily test based on the isolation test. In case of date palm also, if there is a no seed set in the isolation, that is dioecious. Okay, there is need of cross pollination for that. ठीक है. Based on that, you can easily say whether it is cross pollinated, whether it is self pollinated. If it is setting the seeds, then it is self pollinated. Okay. Then inbreeding depression study. If there is an inbreeding depression when you are going for selfing, then you can easily tell that it is cross pollinated crop plants. ठीक है. Pigeon P often cross pollinated extended up to 70%. अरे विजयाली, ये तो जनरली 5 to 30%. ठीक है. If it is going 70%, then the remaining 30% should be self-pollinated. Okay, when they are talking about both self and cross-pollinated, then we can easily tell that is often cross-pollinated. Okay, पेपर में दिया था 70%. ठीक है, ranges from 5 to 30%. We are telling not exactly, we are telling this much percentage. It will ranges. Okay, no seed set in isolation. That's what. Okay, since it is cross-pollinated. Now, the amount of cross-pollination. If you want to tell the 30% in often cross-pollinated, 40% cross-pollination, how we can say the amount? The easy test. Okay, you will grow the recessive homozygous condition plants in the center, and around that the dominant homozygous. Okay, recessive homozygous, then around you will grow capital A. Okay, and your dominant, okay, condition will be yellow seeds. Okay, yellow seeds. Your recessive will be white, for example, or you can take green. Okay, green. You can see the green plants, the green seed color and yellow seed color in the surrounding. You will grow like this. Then there will be a crossing. There will be a somewhat cross pollination, since we are finding the amount of cross pollination. Since it is a cross pollinated crop plants, we need to know the amount of cross pollination. Then there is a cross pollination will occur. Then we will harvest the seeds from this particular plants. Okay, where after that you will see the seeds. Okay, you will test the seedlings. Okay, where there is a yellow seeds. Okay, out of the we are only harvested the seeds from this particular green plants. Okay, if you see the percentage of dominant seedlings, okay, where there is a cross pollination, there is a formation of capital A, small a, right? If it is self pollinated, only will get small a. But out of your 100 seeds, okay, if 20 seeds are giving this particular yellow seeds, since it is dominant, okay, that means 20% cross pollination. If you are 100 seeds, out of 100 seeds, all are small a, then you will get green color only seedlings. Okay, then it is 0% cross pollination. Based on this, we can easily identify the amount of cross pollination. ठीक है, ग्राउंडनट क्रिस्टोगेमी ऐसे मैं बताया था ना, pulses, your pulses, your groundnut also we can consider as a pulse. Okay, yes, it is oil seed, but we can push them under the pulse also. It is clistogamy, where you can see the keel petal. Okay, standard petal. ठीक है. What are the relevance of modes of reproduction? As I told, genetic constitution, homogeneous, heterogeneous. It will depends on your modes of reproduction. If it is self-pollination crop plants, they will love homozygosity. Okay, if your plant is cross-pollinated, they will love heterozygosity. They wanted to should be in the heterozygous condition. Your gene action in self-pollinated crop plants, you can see the additive gene action. In case of cross-pollinated crop plants, since they are in heterozygous condition, there is a dominance gene action. Ease of pollination control. If it is cross-pollinated crop plants, then there should be a more effort to maintain, to make the selfing, to maintain the inbreeds. Okay, and stability of varieties after release. In self-planted crop plants, those are very stable. Okay, you can easily release as a variety. But in case of cross-planted crop plants, okay, we will produce hybrids. Okay, again in cross-planted crop plants, there is an very difficulty to maintain that hybrid. ठीक है, वो heterozygosity रहता है, ठीक है, वो तो segregation होता रहता है. There is a complete difficulty. Method of germplasm conservation again. Okay, if it is cross-planted crop plants, then you can't go for field gene bank. There is a segregation, right? So at that point of time, you will go for some other exitive conservation, like you can store the seeds for 50 to 100 years. Okay, method of germplasm conservation also depends on modes of reproduction. Sir, was not something special going to happen on 14th June? अरे वो तो रेडी हो रहा है, आ जाएगा, पक्का आ जाएगा सुबह के पहले. What will be the F1 hybrid? What will be the F1 hybrid and gene action? अपलोड हो रहा है बाय वीडियो. What kind of video is this? अरे आपको ये कंसंट्रेट, इधर कंसंट्रेट करो. आपको तो, आपके लिए तो नहीं है वो वीडियो, ठीक है, वो तो आगे वाले जूनियर्स के लिए वीडियो, ठीक है, आप तो इधर कंसंट्रेट करना है. What will be the F1 hybrid and gene action? अरे भाई F1 hybrid is nothing but crossing between two different individuals. We will call it as a F1. Okay, if you are telling that is a hybrid, ठीक है, वो तो सुपीरियर होना चाहिए पेरेंट्स के सामने, ठीक है, नहीं होता बस F1 है. F1 is nothing but first filial generation. Wherever you will cross any different, not different, whenever you cross two different two plants, you will call them as a F1. But if you want to call it as a hybrid, then there should be some criteria. Okay, it should be better than both the parents or any parent. It should exploit heterosis. Okay, that then you will call it as a hybrid. Okay. Genetic feature of cultivated varieties. ये तो मैं ऑलरेडी बार बार बताया था आपको, PYQ में भी एक्सप्लेन किया था सही से, ठीक है. Your self-pollinated crop plants, okay, the sexual which are self-pollinated, homozygous and homogeneous, those are your pure lines. Okay, homozygous at individual gene level, okay, those are homozygous, capital A, capital A. Yeah, as an individual population level, all will contain capital A only, ठीक है, इसलिए हम बताते हैं उसको homogeneous, that is your pure lines. Homozygous, heterogeneous, okay, at particular gene level all are contain homogeneous, okay, AA or AA, like that. But at a population level, it's a mixture of capital AA and small a, ठीक है, that is homozygous, but at population level that is heterogeneous. Your multi-lines, old pure line varieties or mass selection varieties, these are heterogeneous, ठीक है. At individual plant level, all genes are homozygous condition, ठीक है, capital A, small a or capital BB, small bb, Okay, there will be different genes, right? They're all genes are in homozygous condition, but it is a mixture of different genes. Those are in heterogeneous. Okay, that's why your multi lines, your pure lines, mass selection lines, those are heterogeneous. Then cross-pollinated crop plants, heterozygous and homogeneous, single cross hybrid. Okay, the hybrid between any two parents is heterozygous and homogeneous. All plants are having same genotype. That is capital A, small a. That's why it is homogeneous at population level, and at individual level it is heterozygous condition. Heterozygous and heterogeneous. At individual level also heterozygous, population level also heterozygous. Okay, your double cross hybrids, three-way cross hybrids, synthetics, composites, those are mixtures. Okay, that's why those are heterozygous and heterogeneous. In asexual, heterozygous and homogeneous, that is your clones. Someone is asking, na, clones. But heterozygous and heterogeneous are old clones, are your land races. Okay, after the clone, after some generation, there will be mixture, mutations. So it will convert into heterogeneous from homogeneous material. ठीक है? या अभी हम आ गया सेल्फ इनकम्पैटिबिलिटी. ठीक है? 18 स्लाइड जा रहा है, बस 20 स्लाइड से ठीक है खत्म कर देंगे. 7 से शुरू किया था ना, 4 बज गए अभी. कैसे है जोश? जोश है क्या? जोश है क्या? बताओ बताओ जल्दी. बताओ ना, कमेंट में डाल दो. We will see self incompatibility and male sterility. Then we will wind up today's marathon. ठीक है? yes आई सर. बोलो हो रहा है सर जी. अरे ऐसे होना चाहिए ना, अभी नहीं हो तो पूरा रिवीजन कैसे कंप्लीट करेंगे? ठीक है? आई सर यस सर, वेरी आई सर, पूरा है. ठीक है चलेंगे. शेयर कर दो, सबको उठा दो, थोड़ा नींद से उठा दो, सबको शेयर कर दो. ठीक है, अब आपको भी जोश आएगा. ऐसे लगा दो, ऐसे फेंक दो सारे ग्रुप में. Why does inbreeding depression occurs in cross-pollinated crops only? Why not in self-pollinated crops? ठीक है, ये तो ऑलरेडी एक्सप्लेन किया था, हम फिर से हम एक्सप्लेन करेंगे. ठीक है, उसके पहले थोड़ा दोनों टॉपिक खत्म कर देंगे. एक्चुअली we can discuss this in our heterosis and inbreeding depression class, means in marathon, but now I will say in simple way. ठीक है, why does inbreeding depression occurs in cross-pollinated crop plants? आपको cross-pollinated crop plants का genetic constitution क्या है? हेटेरोजाइगस रहता है हमेशा. आपको self-pollinated crops homozygous रहता है. I am taking the single gene example, but in general, in normal case, it will be not like this. Okay, there will be a number of genes will be controlling your trait. ठीक है, or small a. ठीक है, what will happen if it got selfing, if it is entering into inbreeding? Okay, there will be a capital A, small a. From so many years, okay, this particular small a is masking by the dominant a. But when you are going for inbreeding, okay, inbreeding is nothing but you are manually doing it, okay, artificially. So this particular deleterious A allele will expose to the environment, so that it will try to, okay, interfere in the plant development. So there is a loss of vigor, okay, loss of vigor or maybe ultimate death of the plants if it is more dangerous. Okay, that's why you can see the inbreeding depression in cross-pollinated crop plants. But in case of self-pollinated crop plants, since they love the homozygosity, okay, in their evolution itself, okay, there is small a and small a will already exposed to the environment. Okay, if it is very dangerous, okay, it is already got eliminated from the population, okay, during the crop evolution. Okay, that's why if it is small a only, okay, it is not that much deleterious. it is already exposed. Okay, if it is reduced, it is eliminated. If it is not eliminated, then there is not a problem of that particular homozygous allele. Okay, the recessive allele. Okay, but in case of cross-nominated components, it is in the particular genotype from the so many years. Okay, it is maintaining by the heterozygous condition. Okay, it will expose if you go for inbreeding. Okay, that's why you will get a loss of vigor and fertility. Okay, today full syllabus test type of gene action in F1. अरे फिर से पूछ रहा है type of gene action in F1 hybrid. In F1 hybrid there is a dominant gene action. In F1 is nothing but heterozygous, right? How we can exploit additive gene action there? It is completely dominant gene action. ठीक है, कोई पूछा था, कौन पूछा था वो? राइता देवो भवा. ठीक है, कहाँ से आप? राइता देवो भवा, कहाँ से आप बताओ? Self incompatibility. What exactly the difference between self incompatibility and male sterility? Your self incompatibility will produce functional pollens, but in case of male sterility there is no functional pollens. इतना difference है दोनों में. ठीक है, it is genetic mechanism in flowering plants that prevents the self fertilization even when the plant produces perfectly functional male and female gametes. Okay, but there is some chemical reaction will happen that will leads to the incompatibility. But you will first what you can see the functional pollens only. But in case of male sterility there is non-functional pollens. ठीक है. Earlier record of self incompatibility was made by Cold-Ritter in the mid 18th century. Okay, while studying the Verbascum Phenoecium. The term self incompatibility was given by Stout. These are two very important. Okay, the earliest record will be from the Cold-Ritter, okay, in Verbascum, but the term is given by self incompatibility is by Stout. Classification, okay, based on the interaction between pollen grains and pistil, complementary and oppositional. In complementary system of self incompatibility, what will happen? Both pistil and pollen produces some specific substances, okay, that actively promote the pollen germination. Complementary, okay, what it will happen? There is a chemical will produce from pistil and pollen specific substance which will promote the germination of particular pollen. When it will happen? Okay, when there is an S1 in the pollen, sorry, in the pistil and S2 in the stigma. Okay, S1 allele in pollen and S2 in the stigma, then they will produce some chemical. Okay, so it will promote the germination. That's why we will call it as a complementary system. If it is both are S1, they are complementary. If S1 and S1, okay, since the both the specific substances will not match. Okay, when they will match? When there are different alleles. Okay, but in case of oppositional system of self incompatibility, when the pollen and pistil share the same SI alleles, that means S1 and S1, the pistil produces inhibitory substance here. Okay, there will be some inhibitory substance that stop the pollen germination or burst the pollen tubes. Okay, S1, then there is no. In case of S1 and S2, there is a chemical. Both chemical will produce. Okay, it will improve the germination. Okay, it will cause the germination of pollen and there will be a compatible. But here, in case of oppositional system, when they are both are S1, they will produce chemical, they will interact and they will inhibit. Okay, when there is an S1, S2 condition, okay, there is no that chemical, inhibitory chemical, so there is an, okay, there is an germination. The simple definition is like in complementary system, okay, in complementary system there is a substance, specific substance, okay, it will promote the germination compatibility. When in always in the both the cases when compatibility will occur, when there is an compatibility will occur when there is an different two alleles. ठीक है, different two alleles in case of your oppositional system also, when there is an two different alleles, you can see the compatibility. But here the chemical will produce, it will promote, but here the chemical will produce, it will inhibit. ठीक है, ये डिफरेंस आपको क्लासिफिकेशन का. ठीक है, कुछ बताना है क्या?
गैप ले लो थोड़ा.
आ जाओ आ जाओ.
61 students are here. Oh my god. हेलो गाइस, हेलो स्टूडेंट्स, कैसे लग रहा है प्लांट ब्रीडिंग का सेशन? प्लांट पैथोलॉजी से भी खतरनाक है क्या? एंड गुड टू सी यू, लाइक मोर नंबर ऑफ स्टूडेंट्स आर स्टिल इंटरेस्टेड इवन इट इज लाइक 11:30. 4 घंटे का मैराथन, शाबाश. कैसा लग रहा है? एक्सेलेंट. मेरा तो मेंटल हेल्थ, फिजिकल हेल्थ सब कुछ बर्बाद हो गया. प्लीज आपको तो अच्छा है. वॉज इज कमिंग. अच्छा, चलो बढ़िया है. अरे गाइस, ले क्या बोलूं मैं. आप लोगों का गुड है, सुपर है, मेरा तो खराब जा रहा है. मैं साला टीचर से वीडियो एडिटर बन गया, बैठ गया था पूरा वीडियो एडिट करने में. जो एडिटर था हमारा, उसका कुछ काम आ गया तो चला गया. मोर एनर्जी ड्रिंक गिव टू अभी सर. नाइस नाइस. तो मुझे तो लगता है कि आप लोग इसी में खत्म कर दोगे. प्लान ब्रीडिंग पढ़ने का जरूरत ही नहीं था सालों से. जो 6 महीने 7 महीने से पढ़े हो, लाइक whatever you have read from 6 to 7 months. So the way everything has been covered in very crisp manner, I feel like sir I am tired. Tired हो तो तुम भी एनर्जी ड्रिंक पी लो या खाना खा लो. इसने तो अभी खाना भी नहीं खाया अभी तक, आपके अभिषेक सर ने. तो मैं उसका वेट करके मैंने भी नहीं खाया. ओके, होप यू ऑल आर एंजॉयिंग इट. जैसा कि लाइक यू 63 स्टूडेंट्स को देख के लग रहा है कि आप एंजॉय कर रहे हो. यस, आप लोग तो खाना खा सकते हो ना. खाना खाते खाते तो देख सकते हो, तो खाना तो खा लो. ओके गाइस, तो नाउ वी विल कम टू सी हरी सर्स एडिटिंग स्किल्स टू. वो तो मैं देखो, बाल सारे जा रहे हैं मेरे, पता नहीं क्या होगा मेरे साथ. कल तक वेट करोगे ना सब लोग पेशेंट से. मैं तो एक्साइटेड आप लोगों के लिए ज्यादा हूँ, नेक्स्ट ईयर बैच के लिए कम हूँ. नेक्स्ट ईयर बैच वाले क्या बोलेंगे क्या नहीं बोलेंगे, उससे ज़्यादा मुझे ये इस चीज़ में क्यूरियोसिटी है कि आप लोग क्या कमेंट करोगे, आप लोग कैसे इसको अपलिफ्ट करोगे. तो आप लोगों से बहुत उम्मीद है, लाइक आई एम एक्सपेक्टिंग टू मच फ्रॉम ऑल ऑफ यू, स्पेशली फ्रॉम माय प्लांट साइंस स्टूडेंट्स. इतना तो एक्सपेक्ट करना है ना, कि मिनिमम इतना एक्सपेक्टेशन तो कर सकते हैं ना. ठीक है. यस यस, बिकॉज आई हैव टू ईट इन द मेस. अच्छा, कुछ जुगाड़ कर लो कल से कि मेस का, कि पार्सल मिल जाए, कुछ ऐसा हो जाए कि टाइमिंग. देखो, इफ वी डोंट स्टार्ट फ्रॉम 7 PM, ऑब्वियसली लाइक इट मे गो बियॉन्ड लाइक 2 AM. इतना भी नहीं करना है ना. तो 7 PM आई गेस लाइक टू द राइट टाइम टू डू इट. सो खाने का बीच का देख लो कि थोड़ा अरेंज कर लो, पार्सल खाते खाते सुन लो. Let the video come, then watch it. I will comment on my neighbor's phone also. Thank you so much. ठीक है, जिंदा हो भाई?
कौन? मैं तो जिंदा है.
अच्छा, अभी कितना
ठीक
स्लाइड्स
है.
बचा हुआ है तो बोलो तो सही उनको.
बता दिया ना, अभी बस 15 है.
अभी 15 स्लाइड्स है क्या? अच्छा, आज कितने चैप्टर्स खत्म हो रहे हैं?
आज तो 8 चैप्टर खत्म हुआ है.
अरे इन लोगों को देख के मुझे डर लग रहा है भाई. मैं कैसे कर पाऊंगा इतना मैराथन? मेरा तो मैराथन इतना नहीं था. लास्ट ईयर मैंने जितना क्लास लिया, हाईएस्ट मैंने क्लास लिया था 3.5 घंटे का, वो भी पानी पी के 5 मिनट में 2 मिनट में पानी पीता हूँ. पानी तो बहुत पीता हूँ मैं.
मैं तो एक ही शॉट में दे रहा है पूरा प्लांट ब्रीडिंग.
ओके,
सब छोड़ के बस क्लास सुनना है बस.
मैं तो इन लोगों को देख के डर लग रहा है मुझे. पारुल ने 4.5 घंटे तक खींच लिया. मुझे तो चक्कर आ जाएंगे, लाइक आई विल बी लाइक हैविंग डिजीनेस. इतना कर दूंगा मैं तो. थोड़ा प्रेशराइज फील कर रहा हूँ, लाइक आई एम फीलिंग प्रेशराइज नाउ बिकॉज ऑफ ऑल दिस मेंटर्स. ओनली थिंग कीपिंग अस एक्टिव. नाइस. आप तो ऐप में 3 घंटे ले लेते हो सर. अरे वो तो ऐप में तो ठीक है भाई. और जोश तो बहुत हाई है. आप लोगों से अभी बात कर थोड़ा जोश आया, वरना तो मैं पूरा मूड ऑफ ही था. फीलिंग गुड राइट नाउ. ओके गाइस, ठीक है, कंटिन्यू कर लो. जो 15 बचा हुआ है तो ख़त्म कर लो. सो विल मीट टुमारो. ठीक है,
कीप एनर्जी ड्रिंक तो 5-10 मंगा लिया था हमने। रेड बुल तो पीते नहीं है, तो जो है ऑरेंज से ही काम चलाना पड़ेगा।
Okay, this is about the complementary system and oppositional system of self incompatibility, the simple definition. ठीक है, उसके बाद और थोड़ा क्लासिफिकेशन है, इसलिए इधर देखो, इधर हेटेरोमॉर्फिक, होमोमॉर्फिक based on the flower morphology. Your heteromorphic will be self incompatibility associated with the difference between flower morphology. ठीक है, you can see in the flower morphology itself, distyly and tristyly. Styles and stamens are two types, short and long. In tristyly, short, medium and long, we'll see what exactly those are. And in case of homomorphic, means there is no change in the floral morphology. Okay, there is only change in the particular genetic level. Okay, sporophytic and gametophytic. Self incompatibility is governed by the genotype of the pollen producing plant. Okay, in gametophyte, self incompatibility is governed by the genetic constitution of the gametes itself. Okay, your pollen, the reaction, the incompatibility reaction will controlled by your pollen, not pollen producing plant. ठीक है, हम सारे डिस्कस करेंगे स्पोरोफाइटिक और गैमेटोफाइटिक सही से. जीन्स इन्वॉल्व बेस्ड ऑन द नंबर ऑफ जीन्स, मोनोएलिलिक, डाइएलिलिक, पॉलीएलिलिक. ठीक है, मतलब 2 जीन्स, सेवरल जीन्स, सिंगल जीन. ठीक है, साइट ऑफ एक्सप्रेशन, स्टिग्मेटिक, स्टाइलर और ओवरियन. बेस्ड ऑन दैट आल्सो वी कैन classify. Then pollen cytology. Okay, when there is an incompatible reaction, it is in binucleate stage or trinucleate stage. Based on that also we can make the classification. ठीक है, अरे कम हो रहा है क्या व्यूअर्स? अरे ये तो बहुत इंपॉर्टेंट है, मेल स्टेरिटी और सेल्फ इनकम्पैटिबिलिटी. ठीक है, क्वेश्चन तो पक्का आएगा. Heteromorphic system, distyly, we will see what exactly the distyly is. Okay, you can see there are two different types of flowers in case of your primula. Okay, pin and thrum. In case of pin flowers, you can see your style will be long and your stamens will be shorter in length. In case of thrum flower, okay, the stigma will be, means your stamen will be long and style will be short. ठीक है, reaction कैसे होता है? You can see pin and pin. Your pin will be smallest, your thrum will be capital S, smallest. Pin and pin both are SS and SS, smallest. Okay, there will be complete incompatibility. There is no crossing between pin and pin. Okay, thrum and thrum, again capital S, smallest. There is no crossing again incompatibility. When there is a thrum into pin, you can see capital S, smallest. Okay, you will get, if you cross capital S, smallest, what you will get? There is two gametes, capital S, smallest. Here only smallest. So there is a capital S, smallest and smallest. So one thrum and one pin. In pin and thrum, that cross also you can see smallest, capital S, smallest. You will get one thrum into one pin. Okay, that is your heteromorphic system, distyly. In heteromorphic system, tristyly, that is in Lathrium salicaria. Okay, that is your heteromorphic system. Mating is only successful between flowers of different styles of length. Okay, there will be a short, medium and long. In that case, compatible will be like between different phenotypes, long into medium, long and short, medium and short. In that case, there will be a compatibility. Where there is incompatibility, any cross between the same style. Okay, if it is medium and medium, long into long, short into short, okay, during that time there is an incompatible reaction. ठीक है, ये तो heteromorphic system में tristyly, Lathrium salicaria, there will be so many genes will be involved. Okay, there will be so many combination of genes. Okay, that is exact, means mostly it is not important for you guys. Okay, what it is important, which is the species, we can see the distyly and tristyly, and distyly is very important. Okay, and also in the, you can see there is a pin into thrum into pin, where there is a long into medium, medium into long. In that case only there is a compatibility. That is your heteromorphic system. Homomorphic system, we can, as I told, gametophytic and sporophytic. In gametophytic self incompatibility, it's the most widespread form of self incompatibility of the plant kingdom. Okay, you can see in Solanaceae, Rosaceae, Fabaceae families. Okay, in case of your tobacco, potato, okay, in those cases you can see this particular gametophytic self-incompatibility. Okay, first discovered by East and Mangelsdorff in 1925 while working with the Nicotiana. Okay, that is your tobacco. The incompatibility reaction of the pollen grains determined by its own applied S allele. Okay, for example, plant to S1, S2. ये तो mother plant है. ठीक है, from this plant there is a production of pollens. Okay, what are the types of pollens it will produce? S1 and S2 pollen. Okay, you can see the incompatibility reaction है ना, वो determined by its particular S1 or S2 allele, not in the particular mother plant. Okay, so if it is determined by the own applied S allele, that means, okay, that incompatibility substance रहता है ना, वो chemical, it is synthesized after the meiosis. Okay, in the after meiosis, when there is a production of pollen grains, after pollen grains production there is an chemical production. That's why it is completely determined by the particular allele. If it is S1 there is a chemical, if it is S2 there is a separate chemical. Okay, if it is producing that particular substance in the before meiosis, okay, then what will happen? Then it will be dependent on this particular S1, S2. Maybe if it is dominant, then all will be S1. If it is codominant, then there is a mixture of S1 and S2. There is no question about it. Okay, that's why here, since that particular substance is producing after the formation of meiosis, okay, it is completely dependent on the allele of that pollen grain. If it is S1, then S1 chemical, S2, then S2 chemical. There is no question of dominance here. Okay, here you can see in the pistil, that is in case of your style, okay, the S allele show codominance. Okay, meaning the style will reject any pollen grain that matches either of the two S alleles present in the diploid maternal tissue. Okay, your style will contain that stigma. Okay, it is in the your female parent. If it contains S1 we'll see in the next slide. Okay, then it will show the codominance. Okay, if it is S1 allele, S1 is coming, your S1 pollen, okay, S1 pollen is coming, then it will reject since it already contains S1. If it is S2 allele, your S2 allele will reject again since it is contain S2 also. We'll see in the next slide. What about the genetic control of your gametophytic self-incompatibility? Maybe it is monofactorial, single gene will control, bifactorial, 2 genes will control, polyfactorial, okay, maybe there is a more number of genes will control, okay, that is genetic control. We will see exactly what will happen in gametophytic self-incompatibility. Okay, in gametophytic self-incompatibility, as I told, the chemical or substance which will produce after the production of meiosis, then after the release of pollen grains. ठीक है, इसलिए वो तो उसको मतलब ये डिपेंड होता है पर्टिकुलर allele का, not on mother plant. It will not dependent on mother plant. ठीक है, you can see the genotype of plant S1, S2, S3, S4, for example. The genotype of pollen S1 and S2. So your reaction will be S1 and S2 completely. Okay, it will not dependent on this particular mother plant genotype. Here also S3, S4 completely the individual reactions. You can see when there is an S1 and S2, okay, if S1 falls, then no germination since it is already contain S1 allele in the style. If it is S2 pollen grain, then again there is a no reaction. S2 will match with S2 chemical, so there is an complete incompatible reaction. Okay, incompatible. You can see fully incompatible in this particular case. In the mating between S1, S2 and S1, S3. S1, so there is incompatible reaction. Okay, when both will match. Your S3, there is no S3 here, only S1, S2. Okay, S3 has their own chemical. So since there is not matching with any mother plant or any female allele, okay, it will grow. It is compatible. So it is like partially incompatible. Here you can see S3, S4 allele. It contains S3, S4 in the ovary or style. So both S3 and S4 will, कुछ mistake हुआ है क्या इसमें? Sorry, it is actually S1 S2. Here S1 S2, ठीक है? Alleles तो S3 S4 pollens both will germinate. Okay, fully compatible. You can see fully compatible. There is a fully compatible, fully incompatible, and partially incompatible. There are three different compatible reaction in case of your gametophytic self incompatibility. What about pollen? Okay, what about pollen will be the stigma will be like wet stigma, where your incompatibility reaction will occur. Since stigma is wet, there is no problem with the germination. Okay, they will germinate, but they will arrest in the pollen tube. Okay, maybe there is an burst of your pollen tube. Okay, or it will not germinate. If it is germinate also, then there is no growth of this particular pollen tube. Okay, in stylar region. Okay, where your incompatibility reaction will occur in this particular stylar region. Okay, your stigma is wet stigma. And what about mechanism of incompatibility? As I told, in the stylar region, incompatibility will occur. Maybe pollen tube burst. Okay, your germination will happen here. Okay, in case of gametophytic, germination will happen, but it will immature pollen burst, or it will your RNAs will degrade the particular pollen tube. Okay, in stylar region there will be an inhibition. Okay, how we can break down this gametophytic, particularly gametophytic self incompatibility? If they ask gametophytic self incompatibility, you can go for polyploidy. Okay, your sporophytic self incompatibility will contains more number of S alleles. Okay, and it is like controlling by more number of genes. In that particular case, you can't go for polyploidy. If it is polyploidy, will no use to break down the genetic gametophytic sporophytic self incompatibility. But in case of gametophytic, since it is controlling by single gene with some other loci, many loci, you can go for polyploidy. If you double the chromosome, okay, it can break the GSI due to the interaction of or competition between the different S alleles. Okay, for example, here it is your S1 S2. If you go for doubling, then S1 S2 S2 S2. So there will be competition between the alleles. So then we can break down the particular incompatibility. In case of pseudo self fertility, incompatibility can be temporarily suppressed by the treating buds or pollens with the X-ray or gamma rays. It is temporarily breakdown. Okay, your pseudo self fertility and self fertility alleles. Okay, pollens with the allele bypasses the self incompatibility system entirely. There will be a SF alleles. Okay, your S1 S2 are self incompatibility alleles, but your SF alleles are self fertility alleles. If those alleles are present in case of your crop plants, maybe you can break down the gametophytic self incompatibility. Yes, in gametophyte, polyploidy is the effective method to break down. But in case of sporophytic, it is nothing. It is not useful. Okay. And one more thing, as I told, there will be three groups here: fully incompatible, partially incompatible, and fully compatible. Okay, three groups होता है. And if you see, if you look into this particular slide, okay, monofactorial, bifactorial, and monofactorial sporophytic. Your bifactorial gametophyte, that means it is controlling by two genes, and your monofactorial sporophytic. These are like similar in the some points you can see here. Genes governing SI reaction. One, here also it is gametophyte only, but it is controlling by two genes, and your sporophytic will be one. Number of alleles, multiple alleles. Okay, here also multiple alleles, here also multiple alleles. No problem with that. Pollen grain. If you see the pollen grain, the SI reaction controlled by pollen genotype, the applied genotype. But here also pollen genotype, but there are two genes are controlling. But in case of sporophyte, the sporophyte genotype is nothing but the stigmatic genotype or stylar genotype, the mother plant genotype. Okay, it will controlling by the particular sporophyte, but here the pollen itself. Okay, that means your pollens will release in case of sporophytic also. That the chemical will develop na that in that pollen. Okay, it is decided by the sporophyte, that is S1, S2. Here S1 already pollens already in haploid condition. Your S1 will decide that chemical. Your S2 will separately that will decide that chemical of that particular pollen. But here in S1, S2 they will produce pollens. The mother plant, from that mother plant they will produce pollens. But you will get S1 and S2 allele. But this chemical na, the incompatibility substances, it will decide on this particular, okay, this particular mother plant. Okay, if it is there, is there any dominance in this case, okay, what about dominance? S1 is dominant over S2. So since your chemical will produce before meiosis only, maybe your alleles are S1, S2, but the chemical, okay, since it is dominance, S1 is dominant over S2, both are will act as a S1, S1. Okay, both will act as a S1, S1. That's why in case of sporophytic, okay, the reaction, okay, the self-incompatible reaction will controlled by the mother plant, that is sporophytic. But in case of these two, in case of gametophytic, it is controlled by pollen genotype. That's what the main point in this self-incompatibility. Okay, number of nuclei at the time of pollen shed, 2. Here 3 nuclei. That means your from vegetative nucleus there will be 2 nuclei, and from one tube nucleus. Okay, that is 3. This is also 3. As I told, this it is similar to the sporophytic. Pollen coating, simple. It is somewhat complex. Pollen germination, slow. Pollen germination will be very slow. If it is germination will be fast, but the reaction will be happen in the stigmatic surface itself. Okay, they will germinate. If they germinate, there will be fast germination. Otherwise, there is no germination at all. Okay, but in this case, they will germinate. All pollens will germinate, and the incompatible reaction will occurs in styler region. Okay. Exine perforation. Exine means the coating, outer coating of pollen. Imperforate or micro perforate. That means there is no holes in the pollen grains. Okay. But it is perforate. Both are perforate. That means there is a holes in the pollens. Okay. During hydration, it will help. Okay. Stigma, wet stigma, you should remember in case of gametophyte. That's why pollens will germinate, all pollens will germinate, but it will incompatible reaction will occur in styler region. But in case of bifactorial gametophytic and monofactorial sporophytic, in both the cases your stigma will be dry. These are very important. Okay, the site of inhibition, style. The site of inhibition will be in this style, as I already told, styler region. But in case of bifactorial gametophytic and monofactorial sporophytic, it will be in the stigma itself. Okay, in the stigma itself, it will decide whether that pollen is got germinated or not. If it is germinated, then there will be a fast germination. Otherwise, there is no germination at all. Okay, in the stigmatic surface only, it will decide. Effect of polyploidy, as I told, may be eliminated in case of monofactorial. But in case of your both gametophytic and sporophytic, these are unaffected. Means your polyploidy will be no effect. Okay, since there are no more number of genes and more number of alleles. Okay, sporophytic system, behavior of the pollen is governed by not by its own genotype. Okay, by the diploid genotype of the parent plant that producing it. It will be completely dependent on the genotype of the parent plant which is producing it. But in case of GSI, it is completely dependent on its own genotype. Okay, maybe it is S1 or S2. It is completely dependent on that particular genotype. But in this case, it is completely dependent on mother genotype. Okay, first reported by Hughes and Babcock in Crepis foetida, then Parthenium from in the Parthenium argentum from the Gerstal. The system is technically more complex, okay, compared to gametophytic. and we'll see this as an brassica, okay, and mustard family. That is your Brassicaceae family. Mustard and cabbage, उसमें सब में आपका sporophytic self incompatibility आएगा. ठीक है? You can see how it will happen. Since as I told, ठीक है, genotype of the plant will be S1, S2. Genotype of the plant S1, S3, S1, S4. There will be a different combination. You can see genotype of the pollen is S1 and S2, means there will be two different pollens. But you can see the incompatible reaction of the pollen. Okay, when there is an dominance, your S1 is dominance over S2, S2 is dominant over S3, S3 is dominant over S4. ठीक है, dominance reaction ऐसा मतलब we are taking like this. Okay, generally. What will happen here? As I told, your S1, S2. Since this reaction is already decided in the plant itself, before meiosis itself. So, yeah, your gametes will be S1, S2. But since there is a dominance, okay, both will act as a S1. You can see all S1, S1, S3. In S4, S4, again all S1. In S2, S3, your S2 is dominant over S3. So, all S2. Okay. उसका फेट है ना वो ऑलरेडी प्लांट में मतलब प्लांट से बाहर आने के पहले ही डिसाइड होता है उसका फेट वो क्या होना चाहिए उसका केमिकल. ठीक है, जीएसए में ऐसे नहीं होता है, वो रिलीज होने के बाद उसका फेट डिसाइड होता है. इफ इट इज S1 अलील देन S1, इफ इट इज S2 अलील देन S2. ठीक है, उधर डोमिनेंस का मतलब नाम ही नहीं आता है, उसका डोमिनेंट का रिलेशनशिप भी नहीं होता है उधर, इधर आता है डोमिनेंस का रिलेशनशिप. ठीक है, ये इसका रिएक्शन का वो केमिकल रहता है ना वो प्लांट से रिलीज होने के पहले ही डिसाइड होता है मदर प्लांट से. ठीक है, दैट्स व्हाई यू कैन सी द डिफरेंस. यू कैन सी द incompatibility reaction now. S1, S2 in your styler region. Okay, that is your female parent. And if you are crossing with S1, S2, you can see all there will be S1, S2, but all will react as S1 as S1, as I told, since there is a dominance. Okay, both will not germinate since it is already contains S1. Okay, means here also there is a dominance relationship. Here S1, S2 due to dominance S1 allele. Here there is an S1, S4 allele, but S1, S1 since dominance, so complete incompatibility. Here you can see S1, S2, so it will act as S1. Okay, when there is an dominance, and your alleles are S2, S3 here, but they will act as a S2 since there is a dominance among S2, S3. They will act as S2. Here S1, they will act as S1. So both will germinate. So complete incompatibility. Here there is a no question of partial incompatibility. ठीक है, बस वही आप याद करना है इधर. There is no partial incompatibility here. Complete incompatibility or complete incompatibility, अरे complete compatibility, बस दोनों रहता है. कुछ डाउट्स है क्या? एक्सप्लेन द फॉर्मेशन ऑफ कंपैटिबल जीनोटाइप. अरे वही है ना, आपका स्टाइलर रीजन में S1 है, S1, S2 है. ठीक है, व्हेन इट विल क्रॉस विथ S2, S3, since there is a dominance, okay, both the pollens will act like a S2. Here due to dominance your styler region is acting S1. Okay, since there is dominance, see here S1 and your pollens are acting as S2, so both will germinate here in this particular case. That's why it is completely compatible. ठीक है, there is no question of partial here. ठीक है, what about pollen structure? There is an layer, we will call it as a superficial, callose superficial, cellulose superficial layer in the pollen, and there will be a tryphin. Tryphin is nothing but some protein chemical in the exine of that particular pollen. What will happen? Your stigma will be papillate. papillate is nothing but some airy like structure, your stigma, which is dry. Stigma is dry, but it is papillate surface. And pellicle, it is again proteinaceous sheath in the stigma. Then there is a cuticle layer, waxy layer. Okay, what will happen? Your pollen will land on the stigma. In case of sporophytic self incompatibility, ठीक है, आपका स्टिग्मा तो ड्राई है, ठीक है, ड्राई स्टिग्मा. Your pollen will come and will land on the stigma. Since there is a dry stigma, there is no germination. In case of gametophytic self incompatibility, since it is wet stigma, so there is no other words. It will easily germinate. Okay, but it will be inhibition in the stylar region in case of GSI. But here what will happen? Since your stigma is dry, there is no need to be some reaction here. Okay, your pollen will land on the stigma. If it is incompatible, incompatible pollen means your pollen is incompatible. Okay, nothing will happen. Okay, there is no further process. There is no germination at all in the stigma itself. But if your styler is S1, it is producing S1 chemical. If it is your pollen is S2, then there is a compatibility between these two. What will happen? What will happen? It will match the both reactions. Then there is a this particular layer है ना, उस layer से आपका ट्राइफिन प्रोटीन है ना, ये बाहर आता है, ठीक है. It will ट्राइफिन protein will out of your pollen. Since your pollen is your pollen is, you can see here, in case of your sporophytic, your perforate or reticulate, that means it contains holes, so that your tryphin will flow out only when there is a compatible reaction. Your tryphin will flow out, then tryphin will fuse with this particular papillate surface, then this with the pellicle. Okay, it will flow out and your pollen will get hydrated. Okay, there is gel, it is like a gel like structure, tryphin. It will flow out and there is a hydration condition and there is a release of cellulase enzyme. Okay, cellulase enzyme. Those will destroy this waxy layer, cuticle layer. So your pollen will germinate. The simple phenomenon. Okay. In case of GSI, it is, since it is wet stigma, okay, it will easily germinate and inhibition will be in stylar region. But in case of this sporophytic, okay, based on the compatibility reaction, okay, there will be hydration occurs. If it is compatible, there will be hydration and there will be germination. If there is no compatibility, since it is dry stigma, there is no growth and there is no reaction. Okay, there is no germination at all. Simple thing. Your tryphin will be protein molecule in the pollen structure and pellicle, it is proteinaceous sheath in the stigma. Both will interact and they will release a cutinase enzyme, cellulase enzyme. It will destroy the cuticle layer and there is a germination. माधुरी खत्म करेंगे मेल स्टेरिलिटी बचाओ बस. Okay, we will see the difference between the gametophytic and sporophytic. Someone asking reciprocal class, that's we will see in the difference. You can see self incompatibility controlled by gametic constitution of pollen. Here genotype of sporophyte, genes involved, S gene with multiple alleles, S gene with multiple alleles, site of gene action, style. Okay, here pollen matlab in stigma itself. Okay, in stigma, site of gene action, type of gene action individual, here dominance or individual or interaction. In sporophytic, you can see dominance, codominance, everything. But in case of your gametophytic, there is no question about your dominance, right? Since it is controlled by particular S allele, single S allele, there is no dominance, codominance. Pollen structure binucleate, here trinucleate. Reciprocal differences you will not observe here. Okay, since your S1 allele, S2 allele, they are separate. Completely it is, if it is reciprocal class also, there is no difference at all. But in case of your sporophytic, okay, there is an reciprocal difference. Okay, if you change the female and male parents, then again definitely there is a changes in the compatible and incompatible reaction. Recovery of parents from the causes, only male parents. Here in case of your sporophytic, you can recover both male and female parents. Okay, production of homozygotes is not possible in case of your gametophytic. it is always, since the incompatible reaction is based on the pollen, okay, it is completely impossible. Here there is a possible of recovery of parents, production of homozygotes and parents also. Types of crosses: sterile, partial fertile and fully fertile. Here only either fertile or sterile. Effect of polyploidy is restored here. You can restore through the polyploidy. Here it is difficult. Production of chemicals associated with the self-incompatibility during pollen formation or after pollen formation. Here before pollen formation. Okay. Example: potato, tomato, olive, solanaceous. Okay. White clover, red clover. Here radish, cabbage, cauliflower. Those are Brassicaceae family. Molecular basis, this is not that much important for you guys, but if I want to tell, in sporophytic self-incompatibility in Brassicaceae, okay, your male determinant protein will be S locus protein. Okay. In female determinant protein, there is a SRK locus. These are two different proteins from the male side and female side. What will happen? Molecular mechanism: when there is an incompatibility reaction, your SLR binds to SRK, that is your SLR, your male chemical will bind to this particular SRK, that is your female chemical, okay, in this specific manner, haplotype specific manner, and initiate the phosphorylation cascade. Phosphorylation cascade is nothing but it will destroy, okay, that it will happen in the sigmatic surface, na, okay, it will destroy that particular chemical so that the typhine will not flow out, okay. So there is no hydration and complete incompatibility. Okay, it is not that much important, but you should know the how it will work in the sporophytic. In gametophytic self-incompatibility, this is very important and very simple also. Your male determinant is S locus protein, okay. Your female determinant is sRNAs. When there is an incompatible pollen will germinate and pollen tube, okay, your sRNAs will activate. Okay, your sRNA will enter all the pollens, okay, which are germinating, okay, and your RNase is nothing but it will degrade RNA, right? It will enter your female protein, will enter all the pollen which are germinating, but it will selectively degrades the RNA only in the incompatible pollen tubes. Okay, this is the mechanism. Okay, sRNase mechanism. This is very important in case of gametophyte. In case of sporophyte, this is phosphorylation cascade will happen. Okay, at the stigmatic level, it is in stigmatic level and it is in stylar level. That what you should know. Simple thing. Jada sochna mat. Okay, they will not ask this much deep molecular mechanism also. Then interaction between the alleles, S alleles. In case of your sporophytic, okay, in sporophytic self-incompatibility, there will be a complete dominance in both stigma and pollen. There will be different condition will happen. Okay, dominance in pollen and codominance in stigma, and type 3 dominance in stigma, codominance in pollen. There will be different possibility. Okay, codominance in both stigma and pollen in sporophytic self-incompatibility, since it will be decided on the both in the parent itself. Always there will be two alleles na in sporophytic. There will be interaction, but in case of gametophytic self-incompatibility, the reaction will be based on completely your haplotype. Your haplotype is nothing but your haploid pollen. So if it is only one allele, there is no word for the dominance relationship, right? So your gametophytic self-incompatibility, codominance of S allele occurs in the style. In the style, codominance. But in case of pollen, it is applied. So dominance in pollen does not apply. Okay, self-incompatible response depends only on the pollen genotype. Okay, codominance तो मतलब stylar region में तो दो alleles रहेगा हमेशा, इसलिए there will be codominance. But pollen, there is no question of dominance, codominance relationship in case of your pollen. In case of gametophytic self-incompatibility, okay, there will be some exception in Brassica oleracea, Brassica nepus. One allele may be dominant in the stigma but recessive in pollen. There will be different things. Okay, they will not again ask about these exceptions also. But what you should know, in case of gametophytic self-incompatibility, at this point of time, you should know that there is no question of any dominance relationship. Okay, relevance of self-incompatibility. What are the relevance? Use of self-incompatibility in hybrid seed production. Okay, if both are self-incompatible lines. Okay, the hybrid seeds which are formed in the both the lines, self-incompatibility line 1 into self-incompatibility line 2. If both are self-incompatible line, okay, the seeds will form in this particular line and seeds will form this in particular line. Both are hybrid seeds. Okay, one line is self-incompatible, another line is normal fertile line. Okay, the hybrid seeds which will form in the self-incompatible lines are only hybrid seeds. ठीक है, इसमें दोनों hybrid seeds है, but here the hybrid seeds only will form in self-incompatible line. Okay, in this fertile line, if they are setting the seeds, maybe they are like self-wood seeds also. Okay, maybe ये question पूछ सकता है, पूछा भी है in 2 or 3 papers. Crops where SI is used, gametophytic self-incompatibility limited use. Okay, in clover they are using self-incompatibility ना, ये तो सारे क्रॉप्स में उतना कमर्शियल तो नहीं है. ठीक है, मेल स्टेरिलिटी पूरा कमर्शियल है. ठीक है, sporophytic self-incompatibility widely used in brassica species, particularly in Japan. Okay, Solanaceae composite है, they are like mostly in they will use. Okay, temporary elimination, that means pseudo fertility. ठीक है, refers to a condition where self-incompatible plants temporarily become self-fertile. Okay, if they are self-incompatible, then how we can maintain? To maintain those self-incompatible line for next generation, okay, we need temporary suppression. If it is complete suppression, then it will convert into normal self-fertile plant. Okay, but we need just to self-it so that we can maintain. In that particular condition, we can go for temporary elimination of self-incompatibility. What are the methods? Bud pollination. Bud pollination is nothing but before it is got maturing, we will do the pollination. So we can do the temporary elimination. Surgical techniques, we will take the stigma out. Okay, in case of sporophytic self-incompatibility, your stigma is the site of reaction. If we remove that stigma, then we will apply direct pollen to the pollen tube, styla region, so it will germinate. We can temporarily eliminate. Then end of season pollination, during at the end of maturity stage, we can do the pollination. High temperature, more than 45 degrees Celsius. If we go for more than 45 degrees Celsius, if you use the pollination, then there is the germination will happen. Then increased CO2 concentration, high humidity, and salt NaCl spray. If you use the salt condition, then also there will be germination. So temporarily, these are the temporary techniques. Your irradiation through X-ray or gamma rays. Double pollination. Double pollination is very important. Okay, what exactly double pollination? We will use compatible pollen or incompatible pollen, दोनों. ठीक है, दोनों हम डालता है स्टिग्मा में उधर. Okay, there will be a possibility of germination. ठीक है, ये तो आपका डबल पॉलिनेशन है. ये तो सारे टेम्पररी पॉलिनेशन. ठीक है, मेल स्टेरिलिटी. अभी बस 8 स्लाइड बचा हुआ है. ठीक है, बताओ, अभी बताओ, कंटिन्यू करेंगे या नहीं? बस मेल स्टेरिलिटी. अभी सेल्फ इनकम्पैटिबिलिटी तो हो गया. ठीक है, बिटवीन जीएसआई एसएसआई सारे. Sir, shall we revise male sterility topic tomorrow? Sir, it's already overloaded. Okay, what about others? We will go with majority. Continue, sir. Okay. Since it is the mechanism, we can complete now, so that we will enter breeding methods tomorrow. बताओ, मतलब बाकी लोग बताओ. Finish it, sir. Finish it. Last topic. Yes, it is last topic. Male sterility. Nemani. Okay. What about male sterility? As I told, non-functional pollen grains. In case of self-incompatibility, there is a functional pollen grains, but there is a reaction. But in case of male sterility, non-functional pollen grains, it occurs nature sporadically, perhaps due to some mutation. Colorator again is reported male sterility in flowering plants. Classes: structural male sterility, male organs either missing or deformed or have turned into other plants. That is your structural male sterility. Maybe your anther is deformed. Okay, missing. There is no anther. In that case, you will get a structural male sterility. What about sporogenous male sterility? The production in the production line itself, the stamens look like a normal outside, but the internal tissue, okay, the pollen mother cell, there is an problem with that particular tissue. Pollen mother cell tissues are defective. That is your sporogenous. What about functional male sterility? Problem is the delivery. The male organs and the pollen grains are both developed, okay, and appear healthy, but the pollen is trapped or non-functional. Okay, it is trapped under the anther itself, or it is not releasing, or it is, if it is released also, there is a non-functional pollen. That is your functional male sterility. Okay, sporogenous, structural, okay, sporogenous and functional. It is generally classified into following 4 groups: GMS, CMS, CGMS, and chemically induced male sterility. GMS is nothing but it is controlling by nuclear genes. Okay, the normal GMS, the MS gene, okay, the small MS allele will control the sterility. Okay, and again, if the molecular mechanism about your sterility is nothing but there will be some genes, okay, in the nuclear genes or mitochondria or cytoplasm, wherever it may be. Okay, those genes are, okay, will interfere in the production of pollens. There will be different pathways in the production of pollens, indirectly or directly. Okay, in that particular any genes there is a mutation, then there is a non-functional pollens. Okay, that is the basis of your male sterility. Again, in case of genetic male sterility, as I told, your alleles are present in the nucleus. That is your normal GMS. Then again we will convert it into TGMS or PGMS. Those genes will respond with respect to temperature. And your, again, another one, that is your transgenic male sterility. This will also comes under your genetic male sterility, since your genes are present in the nucleus. Cytoplasmic male sterility, your genes are present in cytoplasm, that are in the mitochondria or cytoplasm, but majorly those are present in mitochondria. Then cytoplasmic genetic male sterility, there is an sterility genes in the mitochondria, but to restore that, there are restorer genes which are present in nuclear genes. Okay, that is your CGMS. Then chemically induced male sterility. Okay, you will use the chemicals to kill the pollens or to make the non-functional pollens. Genetic male sterility, as I told, system where the inability of produce the functional pollens controlled by the genes in the nucleus. Okay, it is almost always caused by the single recessive gene, that is MS. Where there is a dominant gene, there is one exception. कोई बताओ, comment में बताओ. हाँ, already लगा दिया. Safflower is the exceptional GMS for dominant gene for male sterility. Yes. ठीक है. how NaCl spray works, sir? When you will spray the particular salt in the, that too in the concentrated version, okay, like 50%, 60%, okay, NaCl solution, okay, that salt will, okay, promote that particular hydration condition. Okay, it will increase that humidity, or you can tell that the hydration, so that temporarily you can avoid the incompatibility. That is about related to incompatibility, guys. Someone is asking NaCl spray. Okay, it will make the pollen got germinated and hydrated. ठीक है, इसलिए वो NaCl spray और ऐसे वर्क होता है. ठीक है. Safflower. All other are small ms. Your small ms will be A line. You can use as a male sterile, completely sterile, and B line, that is your capital MS, small ms, that is your fertile. You can maintain small ms will always produce small ms allele. It will produce 2 types. Since it is fertile, since your capital MS is there, plant will be fertile. So there will be a small capital MS, small s, small ms. There will be 2 type of gametes. So there will be 2 types of plants: capital MS, small ms, and small ms in 1:1 ratio. So you just have to remove this particular small ms plants which are sterile. Sorry, you should remove this particular capital MS, small ms. You can only save these seeds so that you can maintain your sterility. Okay, 50% sterile plants, these are saved and used as A line for the next season. Fertile, 50% fertile, these are rogued out, removed from the field, so they don't interfere with the hybrid seed production. ठीक है? How we will produce hybrid seed? Okay, the simple thing, if you cross with small ms with normal capital MS, okay, your all hybrid seeds will be capital MS, small ms, so that you can give the seeds to farmers, then there will be seed set again. Okay, since it is fertile plant, F1, whenever you are giving seeds to the farmers, your F1 should be fertile. You should be think like that. Okay, if you give the F1 sterile plants, okay, for example, in case of rice, if you give the sterile plants, but it is hybrid, but it is sterile, okay, what will happen? There is no seed set. In case of your farmer's field, in the next generation, there is no F2 seeds. For farmers, seeds are the yield na for them. You should be very careful. Okay, sir, what is the primordial observation in hybrid seed production of rice? Primordial observation. ठीक है, मैं तो पता नहीं है क्या बोल रहा है तू. Primordial observation मैं तो नहीं आ रहा है दिमाग में, कल मतलब देख के बताता हूँ. ठीक है, Primordial observation. Then in genetic male sterility, as I told, first one is your TGMS. अरे भाई ज्यादा सोचना मत. ठीक है, TGMS PGMS में ज्यादा दिया है बुक्स में. ठीक है, ज्यादा सोचना मत. It is TGMS plants are male sterile at high temperature. ठीक है, it is based on temperature. Your genes तो न्यूक्लियर में है. ठीक है, वो they will respond to temperature. ठीक है, high temperature and fertile at low temperature. ठीक है, sterility is controlled by the again MS genes which are located in the nucleus itself. In case of rice, okay, this particular PA64S, okay, Pi64S, which is again from China, okay, this line will act as a TGMS line. Now in India also we are working on it. Okay, there will be a recently in 2 to 3 years there will be a hybrid based on the TGMS system. In case of rice, the research is going on. Sterility above 23.5 degrees Celsius. They will fertile in the below that 23.5 degrees Celsius. Simple. Okay, widely used female parent in the 2-line hybrid seed rice in the China. Okay, temperature sensitive stage. Okay, PMC formation to meiosis. That particular meiosis na pollen mother cell pollen production. This is sensitive to the temperature. So the day length also little effect when temperature is low. In case of PGMS, it is completely dependent on the day length. Okay, when the temperature is within a critical range. The temperature should be critical range 23 to 29 degrees Celsius. Long day, complete male sterility, more than 13 hours. Short day will be normal male fertility. Okay, if you want to maintain this particular line, okay, where if you give the long day, okay, complete sterility. If you decrease the day length, okay, the normal male fertility. The same line, okay, based on the temperature, you can use as male fertile and male sterile, and then and another line to make it hybrid. That's why it is 2-line system. Okay, your CMS, CGMS, that is 3-line system. You will use A and to maintain again 1 line, and to make it hybrid again another line. That is again 3-line system. Here there is no for maintenance, there is no need of another line. The same line will act as both fertile and sterile. You need only another line for the hybrid seed production. Okay, PGMS is 2-line system is used in the mainly in China again. Both PGMS TGMS are used in China. As I told now, we are using TGMS. Okay, to produce the hybrid in India also. Next will be transgenic. Okay, in transgenic male sterility, सिंचन बता रहा है प्राइमोडियल में ब्रीडर डिसेक्ट करता है राइस के फ्लावरिंग टाइम एंड सिंक्राइजेशन को. डिसेक्ट करता है मतलब डिसाइड करता है और डिसेक्ट करता है. ट्रांसजेनिक मेल स्टेरिलिटी, ठीक है, एवरीथिंग विल हैपन इन द टेपटम सेल्स. दिस इज द क्रॉस सेक्शन ऑफ योर एंथर. ठीक है, you can see there will be tapetum cells here, tapetum cells. Okay, in that particular cells you can see the your pollen mother cells. in particular tapetum cells. Normal tapetum development here you can see there is normal tapetum development. In male sterile plants, okay, what will happen? We will introduce Barnes gene. Okay, Barnes gene. In the name itself, it is having Barnes, that means RNase. Okay, if that particular gene is present, Barnes gene, okay, what it will do? It will kills the RNA in the particular tapetum cells. Then it will completely destroy that particular tapetum cells. If there is no tapetum cells, there is no pollen grains, non-functional, it is sterile. Now my question, if I ask you the question, okay, sir, why it will only destroy tapetum cells? Maybe it can be destroy your complete anther, complete flower, or complete leaf, stem, RNase. बारनेस तो आरएनएस प्रोडक्शन करता है, आरएनएस सब जगह में आरएनएस रहता है सेल्स में, पूरा प्लांट में. There will be a destruction of complete plant, right? But it will only kill tapetum cells. Why? बताओ, कोई बताओ जल्दी. TA29, good. TA29 will be the promoter. Okay, when we will also introduce this promoter, okay, it will only express in the tapetum cells. Okay, that Barnes gene only express in the tapetum cells. It will kill the tapetum cells. So you can see here your Barnes protein, that is your RNase protein. Okay, it will destruct the RNase in the tapetum. Your tapetum will be destroyed. There is no pollens, complete male sterility. In case of, you will use bar star, that is counterpart. Okay, TA29 Barna means Barnes, and we will also introduce bar star. What it will do? It will produce Barnes protein. It will also produce bar star protein. It is tapetal specific bar star. It will interact with this Barnes enzyme. It will make inactive. Then normal tapetum development. There is no functional Barnes now. Your bar star will counteract it. It is the normal phenomenon. Okay. Now how we will develop this particular line in case of our transgenic male sterility? We will include TA29 and Barnes gene, and another one CAMV35S, that is also means tapetum specific promoter with the bar gene. Okay, in the hemizygous condition. Okay, wherever you will introduce some event, okay, it will be first event will be hemizygous only. After that it will diploid, means after that it will homozygous. But in this case, since it is for male sterility, there is no selfing, so it will be always in hemizygous condition. Okay, then you will cross with normal line. Okay, normal isogenic lines, that is male fertile. When you will cross with this particular male fertile, you will get in 1:1 ratio male sterile and male fertile. Okay, what will happen? You can see from this strand, and it will from another allele. Okay, it is in diploid condition, it will produce this type of gamete. So you will get a particular plant, and another condition you will get from this particular another allele, and another allele will produce normal fertile plant. But you should discard this fertile plant for the maintenance. What will it do? It will do, you have introduced bar gene. What it will do? Where there is a bar gene, it will give an tolerance for the phosphotyricin herbicide. Okay, your bar gene will give resistance to phosphotyricin herbicide. Okay, when you will spray the phosphotyricin in the for mixture of this particular male sterile and male fertile plants, it will kill the male fertile plants. Since they don't have bar gene, we included bar gene with the Barnes gene. So where there is an sterility, there is an bar gene, so it will give tolerance or resistance to phosphotyricin chemical herbicide. So it will only kill this particular fertile plants. Okay, your bar gene will be using for the to screen fertile and sterile plants. Okay, now we are having, you are maintaining it, but how we will do the hybrid seed production? Okay, this is your hemizygous condition. Okay, it contains Barnes and bar. then you will cross this particular female parent with the male parent which is having bar star, okay, your male fertile line, that will restore it. You can see if we cross this, all your, it contains bar star, all your hybrid seeds are fertile. Why? How they will fertile? You can see one allele will come from this and another one will come from this. It contains bar star, so it will counterattack this barrenness gene, so you will get fertile. But it is hybrid, crossing between two things, two parents. In case of this, one will come this allele and another from this. Again, it is hybrid only. Both are hybrid in this case. Okay, by this you can produce the hybrid seed production. To maintain, bar gene is necessary. Okay, you will normally cross with some isogenic lines of this particular line. You will get in the 1:1 ratio. You will kill this fertile lines with the spray. So by using this particular line, you will cross with the bar star gene. Okay, the bar star should be in homozygous condition. In both the is there should be bar star, so that in both the cases it will restore the fertility. So your F1 will be fertile plants. Okay, your barnase and bar star genes from Bacillus amyloquafaciens, but your bar gene, okay, which is tolerant for phosphonothricin, from the Streptomyces hygroscopius. Okay, your barnase bar star from Bacillus amyloquafaciens, your bar gene from Streptomyces hygroscopius. ठीक है, ये तो आपका ट्रांसजेनिक मेल स्टेरिलिटी. नेक्स्ट इज योर साइटोप्लाज्मिक मेल स्टेरिलिटी. सिंपल, ठीक है. It is type of male sterility is controlled by the cytoplasm, mainly mitochondrial genome DNA. Mitochondrial DNA. You can see mitochondria will present in cytoplasm. Okay, sterility will be in the cytoplasm. Cytoplasm sterile. Nuclear gene is non-restorer in case of cytoplasmic male sterility. But when you will identify some restorer lines in the nucleus, then it will convert into CGMS. Simple. CMS to CGMS. Till you are not having restorer genes, we will call it as a CMS. But when you will identify restorer genes, then it will be converted into CGMS. Fertility bar gene. Fertility bar star gene. Okay. Hemizygous means only one set of genome. Hemizygous means only one copy of that particular barnase gene. Okay. When you will introduce some gene with the transgenic technology, okay, you will introduce to any one of the homologous chromosome. In another copy, there is no particular that copy, that particular transgene. When you will go for selfing, then you can get homozygous condition. But in this case, there is no possibility of selfing itself. This, since that is sterile plant, okay, it will be always in hemizygous condition. Okay. Sir, what should I remember about the barnase bar star system? Your barnase is responsible for male sterility, bar star is responsible for restoration. Okay, both are transgene, from we took it from bacteria, okay, for the hybrid seed production. उतना मतलब ज्यादा सोचना मत, ठीक है. Cytoplasmic male sterility and male fertile. When there is a fertile gene in the cytoplasm, it is fertile. Okay, you can see male sterile, okay, will cross with fertile, normal any fertile plant. Okay, you will get an cytoplasmic male sterility only. Since your sterility from cytoplasm, your female parent always transmit the cytoplasm, so your sterility will come to the next generation. Okay, there is no, your fertility will not, if it is using as a male, okay, only your nuclear genes will transfer, not your cytoplasm genes. That's why it will always pass in the female parent. Okay, this is the basic phenomenon. It is controlling my sterility genes. Okay, how we can transfer cytoplasmic male sterility to a new strain. This is your strain A. it is contains cytoplasmic male sterility. Okay, this is your strain B. What it will do, you will cross these two. Okay, F1, then again you will cross this with this one by doing repeated back crosses. Okay, what you are doing, the repeated back crosses are nothing but your sterility is transferring. Okay, I will agree, but since in every back crossing you are taking 50% genome of this particular strain B, like thus 50%, 75%, 87%, 90%, 99%, like that at the end of the day. Okay, after some BC6, BC7 generation, okay, your nuclear genes completely from the strain B and your sterility from the strain A. Okay, now it is the new version of your sterility, okay, of that strain B. Okay, this is the basic phenomena. That's what they told. This is the male sterile version of strain B. More than 99% of the nuclear genes from the strain B. Okay, simple. How you can means transfer by repeated back crossing. Then use and significance of CMS. CMS is used for hybrid seed production in ornamental crops. Where you can use this particular CMS. Okay, we will, if you want this hybrid seed, okay, of this CMS, what it will do, it is RR and sterile, RR sterile. You will cross with the RR. There is no restorer genes in case of CMS. You will cross with any fertile plants. What you will get? You will get an hybrid here. You will get an hybrid. Okay, RR. There is no restorers, so you will get an sterile. It is hybrid. Okay, your F1 is hybrid. You will cross with two different things, but it is sterile. Your F1 is sterile. Okay, where you can use this particular technique, particular CMS technology, where the economic part is not a seed. Okay, for example, ornaments. Okay, for example, your onion. In case of onion, your economic part will be bulb. You don't need seeds. In this case, seeds will not set in case of F1 since it is sterile. But you can use where there is an, your economic part is not a seed. In case of bulbs, in case of other ornamental crops, you should need you need a F1 vigor, but you don't need a F1 seed, F2 seeds. There you can use this particular technology. But where there is a seed is economic part, you need F2 seeds. If you need F2 seeds, then it should be fertile. Your F1 should be fertile. If you want to make it F1 is fertile, then you should need a restorer lines. Then you should go for CGMS. Okay, you can see here. It is not suitable where seed is economic product unless fertility restorer is available. Some CMS system are used commercially, example in chilies, where you don't need seed again. In case of chili, your economic part is chili, and in capsicum, your economic part is like not seed. But where there is a need of seed, like your rice, your maize, there you should go for CGMS. Okay? When restorer genes are identified, your CMS become automatically CGMS. It is believed that all CMS system are actually CGMS. Okay? All CMS are actually CGMS, but we are not able to identify restorer genes. That's why some are remaining as a CMS system. Okay? Yes, where vegetative part is economic value, where we don't need seed, we can use the CMS system. Two-line system. Yes. अरे अगली लॉगिंग वाला डाल रहा है एपिस्टैटिक, डुप्लीकेट एपिस्टैटिस, कॉम्प्लीमेंट्री एपिस्टैटिस, प्लांट ब्रीडिंग चल रहा है. ठीक है, you will see that later in genetics marathon or genetic classes we will discuss about it. ठीक है. Cytoplasmic genetic male sterility. Next is your CGMS, as I told, your CMS plus restorer genes. Simple चीज है. ठीक है. Restorer genes are nuclear genes, usually dominant, okay, and restore the fertility of CMS line. As I told, mutation is responsible. Mutation is causing from the some of the genes which are present in nucleus in case of GMS system. In case of CMS, which are presented in the mitochondria, your male sterility, due to some mutation which is happening. How your restorer genes will restore it? Your restorer genes will interact with that particular genes, okay, in open reading frame, or any other mutation, if it is there any mutations, it will counterpart those mutation of your restorer genes, so that it will restore the fertility. Okay, it will make the functional pollens. Oh, you can see this is male sterile line, capital smaller, and yes, your sterility in the mitochondria. This is male fertile line. Okay, when you will cross, you will get an male sterile line. Okay, but male sterile line, it is male fertile, your RR, the nuclear genes which are having restorer genes. When you will cross with these two, you will get capital R, small r. If it is maybe sterile or fertile, if it is capital R, then it is fertile only. Okay. If you cross your male sterile with the male fertile heterozygous condition, so there is a possibility of segregation again. Small r with capital R, it is male fertile, and small r with small r, it is male sterile. The different segregation. Okay, your capital R is present in the nuclear condition and it will restore the fertility. ठीक है, ये तो basic है CGMS में. Development of new restorer lines. How we can develop new restorer lines? Yes, in case of your CMS, it is simple repeated backcross. Okay, then you will get a new version. But in case of CGMS, there is somewhat complex. What it will do? The strain A, strain B. Your strain A is completely male sterile. You will cross with the restorer lines. Then you will get male fertile line. That is capital R, small r, and the sterile side. First, you should transfer to the sterile background. Your restorer genes, you should transfer to the sterile background. Okay, the sterility. But your genes are restorer, but the cytoplasm is sterile cytoplasm. Then you should cross with the male fertile non-restorer. That is your strain C. You should want to transfer this particular capital RR into strain C. First, you should cross with the sterile cytoplasm. Then you will cross with the particular strain C where you should want to transfer. Then you will get a segregation. Since your RR in heterozygous condition, homozygous condition, you will get an segregation. What it will do? You will discard the sterile lines, then male fertile lines. Okay, to discard the sterile lines, you should cross with this particular sterile lines. If there is no sterile cytoplasm, then it's very difficult to discard those smaller alleles. Okay, if it is fertile here in the cytoplasm, okay, if it is smaller R also, it is fertile line. Okay, and these are also fertile line. It is very difficult to discard those particular small RR condition. So that's why, okay, you will use the sterile cytoplasm in the background. That will come from the strain A. First you will cross with strain A, then you will try to transfer into the new strain C. Okay, then you can see here, you can discard easily due to sterility. Then these are male fertile. Then again you will cross with the strain C again. Now there will be repeated backcross. You will discard sterile and repeated backcross to the strain C. Like at the end of the some backcrosses, okay, 2, 4, 5, 6 backcrosses, okay, you will get capital R, small r in the sterile background. Then you will self it. After the selfing, you will anyway will discard this. There will be segregation. Capital R, small r, capital R. Then you will go for self pollination. Again, it will segregate your capital R, small r. Okay, will completely discard it. Only the capital R in the sterile background. This will not segregate further since it is containing capital R. Okay, will maintain it. Okay, you can like this. You will transfer this particular strain B to strain C. First, what you should remember here. First, you will transfer into the sterile background so that you can discard that small RR condition. Okay, if it is in the fertile background, you can't differentiate between small RR, okay, F, and your small r, F. Both are fertile only. Okay, you can't differentiate. But if you transfer into sterile background, okay, you can see in the sterile background, you can easily discard this. It is sterile. but it is fertile due to R gene. You can maintain it, and you can repeatedly backcross into the strain C, to which you are want to transfer that particular restorer. Okay, clear चीज है. Sir, you teach very well. ठीक है, इतना ही है क्या? मैं 5 घंटे से पढ़ा रहा है. Okay, 2 to 3 slides. Chemical hybridizing agents. आपका CMS तो हो गया. For one thing I should tell there. In CGMS, okay, ABR is the famous system. A line will be your RR, okay, sterile. Okay, your B line will be maintainer line. That is your RR, but it is fertile, so that you can maintain your particular line RR. And again S. This is your maintenance. This is A line. This is B line. This is isogenic lines, but only the fertility and sterility will be the difference. And you will get again A line. You can see again A line. Okay, your R line, you will cross with the capital R, capital R. Okay, it is your sterility or fertility. You will get hybrid, that is your F1 is capital R, small r, and sterility. Okay, this is your hybrid seed. This hybrid seed you will give to the farmers. Okay, farmer will grow. Okay, again the seeds will set. Since it is fertile, okay, again seeds will form. Okay, seeds will set in case of your farmer's field. Okay, since we have restored it from the capital R, seeds will set in the F1 plants. Those are your F2 seeds. Okay, we'll get F2 seeds. Then after that there will be segregation. If you grow that particular hybrid based on this particular system, if you grow on F3, F4, there will be segregation. So you can then you can see the sterility in the F3, F4 generation. Okay, that's why we are not responsible for that. Okay, as a breeder or as a company person, your F1 seed will be fertile and you will get good, since it is hybrid, you will get a good yield. But if you use those seeds again, and if you are complaining that there is no seed set, then we are not responsible. Okay, there will be segregation and there will be a RR condition, and sterile cytoplasm. So we can't do anything. Okay. Then chemical hybridizing agents. The many chemicals can suppress the male reproductive organs. Okay, you just here we are using chemicals. These were earlier called male gametocytes, male sterilants or pollen suppressants. But in 1985, McRae proposed the term chemical hybridizing agents to avoid the confusion. First report, Moore and Nair, okay, induced male sterility in maize using malic hydrazide. Malic hydrazide will be first chemical hybridizing agent used in the crop breeding, okay, plant breeding for the male sterility in the maize plant. Chemical hybridizing agents are nothing but you will spray the chemical, it will kill the pollen production. 50 लोग हैं मेरे साथ, 47 गुड. सब मतलब सारे कौन कौन मतलब ऑलरेडी भाग गए ना, वो एक बार फिर से कल सुबह यार, कल दोपहर, कल शाम कभी भी देख लो. What are the ideal CHS? Induce more than 90% of male sterility, be effective across most or all genotypes. It will be economical, easy to apply with a flexible timing also. Non-mutagenic. Okay, if you are using the chemical, it should only kill the pollens. If you are doing it is also doing mutation, then it is again problem. No persistent in the F1 seeds. In the F1 seeds, that chemical should be not present. Okay, there is no effect on that particular F1 seeds. Cause minimal or no reduction in the seed set. Preferably promote flower opening to enhance outclassing. If it is affecting the flower opening, then there is no use. Systematic action affecting the all plants. Okay, it should be uniformity, it should have some uniformity. Ideally require only one application and work as a seed treatment. Okay, if you are again want to apply again and again, then it is not an ideal. And these all characteristics, okay, will not fulfill all the times. That's why we are having some demerits regarding chemical hybridizing agents. There will be some residual effect, there will be some mutation effects. That's why we are not using it that much commercially. Arsenics, that is your Mg1, Mg2. Okay. In case of rice, we will use ethanol, ethyl, we will use in barley, oats, but important is mendak again. Okay, in the cotton, pearl millet, chilies, we will use. Okay, these are we will use in the commercial companies during the seed production. Okay, or hybrid seed production. And important is like your arsenics we will use in rice. Okay, Mg2 is extensively used in China, and ethanol in the barley and oats, and mendak in the cotton. And in case of malic hydrazide, again for the first chemical we used in maize, cotton, and ibex. Okay, in wheat. In wheat, as I told, the variety is ibex. Okay, which is ibex, the hybrid variety in the wheat which is developed from this particular chemical. ठीक है, उसको तो बारे में उसको छोड़ के तो इम्पोर्टेंट तो ज्यादा नहीं है. ठीक है, ये तो बहुत इम्पोर्टेंट है 3-4 केमिकल. The last slide, guys. ठीक है, the pistillate condition in case of castor. In castor, we will see the pistillate condition. Where we can see pistillate and staminate condition again in the monoecious crops. Okay, where there is an separate inflorescence. There you can see the pistillate condition. In monoecious crops like castor, cucurbits, okay, some mutants will produce only pistillate flowers. Pistillate means only female flowers. If it is producing only female flowers, that means it is male sterile, right? It is male sterile. We can use those pistillate lines effectively for the hybrid seed production. This condition is called pistillate condition, and such plants are known as pistillate lines. Pistillate lines are widely used for hybrid seed production, especially in the castor. There will be 3 types of condition: N-type, S-type, and NES type. Okay, simple. What you should remember: in N-type of pistillate, it is nothing but the simple GMS condition. The simple GMS condition. Small n, okay, will only give the pistillate lines. Only pistillate lines means male. There is no male inflorescence. It will completely act as female plant. In case of capital N, small n, it is like bisexual. It will produce both pistillate and staminate flowers. Okay, it is fertile, male fertile. Okay, pistillate is nothing but male sterile, only female flowers. When you will cross with these two, again it will produce in small n, capital N, small n in 1:1 ratio. Okay, same as your GMS condition. But in case of S type, this is sex reversal variants. Okay, the initial primary branches. In case of castor, there will be primary branches, then there will be secondary, tertiary branches. Okay, in primary branches, you can see the pistillate. Primary branches, pistillate condition. That means pistillate means male sterile, only female flowers. After that, secondary, tertiary branches, it will start to produce. Okay, it will the sex reversal will happen. That means it will try to be, okay, both male and female flowers will come. That is your male fertile. Okay, after primary, secondary branches will happen. This also we can use for hybrid seed production. This is NES system, NES type. It is nothing but your environmental sensitive. NES is environmental sensitive type. Less than 35 degree, more than 35 degree. Okay. In less than 35 degree, only pistillate. That means male sterile. In case of more than 35 degree, it is male fertile. That means both male and female flowers will come. Simple, it is as a TGMS or PGMS condition. Okay, environmental sensitive. Okay, again we can use this as a for hybrid seed production. Okay, when there is a low than 35 degree temperature, only female flowers will come. Okay, use those pistillate lines. You can sow the pistillate lines in the less than 35 degree temperature. They will act as a female flowers, only female flowers. Then you can use another lines, not pistillate lines, any other lines. Okay, it will not affect by the environment. Other lines you can use as a male, means any male plants. Okay, only pollens will come from that plant. There is no pollen in this particular pistillate lines. You can get a hybrid seed. Okay, this is the pistilate condition in case of caster. The app is tired. TGMS PGMS. अरे एक बार देखते हैं। फिर से आ गया कोर्स में। अभी प्रमोशन तो नहीं कर रहा है, ठीक है, ऑलरेडी मैं तो प्रमोशन करवा दिया है। आपको इतना मतलब इतना क्लास देख के आपको भी कुछ लेना हो तो अभी ले सकता है, ठीक है। अब देखो बस मैराथन में ऐसे कंटेंट मिल रहा है कि आपके अंदर कितना है, ठीक है, लाइव क्लास में कितना कवर करवा दिया है, ठीक है, मार्क टेस्ट में, पेड मार्क टेस्ट में कितना कंटेंट ऑलरेडी ये, ठीक है। अभी मैं मार्केटिंग तो नहीं करवाया, आपको इतना सुनने के बाद भी चाहिए तो हमारा पेड मतलब टेस्ट सीरीज और एनी मास्टर कोर्स, यू कैन buy it. Okay, this is all about today's session. We covered from up to self incompatibility. अरे मतलब छोड़ के मत जाना, कुछ बताना हो तो अभी बताओ कमेंट में। ये वीडियो अपलोड होने के बाद, ठीक है, कोई अभी लाइव में देख रहा है, कोई आप रिकॉर्डिंग में भी मतलब अपलोड होने के बाद भी देखते हैं ना, वो लोग सारे कमेंट में डालना है मैराथन का पार्ट 2 चाहिए नहीं चाहिए। And also I need feedback of this particular first part 1 marathon session. I will see each and every comments, then only we will move to the next marathon, that is part 2, completely for breeding methods, principles and methods of breeding methods in self-planted and cross-planted croplands. We will also see quantitative and some biometrical techniques in the simple means the exam point of view. ठीक है, बताओ अभी बताओ न्यू सर एन एंड एस टाइप अगेन सिंचन। वो एन एंड एस टाइप कुछ नहीं है। एन टाइप इज लाइक सेम एज योर जीएमएस सिस्टम, ठीक है। इट विल कंप्लीटली बेस्ड ऑन small n will give sterile, means your pistillate condition. Sterile is pistillate condition, there is no male flowers, that's why we will call it as a pistillate condition. Capital and small n will give the normal plants, fertile plants. Okay, when you will cross again you will get 1:1 ratio, that is your n type. In S type, okay, the primary branches will give pistillate condition. Your primary branches will male sterilate, only they will give pistillate condition. After secondary tertiary branches they will start to give normal male fertile plants, that's it. Sir, if farmers are given F2 hybrid seeds under CMS condition, they will segregate further. Then what is the benefit of giving them F2 seeds? Because in F2 RR nucleus, yes, cytoplasm. Arey bhai, in CMS there is no F2 seeds. In CGMS, okay, I am talking about. In CGMS, you will give F1 seeds. Those are capital and smaller. Those are hybrid again. Okay, you will give to farmers. What will happen? Since those are fertile, capital or smaller, you will get F2 seeds. What farmer will do? You will sell those F2 seeds. For example, in wheat and rice, they are giving good yield. There will be setting F2 seed set. Then those F2 seeds contains capital or smaller, capital or smaller condition. But we are not sowing those F2 seeds. What? You will sell the seeds. You will eat. Okay, the consumers will eat those seeds. Okay, there is no need of segregation, worried of that segregation. If you save that seeds and if you sow those seeds again, then obviously there will be segregation. Okay, there is sterility, then you will not get seed set in the next generation. Okay, that's why hybrid seeds you should purchase each and every year, na. Vidisha Ghosh, new record by Abhi sir. Drink tea, everyone, I have made it. Rocking star Abhishek sir, thank you. War 10 रुपए के फ्रूटी अभिषेक सर क्यूटी है इसने, क्या बोल रहा है यार। न्यू रिकॉर्ड थैंक यू बाय, थैंक यू सर, मच नीडेड, थैंक यू सर। जी बता रहा है उस सर जेनेटिक स्लिपेज, वो तो जेनेटिक्स में आएगा, जेनेटिक स्लिपेज, ठीक है, वो तो जेनेटिक्स में कवर करवा देंगे सिंचन। सर वी ऑल्सो वांट योर मैराथन, ठीक है। इस इधर तो आर्ट सारे लगा रहा है, ये अपलोड होने के बाद आता है ना कमेंट्स, वो जरूरी होता है, ठीक है। प्लीज सर, इट इज नेसेसरी फॉर लास्ट मोमेंट। ओके सर, ओके, थैंक यू सो मच, थैंक यू। और क्या आ रहा है? डोमिनेंट एपिस्टैटिस, अरे भाई ये सारे छोड़ दो ना, वो जेनेटिक्स में आएगा। रिकॉर्ड फॉर ऑल मैम सीरीज रिकॉर्ड 6 hours complete in 8 minutes। ठीक है भाई, ठीक है देन, गुड नाइट सर, एंजॉयड इट, थैंक यू सो मच। मैं ऑलरेडी बताया था, ठीक है। सारे कमेंट डाल दो, क्या चाहिए वो बताओ, फीडबैक बताओ, और क्या बताना हो तो कमेंट्स में सारे डाल दो। So CGMS is 3-line system, CMS is 2-line system, apomixis is 1-line system. Yes, exactly. In apomixis, once you fix the hybrid, then there is no need of maintainer, or there is no need of your again male parent also. If you once fix it, then it will be continuous, right? Apomixis is 1-line system, CMS is 2-line system, okay, and your CGMS. Your CMS is not 2-line system. To maintain it, you will use another line. Again, to produce hybrid, again you will use another male parent, right? Your TGMS, PGMS, those are your 2-line system. Good night, sir. Bye. Good night. Good night. Okay, good night then. Bye bye. Tata. We will see in the next marathon. I will see the comments from each and everyone. Good night.